milk and eggs 9%, root and tubers 19%, and 24% are the cereals. So these are the major contributors of food wastage. So as you know that these food wastage contain a variety of nutritional components. So we can utilize these waste into a variety of valuable compound production. So talking about linear and circular economy, sometimes ago we are talking about linear economy. So what is meant by linear economy? Linear economy means if you have something you can take and then you can make as per your requirement the product, then use that product, then dispose of, and this disposal ultimately leads toward the pollution. So this will ultimately pollute our environment. And as you know that all over the world, the climate change is a big problem. And every year we have to face so many challenges to overcome these climate mitigations. So in the next generation, we are talking about circular economy. So what is meant by circular economy? Circular economy means you have to recycle all these things. For example, if you have certain products, you can make, use, reuse, remake and recycle. You have to develop some sustainable development in every sector so that we can prevent pollution, we can prevent climate change, and these climate changes, they have ultimate effects on human lives as well as on agriculture. So we need to think about circular economy. So these are the modern terms which are being used for the next generation. So you can see in this picture, in the circular economy, what kind of food waste, dairy products, vegetable and fruit, seafood and meat, and when these materials, they are being thrown into the environment, they can cause so many kind of problems. So we have to valorize these wastage to variety of compounds like biofuels, bioactive compound, bioplastic compounds, and these can be recycled and you can make money by this one. What kind of food valorization could be added to our additional products? From all these kind of fruit and food wastage, we can produce bioenergy, we can produce enzymes, we can produce bioactive agents, we can produce some sort of acids, we can produce biopolymers, we can valorize these wastage into pigments. So I will talk about little bit in each segment. So we have conducted one study. You can see that we have utilized waste melon peels, and these melon peels, they have good potential to produce a variety of enzymes like cellulases, amylases, tannases, proteases, and variety of enzymes could be produced by the intervention of food wastage through microbial interventions. We can apply fungi, we can apply bacteria for their production. Another study we can conduct, we have used sweet lemon peels. I don't know in Algeria, do you have sweet lemon peels? Sweet lemon. I think you have sweet lemons. And we have also tried the rotten peach. All these food and food wastage could be valorized into some sort of enzymes. So this is one study which are published, we have conducted in previous years. You know, the citrus is the most consumed food products, and recently we have reviewed these things and we have published an article. In this, we can utilize orange peel, lemon peels, orange pulp, and we can produce a variety of enzymes like cellulases, zelanases, ligninases, lipases, proteases, and pectinases. So all these enzymes could be produced via these waste material by microbial conversion. So all these enzymes, they have a potential industrial applications. Another study we have published that is the valorization of agri-food byproducts for lactase enzymes, and you can see that we have used different food waste products, byproducts like cocoa bean shells, rice husks, coffee husks. All these materials, they have potential to produce because they contain a lot of cellulosic material, lignin material, and hemicellulose as well. So we can valorize these material by microbial through fungi, and we can produce a lot of enzymes. So in this study, we have reported lackase enzyme, which have potential for dye degradation, which is a sustainable approach to mitigate the pollution problem. So another study we have conducted in which we have used grapefruit peels. You know, all the fruit peels, they are the potential cellulosic materials, and we can, in this figure, you can see that we have tested banana peels, banana peduncles, mango peels, orange peels, pea peels, apple peels, grape peels, and mosambi peels. All these are the food byproducts. And you can see that the grapefruit peels, they have a good potential. And we have tested a variety of bacteria like Bacillus ligniformis, Bacillus amyloliquefaciens, and Rotella orthinolytica. So all these enzymes, all these strains, they have capability to produce a variety of enzymes which are being reported already in my research groups. So this is the first topic, that is about enzymes. So all these studies indicated that the fungi, the microbes, they have potential to produce a variety of compound like enzyme. So moving towards the next, these are the biopolymers. Biopolymers are the natural polymers produced by the cells of living organism. They consist of monomeric units that are covalently bonded to form the larger structures. So some microbes, they have capability to produce biopolymers. So these polymers are very crucial for various biological processes and functions, and they can be found in all living organisms, including plants, animals, and microorganisms. So I am giving you some examples. So what kind of biopolymers we can produce from food waste? Here's a list of biopolymers. For example, all sort of fruit and vegetables. They contain pectin, they contain lignin, they contain PHA and PLA. So we can extract these biopolymers from these materials. And if you talk about further processing of these materials, we can produce some chitosan from gelatin and from other materials. We can extract all these material by some sort of other processes, from extraction processes or by microbial valorization. So these biopolymers, which can be extracted from food wastage, is a good example. Here I will report alginic acid production via food waste. So what is alginic acid? Alginic acid occurs as a main cell wall constituent of brown macroalgae in the form of mixed salts of sodium, magnesium, and calcium ion. As you know that we have already utilized sodium alginate as immobilizing agent. We can utilize these compounds for immobilization purpose. So apart from these seaweeds, some alginates can be produced by microorganisms. So what kind of microorganism they can produce alginate? Like azotobacters and pseudomonas strains, they have capability to produce these alginic acid by converting food waste into a specific compounds. So we have utilized apple peels as a substrate, as a waste material. As you know that when we eat apples, some people, they peel off their peels, could be removed and just use pulp. So if the peels are being removed and dumped into the environment, it becomes a wasted problem. So we can convert these fruit peels into a variety of compounds like alginic acid. So we have conducted a recent study and we have used apple peels through solid state fermentation, and we have utilized some microbial strains. You can see that we have gotten 186 milligram per gram of yield of alginic acid via microbial conversion. And we have tested this by HPLC, and you can see in the picture the powder of alginic acid can be extracted and further utilized for various applications. Another biopolymer is polyhydroxybutyrate. So what is polyhydroxybutyrate and polyhydroxyalkanoates? All these are the bioplastics. As you know that bioplastics, plastic is making a big trouble in the environment. And we are dumping so many kind of plastic in the environment, and these plastics are very difficult to degrade by the environment. So we have to change we have to change plastic into bioplastic, or we need to convert into a paper products. So all these paper products and biopolymers, they can be degraded by a variety of microbes present in our environment. So these are the polyhydroxybutyrate could be produced by food waste valorization. Here again, we have utilized apple peels as a substrate, as a waste material, and we have converted this into polyhydroxybutyrate via Bacillus thuringiensis or Bacillus some other strains, which have capability to produce these biopolymers through fermentation, through sunlight fermentation, as well as through solid state fermentation. Because these products, they are produced as intracellular compounds. So, and sometimes they convert the waste material into valorizable compounds. Here you can see that we have optimized the different conditions via response surface methodology, and we have obtained a better yield, and we have optimized all various components, and you can see that we have tested via FTR analysis and HPLC. We can detect all these compounds through fermentation. Another biopolymer is levain. What is levain? Levain is naturally occurring fructan, and the homopolymer of fructose. Has been produced by a broad range of microorganisms as exopolysaccharides. So exopolysaccharide could have potential for further application, and these can be extracted from variety of microbes through biomass valorization. So in these microbes, these polymers contribute to the formation of EPS matrix that helps to microbial biofilm formation. So we are producing such kind of material. For example, in this study we have produced mango peels. I think in Algeria, mangoes are available, maybe. Mangoes, maybe, because mango, they are famous food in Pakistan as well. So our mangoes, they are also very famous. So we have published this study, and we have converted these mango peels to levain through microbial fermentation. You can see that we have utilized a variety of food waste material like Mangofera indica, Musca, Malus domestica, and Saccharum officinarum. So all these wastes, they have capability to produce levain, and we have optimized a variety of nutritional parameters, and we have detected the levain by FTR and HPLC as well. So this is the second part as a biopolymer which can be produced. The third part is some flavoring agent. We can produce some flavoring agents through microbial fermentation. For example, what are these flavoring fermentation? As you know that every food has its flavor and color. So if your food is very good flavor, good smell, you would love to eat these food. But if your food is not have a good flavor and have not good smell, maybe you don't want to eat that food. So the flavor is very important. So, but before, people, they are using artificial flavors. So why we need to move towards artificial flavor? Because we have to, all these artificial flavors, they are not good for health. We need to do some natural compound extracted from our food wastage. So we can produce flavoring agents, and these are the substances which are added to food or drinks to enhance or modify their taste, aroma, or texture. So all these compounds could be produced via microbial fermentation through biomass valorization. So we have published a recent study, production of biovanillin, that is obtained from banana peels. Banana peels could be converted into a biovanillin through valorization. And we have optimized a variety of process parameters like optimum pH, temperature. Here you can see that how much folic acid concentration is optimum for vanillin production, how much time is important, pH and temperature. And all these compounds obtained from microbial fermentation could be analyzed through our standard and produce local product through FTR as well as HPLC. Then we have conducted another study on biovanillin. In this we have utilized pomegranate peels. And when we eat pomegranate, we discard the peels. So why we are going to discard these peels? We can convert these because all these fruit waste, they contain folic acid, and this folic acid can be converted into a variety of compounds. So in this we have used Enterobacter, and we have converted pomegranate peels into biovanillin through microbial fermentation. So in here, again, we have tested 3 variety of substrate. The first one is punica, the second one is musca, the third one is citrus. So among all these tested food waste, pomegranate have good potential to produce biovanillin. So we have optimized further conditions to get maximum yield. And again, we have detected this biovanillin via HPLC as well as via FTR analysis. So these are the third component which I am going to talk about food waste valorization is the extraction of natural pigments. So we can extract natural pigment. As you know that pigmentation is very important, and for this pigmentation, mostly industries or some other people, they are using artificial colors to give different coloration. But as you know that in a variety of food, they have a variety of pigment. Some food they are green in color, some food they are yellow in color, and some foods they are red in color. So when we are wasting these foods, we can extract these compounds for other application. So if we classify these natural pigments, there are 2 kinds. Some are water soluble and some they are lipid soluble. If we talk about water soluble, these are anthocyanin, betalains and cyanidin. There are a variety of different compounds, and all these compounds they have a very good application as bioactive compound. So some natural pigments they are lipid solubles like carotenoids and chlorophyll. All these pigments they have very good application in human health as well. How we do extract all these pigments? What are the different methods which are being utilized for natural pigment extraction? We can apply a variety of methods like solvent extraction, microwave assisted extraction, ultrasound assisted extraction, accelerated solvent extraction, pulse electric field assisted extraction, supercritical fluid extraction, enzyme assisted extraction. So all these, and green solvent as well. So we are moving towards sustainable development. So when we work on these parameters, we must use some sustainable products, we must use some sustainable methods, some sustainable solvents. So the next is about green extraction. So when we talk about green extraction, that means every method, every solvent, every process you are using in your research have no implication on, have no environmental impact. So we need to care about our environment. If we are polluting our environment, they have ultimately a big challenge for us. So we need to mitigate these climatic conditions. So these are different extraction methods which are being utilized for the extraction of natural pigments. So why we need to do work on natural pigment? Because there are some proved bioactivities of these natural pigments. So what are these activities? For example, betalain extracted from sugar beet, anthocyanin, lycopene, chlorophyll, beta-carotenoids, all these compounds can be extracted from food wastage, and they have very good application on human health, just like antioxidant activities. Some compounds they have antimicrobial activities, they are immunity enhancers, they have anti-cancer activities, and they also have hypoglycemic activities, and they do act as scavengers. So that means when we waste it, a lot of food is being wasted. So we can extract these compounds from the wastage and we can utilize as a medicine or as a nutritional component for our further food applications. So how we tackle these things, how we manage these things, what are the different policies? So what are the different holistic solutions for food waste management? How we can tackle this one? So we have to do some awareness, we have to do some good application, good policies. For example, the first one is the strict implementation of Food and Hygiene Act and rules. When you are preparing your food, when the food is being served to the customer, to the consumer, it should be hygienically produced. Then, waste to value products, approach for recycling of domestic and kitchen waste. As I have mentioned in the earlier slide, a big wastage is being produced at our homes. So we need to educate our wives, our mothers, our sisters, why we are wasting a lot of food. So we have to implement this one. Then we can do anaerobic digestion for initial food waste processing in underdeveloped countries. You know, you have seen in my previous slide, Nigeria is the biggest country where the food waste is very high. So we need to develop, we need to make some policies, we need to have some process, some conversion, we can prevent this loss. Then waste to energy approach for tackling industrial food waste. The third sector is a major contributor for food loss is industry. A huge amount of food waste is being produced at industrial level. So industry should know about this, why we are wasting this, and we can convert this waste into a variety of compounds like energy, like bioactive compounds, like pigmentation, and so many other things. Then suppression of food waste from municipal solid waste in developing countries, and food waste sources reduction during domestic and kitchen waste management. So all these are the some applications, some holistic approaches where we can manage the food waste loss. So talking about conclusion, what are the major sources? If we talk about the major sources of food wastage, these came from plant-derived, animal-derived, food industry-derived, and restaurant-derived. Everybody is going to a restaurant, and you can imagine, you can see, when you finish your meal, how much food is being wasted at your table. You can guess, you can imagine. And the restaurant, what they are doing there? Just trying, nothing else. So we have to manage all these things. And what are the bigger challenges and prospects? They are the big sources of nutrients, and this is a major global concern. And this also is ignorance by stakeholders. These are the source of contaminants, and improper management leads to the food loss. How we can tackle this? What are the sustainable options? The major sustainable options for food waste valorization is number one, sustainable valorization, integrated remediation, circular economy, and bioconversion. So these techniques can help to reduce food loss. So this is a major concern for us, for all over the world, for everyone. As you know that after COVID era, the climate is abruptly changed, and our pollution level is becoming more and more high due to ignorance, due to less awareness. So we need to tackle all these things wisely. So what is the take-home message? You can read the take-home message. Let us strive to be a better people and not waste our food. So we can also speak about food waste is cash in the trash. So these are the take-home message. This is my research group, my PhD student, my collaborators, my students. I have a good collaboration in Brazil and some other countries as well. So thank you very much for your attention. If you have any question, you are welcome. Thank you.
Thank you, Dr. Irfan, for your presentation. Now we'll have a second plenary lecture delivered online by the Professor Mary-Lise Rose-Hill. I don't know if she can hear us.
Yes, I can. Can you hear me?
Yes. I will just take a moment to introduce you, and then you can start, okay? Is it okay?
Yes, perfect. Thank you.
Okay. So, Dr. Mary-Lise Rose-Hill is a senior researcher in biotechnology at the Cape Peninsula University of Technology, South Africa. She holds a PhD in molecular and cell biology from the University of Cape Town and has an extensive background in biochemistry, molecular biology, and microbiology. Her research focuses on anti-actinobacteria, particularly on the discovery of novel species, oxidative enzymes such as lactase and peroxidase, and bioactive secondary metabolites with biotechnological potential. Over the years, she has led multiple national and international research projects, including collaborations with Germany, exploring actinobacterial biodiversity in terrestrial and marine environments. Dr. Rose Hill has also contributed to academic leadership at CPUT, serving on various research and ethics committees, and has supervised numerous local and international postgraduate students. Today, she will join us to present her lecture titled From Agricultural Waste to Products, Biofuels, Bioplastics, and Clean Water. Dr. Mary-Lise, the floor is yours.
Thank you very much. Good morning, everyone. Thank you to the organizers for the invitation to present at the first International Conference on Food Security and Sustainable Agri-Food Technologies. For those who know me, I usually talk about bacteria and how we can explore them as bioresources. I will focus on this a little bit, but today I'm going to focus on some of the research our institute have been involved in over the past 10 to 15 years, where the focus has been on bioremediation and beneficiation of agricultural wastes. So first, I will provide some context to the agricultural sector.
Dr. Mary-Lise, please excuse me. There's a technical issue. We can't see your presentation. Can you wait, please, a few minutes?
Sure.
Dr. Mary-Lise, it's okay. We can see your presentation.
Okay, perfect. Okay, so as I mentioned, I will provide some context to the agricultural sector of South Africa. The South African agriculture is quite diverse, as you can see from this image. This is mainly due to our different climates that we have, which ranges from subtropical to Mediterranean. And this allows for the cultivation of a wide range of products, including wheat, maize, and sugarcane, which is mostly around the northern and eastern parts of the country. We have oil seeds such as sunflower and canola, the citrus fruit, grapes, and various deciduous fruits such as peaches, pears, apples, and plums, just to name a few. And we also have some tropical fruits as well, such as pineapples, bananas, and lychees. And then we have a growing olive industry, as well as our own indigenous teas called rooibos and honeybush. The agricultural sector contributes significantly, obviously, then to the economy of the country, not only through exports, but also through implementation of a circular economy approach across the sector in collaboration with other industries. But one of the main challenges that is faced by the industry is water scarcity. This is then basically require careful management and innovative irrigation practices, as well as an increasing amount of solid waste that is being generated is also a problem, which typically then ends up on landfill at a great cost to the industry. One of our first projects started nearly 20 years ago. It was aimed at addressing the problem related to finding innovative ways to produce clean water from winery wastewater for irrigation. The South African wine industry is one of the top 10 wine producers globally, with the majority of the farms being located in the Western Cape province of South Africa. So you can imagine that with such a large amount of wine being produced, there is a huge problem with winery wastewater. This is a basic overview of the winemaking process. Other steps can also be included, such as clarification and blending. But during this process, you have that the crushing and fermentation vats have to be washed out before it can be reused. And they usually use potable or drinking water, which then result in a wastewater stream that is quite high in organic content, such as ethanol, acetic acid, sugars, and phenolic compounds. And it often contains inorganic salts, such as potassium, sodium, calcium, and magnesium, as well as other reagents that are used for the vat washing. So because of the variation in the use of different types of grapes and the seasonality of the process, the chemical oxygen demand, or COD, is highly variable, ranging from 500 to 4,000 milligrams per liter COD. The solid waste which is generated is called must, can feed into composting, which is typically used on the wine farms, or it is utilized by other companies for extraction of tartaric acid. So this solid waste is actually very well treated. The wastewater, however, can be a problem, especially for smaller cellars, where they have insufficient space for wastewater treatment infrastructure. So the winery effluent at the small cellars are typically used to irrigate fields or pastures, but as you can imagine, using this effluent for water for irrigation causes additional problems. The high level of organics causes anaerobic soils. It is toxic to both plants and microbial communities in the environment, while the inorganic compounds that are present causes the development of sodic swells, where an excess of the exchangeable sodium negatively affects water infiltration and aeration in the soil. So then further downstream, you also have the problem that the effluent can also cause eutrophication of other aquatic systems, which then leads to algal blooms and a toxic environment for vertebrates and invertebrates. There's therefore a need for a simple yet affordable system to treat the winery effluent from small cellars. One way to address this is to ask how nature achieves this. So in collaboration with Biomimicry SA, we approach the problem by asking the relevant questions. So what is the problem? How does nature solve this particular problem? Are there any examples in nature? And can we find the process in nature that achieve reproducible success? Can we then develop a solution based on this natural model? And then does our design emulate nature? In other words, can it adapt and evolve? Is it conducive to life? And is it sustainable? So then, taking these questions into consideration, our team visited various small vineyards in the Western Cape province to see how farmers deal with winery effluent and if a successful process in nature can be found. So on one of the farms, which is called Nietvoerbij, had a natural wetland located next to their processing facility. The winery effluent entered the wetland system as a stream high in organics and inorganics, but showed a reduced levels once through the wetland system, suggesting a natural bioremediation process taking place. But now, unfortunately, not all small vineyards have wetlands on their properties. So how can we mimic this successfully? Based on this observation, we started with setting up biological sand filters using locally available sand to try and mimic the natural wetland system. During this process, the effect of sand physicochemistry, as well as the effect of microbial communities on winery effluent treatment, was monitored. This then reflected both the biotic and abiotic processes that takes place during bioremediation. We also monitored whether organics and inorganics and salts were removed, and we monitored this all by using HPLC and a spectroquant. This then ultimately allowed us to select the optimal sand type and mixture, the load, and the optimal retention time to ensure the removal of harmful substances. The removal efficiency was monitored at the different levels of the particular biological sand filter, so at the inlet and the outlet, as well as the top, middle, and the deep sediments in the biological sand filter to make sure, or so that we can understand what is actually happening within the whole system. The key points highlighted for a successful system included the need to acclimatize the microbial population to the winery wastewater. It was typically a 2-week exposure period that is required. You also have to understand the role of the hydraulic flow and redox status within the system, and the use of a suitable substrate or sand, and then obviously developing a system that not only remove organics, but also salts. Here are some of the examples of results obtained while monitoring the functionality of the biological sand filters. In the first figure, the link between redox status and removal of COD at the different locations, such as the outlets and inlets in the system, can be seen. Within 24 hours of a high COD influent, more than 70% was removed, while in the second image, insights into potential role of microbial communities could be monitored by evaluating the effect of high COD effluent on these communities at the different sites of the system, with the top plot indicating the communities before and the bottom plot after exposure. How the communities are affected can then provide guidance to the development of more resilient communities that acclimatize to a high and variable COD. So ultimately, the work over the years led to the development of a treatment system that could be implemented. This final treatment design, consisting of a dual sand filtration system, was implemented on a small vineyard called Neertlingshof, and it is currently still in place and it's functioning. Some of the design highlights are that it's quite simple to install. As you can see, it's very few components that are required. It's very simple to operate and maintain, and all of the components were available off the shelf, and a locally available sand resulted in ideal flow characteristics, and no energy was required for the operation of the system since it's entirely run on solar-powered pump and gravity. The system was also monitored for a 2-year period, going through 2 different crush seasons. The specific highlights include the fact that the removal of the organics made the treated effluent compliant with legislative standards and could be used for irrigation. The system also adapted quite well to a variable influent, so whether or not white grapes or red grapes were crushed. And the removal performance improved over time. There was no sign of clogging, and there was an effective salt removal that took place, and this was all thanks to the abiotic processes that happened within the treatment system. The work presented here, I mean, it's just in a few slides here. It actually required extensive collaborations, bringing together microbial ecologists, biotechnologists, engineers, and the wine farming community to produce this final product for the generation of clean water. Here are just some of the selected outputs that's linked to the study and contains more details on the various approaches taken in this project. I would be happy to share this information with anyone interested. Moving on to the next example. Besides the processing of grapes, in the Western Cape province also have factories involved in the processing of apples and potatoes. So apples are processed into a wide range of products, but the factories that this study focused on was involved in the making of apple juice, while the potato factory was involved in the production of potato chips and other potato-based products for restaurants and the general consumer. Waste that is generated from fruit or vegetable processing has great potential for beneficiation. So many of these are quite high in cellulose and starch and sugars. And as such, when we performed a literature survey, it was clear that there are many different paths that can be followed to obtain value-added products from agri-industrial wastes, an overview of which can be seen in this diagram. With limited resources, however, it is impossible to focus on all potential avenues towards beneficiation. In this study, we therefore focused on the 3 wastes mentioned, which is apple, grape, and potato, and exploring 2 potential avenues, the production of enzymes and the production of bioethanol. So for this study, we focused on the fate of solid waste that is generated during the processing. For the potato processing, 3 sets of solid waste is generated after the removal of the skins, after cutting, and after the sorting for the final quality check prior to packaging. This waste either gets disposed of on landfill or it is sent to other farms for animal feed. During apple processing for the preparation of apple juice, the solid waste that is generated is referred to as apple pomace, and this in turn consists of apple skins, which is quite rich in pectin and lignocellulose, apple pips, and apple stalks, which are also lignocellulose rich. Since the aim of this study was to determine the potential use of the different wastes in the production of enzymes, we first had to evaluate the key properties of the waste. So if there are any phenolics present, and that could potentially hinder the growth of our bacterial strains or fungal strains, and the presence of sugars, which can serve as a carbon source for growth. So the high phenolic content of the grape waste made it an unsuitable resource, while the apple and potato waste, with their low phenolics and high sugar content, being more optimal substrates. 3 different teams worked on this particular project. At the Rhodes University in South Africa, the aim was to look at the application of lignocellulose degrading enzymes that was produced by recombinant Aspergillus niger strains cultivated on the waste and tested in the study, and then basically looking at whether or not they can use it for the release of reducing sugars from lignocellulose rich substrates. At Stellenbosch University, the aim was to look at whether the waste can be used for bioethanol production, while our team at the CPUT evaluated the waste as potential sources for the production of lacase from Trametes pubescens and the application of the lacase then in biocatalysis. So the 3 recombinant Aspergillus niger strains developed by the team at Stellenbosch University and a control strain was used in the study, and they're all listed in the first table on the slide. The strains were cultivated in a fungal growth medium containing various concentrations of apple pomace waste and potato processing waste. Optimization studies included determining the optimal temperature, pH, and waste load for the production of the enzymes endoglucanase, mannanase, and endoxylenase. Optimal production was observed in a 20% potato waste medium, even when potato wastewater instead of distilled water was used in the preparation of the growth medium. Similar levels of enzyme production was observed, as highlighted on this slide. These enzymes were then purified and applied in synergy studies for the degradation of model lignocellulose substrates. So synergy studies were performed at the Rhodes University. The enzymes were combined in different ratios to determine the optimal conditions for the release of reducing sugars from model substrates, such as beechwood xylan. And the highest degree of synergy was observed for the mannanase-xylanase combination, resulting in more than 2 milligrams per mole of reducing sugars released within a 30-minute incubation period. Synergy was also observed for the endoglucanase and mannanase when a substrate mixture was used of carboxymethylcellulose, locust bean gum, and beechwood xylan, which is a bit more complex. But again, it resulted in release of approximately 2 milligrams per mole of reducing sugars, making these the production of these enzymes on the waste streams quite suitable for subsequent application. The commercially available bioethanol producing yeast strain, ethanol red, was used or cultivated in the presence of various apple and potato waste by the team at Stellenbosch University. The optimal release of ethanol occurred after 70 hours of fermentation in a growth medium prepared from a starch-rich wastewater, reaching nearly 100 grams per liter of ethanol production, while a level of 20 grams per liter was obtained when a 20% solid potato waste was used. Calculating the theoretical yield based on the dry weight of substrate used, it showed that 200 grams per liter of potato solids yield an approximate amount of 30.6 grams per liter of ethanol, making potato waste a very good potential waste stream for bioethanol production. The fungal strain, Trametes pubescens, was cultivated in the presence of the 3 waste streams tested in the study by our team at the CPUT. After 3 days of cultivation, 3.5 units per mole of lacase activity was detected in a 50% pressed apple pomace growth medium, while 2.5 units per mole was detected after 7 days of growth in a 20% potato waste medium. The high level of phenolics in the grape waste stream greatly inhibited the growth of the fungal strain and therefore did not produce any enzyme. But when the 20% potato waste were supplemented with the red grape waste as an inducer, the addition of low concentrations of the red grape waste, up to 5%, enhanced the lacase production by Trametes pubescens in both types of potato waste, with more than 4 units per mole with the red grape waste compared to approximately 2 units per mole when no red grape waste was added. Furthermore, while maximum lacase production occurred on day 7 in the potato waste alone, as you can see here, production when red grape waste was added was not only greater, but also occurred earlier at days 3 to 5. So this waste stream can therefore be used as an enzyme production inducer. In a previous study, our team showed that the dimerization of ferulic acid, a phenolic that is found in a range of fruits and vegetables, can result in products that exhibit a higher antioxidant potential compared to the starting material. We therefore tested whether the lacase produced in this study could be applied in this particular biocatalysis reaction. The crude lacase obtained from cultivation in potato waste was applied in this bioconversion. This was then compared to the purified lacase under the same conditions. Analysis by thin layer chromatography, or TLC, as can be seen here, revealed that similar products were obtained irrespective of whether a purified Trametes pubescens lacase was used or whether the supernatant from our proof of concept airlift or bubble reactors were used. Staining of the TLC plate with DPPH, which can be used to detect antioxidant activity linked to hydrogen ion donation, highlighted the antioxidant activity of the products that are produced, as seen by the white spots against the pink-purple background. So this study ultimately allowed our team to develop an integrated bioremediation and beneficiation proposal for bio-based waste streams, where value-added products such as enzymes, antioxidants, and bioethanol can be produced for application in a circular economy. Here are some of the outputs that were generated from this study. And again, for more details, please feel free to consult these publications or contact me if you're interested in knowing more about the study. And then my final example, I will focus on our most recent work on the use of lignocellulose-rich waste, canola fines, for bioplastic production. So the South Africa's canola industry is rapidly expanding, and it's quite a lucrative sector, largely centered within our Western Cape province. This is driven by increased global demand, advancements in the seed cultivars, and growing consumer trend for healthy oils. The production has therefore significantly increased over the past decade. Canola is a successful winter crop in South Africa, but it can also be grown under irrigation in summer, and it basically plays a key role in crop rotation and supply of animal feed. So because it's produced all year round, it basically makes an ideal resource then to explore for biorefinery applications. Researchers in our institute have shown that high sugar yields can be achieved from canola fines by pre-treating the biomass with low concentrations of acid and steam at a biomass loading of 5%, followed by enzyme hydrolysis with viscozyme or cellulase mixtures. The high sugar yields after pretreatment then makes the canola fines a promising lignocellulosic feedstock for polyhydroxybutyrate or PHB production. PHB has become quite topical as a bioplastic that can be used to replace fossil-based non-biodegradable plastics. And luckily for us, actinomycetes are well known for their biosynthetic potential and have been reported previously to have the ability to produce the bioplastic PHB. In this study, 3 Gordonia species, 2 streptomycetes, a micromonospora, and a nocardia strain were used. And these strains were all isolated from diverse environments with the aim to see whether the environmental source may influence the ability of the strain to produce PHB. Optimal production of PHB in a defined medium, in this case, the International Streptomyces Project Medium 2, was observed for strain Nocardia humpkensis, which was isolated from soil collected from a riverbank, followed by the Gordonia strains. Interestingly, the lowest yields were observed for the streptomycete strains and the micromonospora, but the study was the first to report on a micromonospora strain with the ability to produce PHB, even if it was at low concentrations. Analysis of the genomes of the 3 Gordonia strains, the Nocardia humpkensis and Streptomyces strain HMT19, revealed that none of the core genes were located in defined operants, but all 3 genes required for PHB production are present, confirming the biosynthetic potential of the strains to produce PHB. It was interesting to see that Gordonia strain JC51, which showed potential when grown in ISB2, did not grow well and produce very low amounts of PHB in a synthetic canola fines hydrolysate. On the other hand, the Gordonia BS2 that did not grow well in ISB2 and produced low amounts of PHB in this particular medium grew very well and produced the highest amount of PHB of all the strains in the synthetic hydrolysate. However, the maximum growth and PHB production took a bit longer than with Gordonia strain BG1.3. And although the Gordonia strain JC51 utilized similar the salabioes in similar amounts to the other strains of Gordonia, it was not able to utilize arabinose and only utilized about 1% of the xylose in the synthetic hydrolysate. So obviously, at the moment, current optimization studies is focused on the 2 Gordonia strains, BS2 and BG1.3. And just for interest, the one was isolated from a lagoon, while the other was from a peatland. So in conclusion, for the winery wastewater treatment project, it can be concluded that the biological sand filtration system can provide a low-cost, energy-efficient, low-maintenance, and a sustainable means of treating cellar effluent at small wineries. And this project required a transdisciplinary approach involving academics from different disciplines and the farming community, ultimately leading to research uptake. And this is obviously what we all want to achieve with our research. For the bio-based waste streams project, we showed the various applications or abilities to either produce enzymes and show the application, and then also the production of bioethanol from the potato waste, making it very promising resources. The study then allowed for the design of a proposed integrated bioremediation and beneficiation process, promoting a circular economy. And then finally, for the canola fines project, we could see that the pre-treated canola fines contain relevant sugars for the growth of actinomycete strains with the ability to produce the bioplastic PHB. And this approach theoretically could then provide both environmental and economic benefits in support of a circular economy. With that, I wish to thank everyone involved in the research presented, as well as the various funders that made this research possible, especially the Water Research Commission of South Africa. And thank you all for listening. If anyone is interested in learning more about these projects and the current work, please feel free to contact me.
Thank you, Dr. Merlies. So just for reminding our audience, there will be a debate after our communication sessions, okay? Now we'll pass to the or communications?
Yes, we will invite our friend, Zahra Azuz. Yes, we've invited to present a study about innovative enzymatic valorization of agricultural product for fermentable sugar production.
Thank you, madam. Good morning, everyone. I'm happy to present today and to participate for this email. And my presentation is lost. Yeah, my presentation today is entitled Innovative Enzymatic Valorization of Agricultural Co-product for Fermentable Sugar Production. It's not working. Sorry. So, in the today wars, the wars material and increased in greenhouse emissions present a significant challenge. In the same time, waste production and the increasing further straining our planet ecosystem. For these courses, the funding in alternative solutions is necessary. For this some funding, we can show the renewable energy. Renewable energy of the sustainable both front and one promise area is the recovery of linocellular waste. This research explores how to this abundant renewable source can be enhanced to produce biofuel, offering a cleaner and more sustainable energy solution for the future. The source of biomass, there are some lignocellulosic biomass. There are a lot of types of the source. We can find forestry residue, animal byproduct, industrial waste, organic waste, wastewater, and agriculture and core resident. In this work, used in the agricultural residents such as white brand and white straw. In general, the composition and the structure of lignocellulose contain three component polymer. The first one is the cellulose. Cellulose is the polysaccharide contain the chain of the glucose linked with the tip of beta-1,4. The second polysaccharide is hemicellulose. Hemicellulose is the heteropolysaccharide contain the chain, lateral chain of the axilla and the different ramification for different type of the sugar. The last component is the lignin. Lignin is the type of phenylpropane derivative. So, for the hydrolysis and saccharification for this polymer necessitates a lot of type of the, or synergy of different type of the enzymes, as like cellulase. In the cellulase, we can find three type of the enzymes, endoglucanase, silibioderase, and beta-glucosidase. For the hydrolysis of the polymer xylan, we necessitate to find the enzyme endoxylanase and beta-glucosidase. For the lininase, generally, ce sont the peroxidase and the lacase. So, the objective of this work was to demonstrate how mix the two types of the strain of fungi, Trichoderma afroarzianum and Aspergillus niger, for to mix for these two both strain for the fermentation and production of the tip of the enzyme before to use it for saccharification. And for the biological material used in this work, two types of the strain, Aspergillus niger and Trichoderma afriozoanum. After sporulation, the spore was used for the fermentation for two substrates, white strain and white bread. Two types of the fermentation, solid state fermentation and submerged fermentation, was analyzed and used for to compare the quantity and of the production of the enzyme. Before this one, it's necessary to test it if there are the synergistic between two strands in the co-culture. And the test was realized in this figure, and we fermentation used just only with Aspergillus, the second with Trichoderma, and the third test with to mix the two strands. After fermentation, and extract was recovered, and the different type of the enzyme was analyzed, which endoglucanase and oxylanase activity, beta-glucosidase, beta-xylosidase, peroxidase, and lactase activity. Also, the protein of the concentration of the protein was measured with Bradford method. After testing the synergistic positive between two strands, the optimization of the production was used with two types of the optimization, as like OFAT, classical approach, one vector type, and RSM, response surface methodology optimization, using box, bank, and design as the matrix of the optimization. 4 parameters was analyzed: incubation time, moisture, incubation temperature, and the relation between two substrates, white strain and white bread. In the condition optimal, the extract was used and utilized for the saccharification with pre-treatment and treatment substrate, and the hydrolysis, the enzymatic hydrolysis was realized at 50 degrees Celsius, and the reduced sugar was analyzed and measured via DNS method. The results of the reduced sugar was confirmed in the TLC. Here, the results for testing if there are synergistic between the co-culture Aspergillus and Trichoderma. We can say here the better activity was measured with the co-culture, and if you compare with submerged fermentation and solid state fermentation, the activity is better with solid state fermentation. And for this reason, continue the other parameter. For the optimization of the OFAT results was shown in this figure about for the different activity on the glucanase and hexaglucanase for the 4 parameter analysis. The first one for incubation time, the humidity, the temperature of incubation, and the duration of between the two substrates, and the optimum was fixed for the both activity as like endoglucanase and oxylanase. The result for RSM based in the box bank design and the analysis of the variance was shown in this table for the both activity. The activity on the glucanase, CMCase, and xylanase. The model is validated with the p-value registered lower than 0.05. Compared with lack of it, it's not significant. The model is valid, and we can start to study the different interaction and the effect of the different parameter. The effect of the linear parameter, some parameter is significant and the other no. The same for the result for the interaction between different parameters. So interaction is positive and significant, and the other no. And we can say here in the response surface plot is just showed the interaction between influencing sugar influencing factor, and the different here we can say the response and the interaction for both parameters, and we can say the remove and the variation of the response depend in the interval of the study. Before to test the saccharification, if you remember, the composition of the hemicellulose is heterosaccharide, and if you need to totally saccharified this one, necessitates a lot of type of the enzyme. This type in general is auxiliary enzyme, and tested different type of the enzyme is present in our extract route, and a lot of type of the enzyme present the activity. This is better for total saccharification of the biomass. For the result of the saccharification is shown in this figure. The figure A and the figure B present the comparison between end-treatment and treatment substrate. The first one is the result for the reduced in sugar with end-treatment substrate, and the B present the treatment substrate with different variation of the reduced in sugar. And in the parallel for each figure, we can show the if you want to compare for the quantity of the reduction sugar compared, it depends in the type of the substance. The first one, for example, the white bran, and the Russian. You can say here the white bran presents the bitter and presents the high concentration and the quantity of the reduction sugar presented. This result was analyzed with the TLC, and the results of the figure of the reduction sugar confirmed in the TLC. The column, the A and the B, A present the untreatment reduction sugar, and the B treatment sugar. For the first column is the standard. We can see the column, the name, the oligosaccharide used as the standard, the glucose and the oligo, donc hexane, C6. And the second column is the sample recovered with the white strand, the white bran, and the third the Russian. It's the same for treatment. By the consistence and the bond depend in the concentration of the dilution sugar. We can say here, for example, in the figure A and the column 2, it's very dense, and this confirm this result showed in the white bran, because in the white bran present a high concentration of the reduction sugar. It's the same, present the different type of the oligosaccharide. The purpose is the total saccharification. And the last one, I need to find the monosaccharides like glucose, arabinose, glycellose. And we can say the last bond, compared with the C1 is the glucose. It's very dense. This is the objective to produce the reduction sugar. So, just I finished for this conclusion. The mixed solid-state fermentation of Aspergillus niger and Trichoderma afroasianum successfully valorized white bran and straw into natural lignocellulosic biocatalysis. The co-culture exhibited strong enzymatic synergy, enhancing saccharification efficiency, particularly for a treatment substrate. This cocoa-friendly approach offered the promising route for converting agricultural residue into fermented sugar, contributing to sustainable bioprocess and circular bioeconomy advancements. For this, I finished my intervention. Thank you for attention.
Thank you, Zahra, for this nice talk. Now we invite Mrs. Radia Bilmana, who will present. Absent. We'll call now Professor Boualem Harfi. No? This was, okay. Yes, we move to online presentation with Miss Edison Ronnie. Miss Edison Ronnie, QCNV Foko. He will present nutritional quality index in quite breast meat fed with pterigori pardalis byproduct meal as an alternative in the circular economy. Thank
you very much. Oh, okay. So thank you very much.
Can I start my presentation?
No, not yet. Not yet. It's going to be.
Can I start my presentation?
No, not yet. It's going to be.
According to the program, it's Mr. Gibson before. According to the program, it's Mr. Gibson before. Can I start my presentation, please? According to the program, it's Mr. Gibson before. Can I start my presentation, please? According to the program, it's Mr. Gibson before. Can I start my presentation, please? According to the program, it's Mr. Gibson before. Can I start my presentation? Hello, are you hearing me? Can I start? According to the program, it's not yet. Can I start my presentation? Hello, are you hearing me? Can I start? Not yet.
Wait a moment, please.
Hello. Okay. Are you hearing me? Can I start? Not yet.
Wait a moment, please.
Hello. Okay. Are you hearing me? Can I start?
Yeah, Minasra, if you can just put your slides in full screen, and then we'll be starting by you, and then we'll come back later to Edelson and Rooney.
Okay, thank you.
Okay, please put your slides in full screen, and you have only 10 minutes for your presentation, and then we'll be starting by you,
So,
and then we'll come back later
before
to
to start, or to begin, I would like to thank the scientific committee for accepting me and giving me the opportunity to participate in this very important congress or conference, which addresses very interesting areas of research. I would like to thank the scientific committee. So, I'm Dr. Nasra Amina from Ammar University. I will participate in this conference or congress with work titled Eco-Friendly Packaging Films based on pectin extracted from agri-industrial fruits residues for my work contains an introduction, generality of about pectin, materials and methods, result and discussion, and will end with conclusion and perspective.
Minasra, please, we are not seeing your screen. Can you just put it in full screen, please?
Yes,
No, your presentation is not yet in full screen.
So,
can you
like
share
this?
Can you share
like,
again your screen
like
and put
this?
it in full screen? No, it's not yet in full screen.
So, I don't know.
Votre présentation n'est pas encore en
Oui,
mode
j'ai
présentation.
bien compris, oui, mais j'ai essayé de faire...
on vous laisse repartager à nouveau, s'il vous plaît.
D'accord.
Et il faut tenir au temps. On n'a que 10 minutes pour votre présentation. Maintenant, il faudra mettre votre présentation en mode plein écran.
J'ai fait ça. Est-ce que...
Ce n'est pas encore le cas.
Oh là là.
Est-ce que il faudra passer par le mode diaporama?
Oui, oui, j'ai pris le diaporama, mais... Voilà, hein, diaporama. Non, pas encore.
Je vous laisse partager à nouveau votre écran et peut-être ce ne serait pas la peine de mettre...
C'est le même problème toujours,
Oui,
je sais pas.
peut-être...
J'ai fait l'animation, le diaporama, mais rien n'a été changé.
Il n'y a pas de souci. Donc il ne faudra pas peut-être passer en mode écran, en mode plein écran, comme ça nous permet de voir les diapositives. Non, c'est l'inverse. Il ne faut pas utiliser le mode plein écran. Comme ça, quand vous défilez vos diapositives, on arrive à vous suivre. Est-ce que vous pouvez passer juste à la diapo 2?
Voilà.
Allez-y, pour voir.
Donc, to start, I will start with a small introduction. So pectin is a natural polysaccharide found in the plant cell walls, primarily composed of D-galactorinic acid, extracted mainly from citrus fruits and apples. It's very varied for its functional property like gelling, thickness, and stability, and its health benefits. So today, pectin is attracting attention for the production of biodegradable films intended to replace food packaging. So, faced with plastic pollution, research is turning toward ecological alternative for food packaging. So this work aims to optimize the extraction of pectin from apple pomace variety Golden Delicious to produce a high-quality polymer integrated for sustainable agri-food application. I will give you a small definition or words about pectin. Pectin is, pectins are complex polysaccharides from high plants, primarily composed of D-galactorinic acid and natural sugar like rhamnose.
Minasara, please, we are very sorry, we cannot see your slides. At this moment, we are only at the first slide of your presentation. That's the reason why we are asking you to stop sharing your but now it's okay. We ask you to use this.
Okay, I understand this.
Yeah, unfortunately, your slides are in French, and this is not helping the audience to better understand and read. Please take only 10 minutes. If you take more than that, I will stop you and we'll move to the next speaker, okay?
Go ahead. I will try to respect the time, okay. So, I will define some words about pectin. Pectin is a natural polysaccharide found in plant cell walls, like fruits and apples. Pectin are complex polysaccharides from high plants, primarily composed of D-galactorinic acid, natural sugar like rhamnose, arabinose, and galactose. Thanks to their thickening, gelling, and emulsifying property, pectin are widely used in the food and biomedical industry. The figure number 2 explains the 4 types of or classes of pectin. We have rhamnopectin 1 and 2, xylopectin, and the fourth one is rhamnogalacturonan 1 and 2, xylogalacturonan. So the main structure of pectin is acid galacturonic bind with alpha 1,4 bond. Pass now to application of pectin in food and in the biomedical domain. In food industry, pectin are widely used in the food industry for their gelling, thickening, and stability property. They are used to the production of jams, jellies, and marmalades. In the nutrition and health domain, they promote intestinal transit, have probiotic action, reduce the absorption of glucose and cholesterol. About biomedical application, we can use pectin as a drug vector for encapsulation and healing, dressing, and other ways. Pass now to materials and methods. In our work, we used the residues, waste of apple, after eliminated the juice or of apples, and we try to find the optimal parameter or to extraction pectin. We work to three parameter or factor: effect of temperature, of time, and of pH of extraction. So we use pH 3 value 1.5, 2.5, and 3.5. For temperature, we worked at 17, 18, and 19 degrees Celsius, and 4 times we have used 3 times 17, 18, and 19 minutes. And at the end, we use the extracted pectin to prepare biodegradable biofilm used in the agroalimentary industry. So we have two parts in our work. The first one is about extraction and characterization of pectin, and the second one is about the characterization of prepared or formulated biofilm. So for pectin, we have evaluated the extraction ration of extraction and some physicochemical properties like esterification degree, methoxy degree, and humidity or water content, and the value of H. For a functional property, we have estimated the water and oil holding capacity. We have also studied the stability or solubility of pectin in different organic and hydrophilic solution or solvent. In the next part, we have studied we estimated some characteristic of biofilm basis of pectin. So we have estimated the thickness of biofilm, some macrobiological characteristic, and water content and color. Pass now to results and discussion. Firstly, we found or we have evaluated some physicochemical properties of pectin. So for extraction yield, the table 3 illustrates the pectin extraction yield from apple pods based on different experimental conditions. So according optimal extraction conditions not only allow the high pectin yields, but also contribute to improving its physicochemical property, notably through a high content of anhydrochloric acid and sustainable degree of esterification. The high extraction yield were found at pH 2.5 at temperature 17. So pass now to degree esterification degree of esterification and methoxy degree. So a high degree of esterification reduce electrostatic interaction and charge density in pectin, promoting to formation of complex with actions, formation of complex with cations of this increasing viscosity. It also influences hydrophobicity of pectin, thanks to the hydrophobia elevated its hydrophobic groups. Our result is in accordance with the work of Y and Misali 2014. About methoxy degree, the degree of methylation of methoxy of pectin is a critical parameter that influences its gelling properties and industrial applications. So extraction conditions such as temperature, pH, and duration or times significantly affect this parameter. A highly esterified methoxy pectin require the presence of calcium ion to form gels. So for H, we have found the H content in our pectin seated between or varied between 0.73 to 1.49. So according to the Codex Alimentary Standards published by Food Agricultural Food Organization in 2009, the total H content of pectin should be around 1%. Pass now to the pectin, color and appearance of pectin. So in the browning observed in pectin is mainly the caramelization or the Maillard reaction. These reactions are favored by high temperature and acidic pH, and also to the presence of glucose, sugar, leads to the formation of melanoidin responsible to the brown color. Pass now for the water and olidine capacity, we have found elevation in these two properties.
Please, Dr. Minasra,
oui.
Can you move directly to your conclusion now? We have to stop you because you are beyond the time. Please go through to the conclusion.
Okay.
Thank you.
So for conclusion, this study aimed to optimize the extraction of pectin from apple plants to produce a high-quality biopolymer intended for the production of biodegradable films for the agri-food industry. The tests showed that acidic conditions, pH equal 1.5 at moderate high temperature between 18 and 19 degrees Celsius and with a prolonged extraction time are allowed for an optimal extraction yield equal 18.7%. So to the produced films present low water content and macrobiological stability and no contamination, thanks to the use of hydrochloric acid in pectin extraction and citric acid in the formulation of biofilm. This biodegradable film appear to be a promising alternative to food packaging, especially pectin biofilm 7, 8, and 9. So pectin film from fruits residues or pomades constitute a sustainable alternative to systematic or synthetic plastic. Optimizing formulation, blend, cross-linking, and active agent improve their performance. I will cite some perspective. So modern technique, we can use modern technique like ultrasound assisted microwave extraction could improve yield and limit thermal degradation and improve quality of pectin and biofilm. The addition of bioactive agent like essential oils and plant extract will also help strengthen the antimicrobial properties of the film. And finally, it is very necessary to study the rheological property of prepared biofilm, their mechanical resistance in order to evaluate their durability and behavior during use in the agri-food industry. And thank you for your attention.
Thank you, Dr. Minasra. Can you stop sharing your screen now? And please, for the future of your presentation, keep this principle, 10 slides, 10 minutes. This should be very helpful for you. Thanks. We'll be moving now to the last presentation of this morning session. Edelson Rooney, Kuzinka Fuuko. Are you online? Are you hearing us? And please, if you can just start sharing your screen and put it in full screen mode. Edilson, are you hearing us?
Yes.
Okay, please, can you share your screen? And once we see it here, and once it's in full screen, then I will let you know when you start your presentation.
Okay.
Thank you.
Wait a minute, please. Can you see my presentation?
Not yet. I will be checking with the techniques here. Edilson, please, can you put that in full screen? Okay, now it's working. Okay, please, you can start, and you have 10 minutes for your presentation. Thanks.
Can you see my presentation, please?
Yeah. Can you just move to one slide to see that if everything is working? That's fine. Okay, perfect. Okay, you have 10 minutes. Go ahead.
Okay.
Edilson, you can start your presentation now.
Can you listen to me?
Yeah, we see very well your presentation, and you can start it now. Thanks.
Okay. Good morning, everyone in the scientific community and the public present today. We will discuss a very important topic, which is the circular economy and animal nutrition. The title is Nutritional Quality Indexes in Wild Breast Meat, with Pteridactis pardalis byproduct meal as an alternative in the circular economy. The poultry meat production has been increasing since in the recent years due to the competition from red meat and import from the other country. The other country, for example, Poland, Spain, and France, to participate to 22% and 30% and 11%. Now we will focus in which Pteridactis pardalis meal uses in cheese and salt meal. The Pteridactis pardalis is a marine catfish in the Loricalidae family, now in the other country as the devilfish, as is very dominant as invasive fish. This species is rich with soft meal, for example, the responsible production and agriculture and protein-rich and source and utilized and prolific species avoid competition for food with native endangered species. And conventional products We use soybean meal production, limited resource as of meal, environmental uses plus 70%, and salt and saturated fatty acid. The Pterodactylus pardalis have the greater nutrient content, availability, biological control, more palatable and economical. And protein and quantity and quality is more digestible, more amino acid balance, and carbohydrates, no anti-nutritional factor, more minerals, for example, phosphorus, no other factor, aminos, steroids. It's easy to produce in aquaculture, easy to store, and fat reaches in omega-3, 31 and 25%, and acid, DHA, docosahexaenoico, docosahexaenoico, and omega-6, no? And acid arachidonic and selenoic. The objective of these presentations evaluate the lipid nutritional index in qual-breast milk feed with different levels of inclusion per byproducts from Pterodactylus pardalis. Then growth performance, biometric index, fat acid profile of this meat, and nutritional index. No? Materials and methods. The location adjustment. No? The Experimental Center Poultry Unit in Jose Faustino Sanchez Carrion National University in Peru. A total to 330 quails we use it with 20 quails per cage, no, with 4 treatments and 3 repetition. The statistical design and analysis of the variance ANOVA was performed with initially with as a coverage using the statistical program statics graphics. Now the results are represented as mean with standard error the level significances. The best treatment in this presentation was the second treatment, the 5%. No, show the 4 days, the 5, 0, 50, and 51, and 53%, and something inclusive the Pterodactylus pardalis and substitution for soybean flour. The experiment analysis, growth parameters, biometric parameters. In laboratory analysis, the proximal composition with methods Dumas, and analysis and nutritional index equations, no? And the analysis of quail breeds, the composition proximal, no, method Dumas, and fat acids profile, the methods of follow with use the chromatographic. The experimental result, the final experimental phase. No, you can see trial 2 grow the nutritional performance result and Japanese quail feed different experiences with 2. The result shows what final weeks the treatment with 50 and 55% flour, and we look at the feed conversion index, the base values to the 50 Pterodactylus pardalis treatment, as well as the protein and the energy efficiency. Comparative results. When the other investigations to compare studies with fish silage, the fish silage and oyster meal, these results are still better, demonstrating they use the Pterodactylus pardalis meal is more efficient with the other studies. Inclusion levels in shrimp byproducts, the first investigation for Kumar Vidal, said that use the 5 and 7 could be beneficial in the diet. And the final experiment, the phase, the table 3 biometric parameter results for a quality feed, the Pterodactylus pardalis for 32 days, shows that training with the 50% had the highest carcasses average yield compared to the control, the 0%. In the table 4, fat acid profile of quail-breast meat feed with Pterodactylus pardalis meal. Using chromatographic tests in quail-breast, the fat acid profile was analyzed and showed very Very notable difference between entrainment with the highest percent to omega-3 and the total substitution with soft meal and omega-6, no, and the diet control. In this presentation, you can see that the indices, the lipid nutrition, the quadrimis from equal, atherogenic index, thrombogenesis index, cholesterolemic index, and full lipid quality. The double 5 nutritional and quality indices in Japanese. All treatments are beneficial that the referential levels no highest. In conclusion, the better final weights were observed in treatment with a 50% inclusion, the level Pterodactylus pardalis byproduct meal. The best results in terms of fat conversion ratio and quality were obtained with the 50% better weights and carcass percentage with observed with inclusion levels 50% the Pterodactylus pardalis produced meal. In terms of sustainability, Pterodactylus pardalis meal can replace the 50% and meal without affecting growth to carcass as well, while high inclusion levels lower final weight. In the case to omega-3 levels in equal breeds, the greater the substitution of meals with Pterodactylus pardalis products meal, the better the result. Nutritional indices vary according to the type to ingredients. For example, subproducts to animals, the vegetable origins use the quality formulation but remain within the reference levels required for consumption. This is the bibliography. Thank you.
Much appreciated, Edilson. Thanks for taking less than 10 minutes to present your very nice work. Stay with us. Now we'll be moving for the debate as we invite all the speakers. Arfan, can you come with us, please? Is any of you who have any question? The debate is open now.
Okay. C'est Mouzaza from École de Chimie de Rennes. For Mohamed Orfan, first of all, thank you very much for the nice presentation. I have two questions. One question is just, I want to have your opinion. Why the poor countries generate waste food in the same quantity as rich countries like Europe and the United States? You have an idea?
Thank you very much for your very good question. But I have no idea in Europe how they tackle their food wastage. But if you see the graph, maybe they have almost same level.
But Nigeria is twice.
Yes, twice.
Yeah. Okay. Yeah. Okay. Another question is about the quality of the waste. You know that you present some transformation of the some waste like peels, like my question is, if I have a waste containing peels and bread and meat, and how we can do to transform this as quality materials?
Yeah, we can do to supplement animal feed, mostly like the waste bread. We can reuse it for animal feeding or some sort of another type of animal feed we can provide. But for peel, sometimes some people or some countries, they process this, wash it, and make because they contain some lignocellulosic material. So this could be implemented as animal supplementation for animal feed as well as for carbon source for microbial fermentation. So the peels could be utilized for further process.
Okay, thank you.
Thank you.
Thank you very much for your presentation. This topic is hot topic, is also important. There are many projects in the European Union or national or international. Now, my question is, can we use this valorized waste for the fly production, okay? Because it's very important for the alternative protein. Lately, I have come across some papers about this, how to, valorize, how to use this waste for the fly production. So what do you think about that?
Yeah, very good question. But I think in some places, these, just like composting, they are using. In some countries, soldier fly composting, have been used for the production of different kind of flies, and these flies could be as a or mealworm. This also is a potential protein source, and all these mealworms and flies, they can be cultivated on these kinds of food waste.
And one more thing, and also, can we use this, the waste for the production of lactic acid bacteria? Okay?
Yeah, sure. So for this, we need to hydrolyze all these waste material into fermentable sugars. When you got these fermentable sugars, then you can easily cultivate any kind of microorganism. It's very simple.
Thank you.
Thank you.
Maybe I have two questions. Yeah, I will first ask one question to the person who is online. Is that okay?
Okay, yes.
Edilson, I have a short question, if you are still hearing us. Edilson, are you hearing me online?
Yes, see, I listen to you.
Okay, I really appreciated your presentation and the way how you are recycling the byproduct as a meal for your quails, and you are hugely improving the ratio of N3 to N6. That's very, very interesting. The only question I have, when we improve or when we have such higher amounts of omega-3 in the meat, then there can be a huge risk of oxidation. My question, did you incorporate any antioxidants such as vitamin E in your feeds? Or do you expect to reevaluate that in the future? Thanks.
Thank you for your question. Yes, the treatment for the total replacement soy meal only used to 2% antioxidants, the VTM.
No, my question was, did you include any antioxidants such as vitamin E in your feeds? Because when you improve the ratio of omega-3 in your meat, it's prone to oxidation. And then how are you dealing with that? Or do you plan to have any test to reduce any oxidation of your meat? This was my question. if you don't have the response now, there is no issue. That's
in the formulation, in the group to antioxidant we use, and it was prepared 2 days, the rate had the premix.
yeah, the premix contains more vitamins, and they are for the health of the animals.
Only vitamins and minerals, but
Yeah.
no antioxidant.
Yeah, but I suggest you to consider that in your future, because the topic on which you are working is really, really relevant, because at the same time you are considering the sustainability and the quality of the meat, but you are just at this moment, I think, improving the nutritional quality, but keep in mind that's the oxidation of meat that contains high amounts of omega-3. They are like prone to this quality defect. Yeah, but very interesting work, and congratulations again. Yeah, thank you. Now we'll be moving and taking other questions from the audience. Maybe we'll be taking 2 more questions because we are in delay this morning. Yeah.
Okay, thank you, Dr. Gagawa. I have just a question for Irfan. Thank you for your nice presentation. As you know, for waste, we have 2 main challenging concepts, which are first the waste diversity and the seasonal variability. Now, do you have any idea regarding one period in both countries, in our country and in yours, which is Ramadan fasting month? Now, for the circular economy approach, do you have any available data regarding this period and the waste amount in Ramadan fasting period? Thank you.
Thank you very much for your nice question, but unfortunately, I do not have any data regarding Ramadan fasting. Yeah.
Yes. Marilise, did you hear me?
Yes, I do.
You're welcome. You're welcome at the Center of Agri-Food Technology. And I thank you for your collaboration with the Applied Microbiology Laboratory and now with the Center. Thank you for your continued commitment. I have a simple question about polluants in wastewater. Did you use green nanoparticles to remove the polluant? Did you try to use it? Green nanoparticles.
Yeah, in this particular study, we didn't make use of the nanoparticles, but we have another study that's currently taking place where we are looking at the use of silver nanoparticles in treatment processes. So it is something that is being explored at the moment, but yeah, this particular study that I reported on was just the biological sand filters.
Thank you.
The last question from the audience.
Oui.
Go ahead.
Assalamu alaikum. Bismillah ar-Rahman ar-Rahim. I am Habiba Drisi from Tamarast University and lecturer in microbiology field. My first question is to Professor Arfan about any trials, if you have done any trials to know microbial dynamic in bioplastic production. Are there any trials to know dynamic microbial for bioplastic production? The second question is For collagen, you have mentioned that you can produce collagen from waste, and if this collagen can be used for health, human health. And one question for Ms. Zahra. You choose only 50 degrees for your experiment, and we know that fungi are not stable at this temperature. Did you any additional information about these details? Thank you very much.
Thank you very much for your question. First, I did not clearly hear what kind of microbial dynamics you want to ask.
Can you repeat, please?
What kind of microbial dynamics you want to ask?
Yes, microbial. Microbial, to know which kind of microorganism.
Microbial diversity.
Yes.
What kind of microbes they have capability to produce?
Yes,
to produce
bioplastic, and if this bioplastic can be used in packaging for fermented food.
Yeah, mostly Bacillus subtilis, Bacillus genus, and some sort of Pseudomonas. They have capability to produce bioplastics. And I think maybe in the world there's some practice this bioplastic could be used for packaging processes. And the second question is you have asked about collagen. Yeah. So I did not mention, I have not worked about collagen production, but this collagen can be extracted from animal waste. Yes. Yeah, this could be used as a polymer for other applications.
Okay. Thank you.
Yeah, thank you.
Thank you for your question. Yes, for the choose for 50 degrees Celsius for scarification, it's not random. There are the old statisticals and characteristics for the different type of the enzyme purified. For the trichoderma and aspergillus, for example, for endoglucanase purified, there are a lot of characteristics and stability effect of the temperature and the stability for different enzymes. And about, according to these results, and compared with the enzyme produced with the aspergillus and trichoderma, the lot of enzyme is stable between 30 degrees Celsius, about 60 and 70 degrees Celsius more than 3 days. And this temperature chooses for the saccharification. The enzyme is stable. More than 3 days, donc the enzyme decreases automatically, the activity decreases in the time. But a lot of enzyme is stable, our production also, cause. This is your question? Nice.
Thanks to all. Any last question? Okay, we'll be taking one from here. Yeah, please. Thanks.
Thank you. So my question is addressed for Professor Marlays, if she is online.
Marie-Lise, are you still with us?
She's not?
She left, yeah, sorry.
Okay, so my second question is addressed for Dr. Azouz. It's about, I would like to understand some points in your interested work. So the first one is about the actual status of the production of fermentable sugar on the industrial scale. If you have any idea of the actual status of the industrial scale of the sources, please?
Yeah, For example, Civitar produce the electricity and export electricity for the sometimes in the Itatic, just producing in the reducing sugar and converted a lot of waste produced in the industrial residues. Just an example, but there are a lot of, so in the moment, because the waste industry causes the problem for the environment, and the Itatic control assist for to valorization this and to find the solution for this, and automatically a lot of industry, for example, Danone, it's, I think they just the last years started to waste treatment the wastewater, and automatically if treatment any was, there are the same results. The objective to find the sources, the new sources for the energy.
Okay, so until now, what is the sources that we are using?
Here in the my
yes,
is the biomass, linocellulosics as white straw and white bran.
Okay.
But we can choose another different type of the biomasses, but linocellulosic biomass.
Okay, so another question, please.
We have maybe to stop here.
Okay,
I invite you just to continue talking during the coffee
Thank
break.
you.
Thanks to all the speakers of this morning and for your questions as well for here, because we are a little bit in delay. That's much appreciated. We invite you for the coffee break and see you at 11:50, please. Thanks.
ميرسي شكرا.
Le pointeur n'apparaît pas sur l'écran.
On va parler après donc de comment on a créé.
Yeah. Good morning, everyone. I hope that you enjoyed your break. Now we'll continue with the second session, and then I will let our colleague introduce the first speaker. Go ahead.
Yeah, we'll continue with the plenary lecture given by Professor Emna Amal, who is a researcher at the University of Sfax, affiliated with the National School of Engineering of Sfax and the Laboratory of Environmental Sciences and Sustainable Development in Tunisia. Her work focuses on microbial biotechnology, waste valorization, and environmental microbiology, with particular emphasis on the treatment and recycling of agri-food byproducts and bioprocess-based approaches to sustainability.
Please, your attention.
She has published extensively in international journals and actively contributes to research on circular economy strategies, organic waste management, and soil remediation, helping to bridge the gap between environmental protection and industrial biotechnology. Today, she will deliver her lecture entitled Valorization of Agri-Food Waste Through Composting, an Approach to Sustainable Development and Circular Economy. Please welcome her.
Good morning, everybody. First of all, I have to thank the organizer for inviting me to reach this conference. It is a pleasure for me, and at the same time, I have to apologize because I have thought that since Algerian people are French speaker, so to be more efficient, I prepared my slides in French language. So I'm going to translate simultaneously. So excuse me if something are not exactly the same. The work that I am going to present is about waste management and valorization, and especially for agri-food waste. So in this presentation, I first give some data about the problematic of the waste from agri-food industries and food security. Then I will speak about the waste management using the composting solution as a technique, and then I present the process, its advantages, and then some applications of this valorization by composting, and I will specifically speak about the olive oil industry waste, and then the confectionery, the data culture, and then poultry industry. And finally, I will finish by my opinion about this subject. If we consider the people, the world people, the population will reach the 10 million inhabitants in 2050. And these people will look for the work. They will be in the urban areas, and urbanization will be about 20 to 100%. And also, these people need industrial activities to live and to get their food security that gives them the disponible availability of the food, the economical and physical access to this food, and its use and stability. So all this industry and activity and life will draw a penury in some resources. And also these people will generate some waste, urban waste as well as industrial waste. And the quantity is estimated about 3.4 million tons per year. And this waste should be well managed and to allow to people to live in the limits of our world. So if we consider the solid waste management, these are this year about 2.2 million tons, and almost the third, 33% of this waste is discharged in the landfill, which are uncovered landfill. This is a practice which we use it in many different the countries in the world, and it raises a serious sanitary problem as well as environmental problems. So also we may notice that 9% of emissions in the world are from the industrial processes, and there is a waste of foods that is estimated about 1 million tons, and the mean of 132 kilograms per inhabitant, and this will have environmental and socio-economical impacts. So, if we come back to this wasted element, food, we may notice that the waste is first from agricultural exploitations, then from the selling of these foods, and finally in the retails and the food services, which represent 70% of this waste.
Also, if we consider this 1.3 million of tons that are wasted, the majority is from fruit and vegetables, which represent 45%, followed by fish and fish products, and then cereals. And if we consider this waste according to the country production, we may notice that America, Latin America, and followed by Europe are the first waste countries, followed by, if you consider the consumer, it will be the north of America and Oceania, then the Europe. And you may notice that this quantity is huge per inhabitant and per year. If we come back to these fruits and vegetables, we may notice that this is the first most important quantity which is wasted. But if we consider the gas emission, it will be the meat and the poultry, which represent 60% of the heat generated, of the gas generated from these materials. That could be valorized, of course. So such waste, food waste, will be will draw the use of arable soils, the use of a huge volume of water, and then the emission of gas without taking any interest or benefit from them. And also there will be an action and an impact on the biodiversity, which will be diminished. If we come back to the rules of sustainable development of ONU in 1915, we may notice that some of these rules will emphasize about the promotion of sustainable agriculture, also the guarantee of durable, sustainable water resources management, and also the fight against climatic changes. All of these rules will let us thinking about a circular economy. And if we consider one process, whatever it is in food, we may notice that all the processes will need entrance. Then there is the process which will have impacts on the environment. So if I consider the transformation of things, of goods, there will be a use of energy, water, packaging, and additives, and a big quantity will be transferred into waste, as well as impact on climate. What is recommended is to first reduce at the source this spoilage, this waste of food, then to reuse it for human beings and to feed it people, then to use it for animals. And the third recommendation is to make value of these elements. Then, if not, we can go to the recycling, and two processes are commonly recommended, that are composting and anaerobic digestion. So, at the least, we can make them incineration or in the landfill, and this will be a big waste. Actually, regarding World Bank, there will be an augmentation, an increase of this waste in 2050, about 70%. And then this problem of waste will be major preoccupations because of public health, or also because of resources and their conservation. So the three things to do is to reduce, to reuse, and recycle, which is recommended. Actually, the Environmental Protection Agency has given some option regarding this waste and their management. And the first, what is recommended is to prevent the production, then to reduce at the source their use, their reuse, their recyclage, then energy recuperation, and finally landfill. If we come back to different waste that are rich in organic matter, we can notice that the goal of this treatment is whether an energetic valorization or an agronomic valorization. So if we consider methanization, biomethanization, we can have the two goals that means the energetic production as well as agronomic valorization. However, for composting, it is only an agronomic valorization that we are going to consider soon. So, many wastes are, organic wastes are available in the world. These are leaves, green waste, wood, many other things, sludge from wastewater treatment plants, as well as excrement from animal breeding, and so on. And my work was made in Tunisia. If I come back to my country, we may notice that all of these waste are rich in organic matter, and they have a rate superior to 50%. These are poultry manure, byproducts of olive oil extraction, the waste, agricultural waste, and also almond solid waste management, solid waste from the city, and then also confectionery waste that are wastewater as well as bark, sesame bark. So, big quantity of the organic waste is generated yearly, and also with this waste there is illnesses, and then you have to find out a solution to prevent these illnesses and diseases. And composting is a way to choose, and I will clarify why this is the best solution for a developed country. There is a political volunteer that encourages this technology of valorization of organic fraction waste. And the impact of this valorization will be the preservation of agriculture as also as energy consumption. So let's know what is composting in 2 words rapidly. It is a microbiological process which is held in aerobic, and it is controlled, and there is different flora that are applied to compost. These are bacteria and fungi which make a bioconversion of organic matter, and then they give very stable products and hygienic products. This is called the compost. So it is the product of decomposition, then mineralization of organic matter, then humidification under the microbial activity, and in the optimal conditions we will find the compost. And this production of compost is followed by heat generation. This heat is very important because it is going to destroy all the pathogenic microbe and germ. So the process is very general. It could be applied to many kinds of waste, bio-waste, municipal solid waste, and so on. Also, as advantages, it is very easy to make and rentable. It allows an amelioration and improvement of soil structure. It increases the water retention in the soil, and also it is act as against the dehydration of the soil. The soil particles will be linked together, and that will improve the disponibility of nutrients for the plant. All this will improve the agronomic valorization of the soils. Also, with this compost, we are decreasing the consumption of chemical fertilizers, and we are going to fight against the heating, climatic heating. And we are in a sustainable development in the framework of a circular economy. Also, if we come back to the literature, and we find that in the world, over 460 patents treating the composting, there is 60 patents which were published in China, and the technical composting way used is aerobic composting. So,
to summer. Co-composting is very easy to handle. You have just to take on the left side, sorry, the pointer doesn't work, but it is all kinds of the waste you may have. You should mix this waste with water, and water can come from wastewater, from industrial wastewater, and then we let the microbial flora acting for 3 to 4 months at least, and then we will get our organic matter stabilized, and this constitutes the compost, which is a fertilizer. Many parameters are applied to succeed in this process. First one are related to the raw materials, that means waste, and it is related carbon to nitrogen, the humidity, the density and porosity of this waste. Then to control this process, there is temperature of the windrow, there is oxygen consumption, enzymatic activities, carbon, organic, and then emission of gases. And finally, to control the compost quality, it will be the rate carbon to nitrogen, the humic substances, and the lack of phytotoxicity. The process will be held in 4 main phases, which are mesophilic, thermophilic, and then cooling and maturation process, where different kinds of microorganisms, bacteria, fungi, and actinomyces are implied. Also, a recent study based on metagenomics has shown that in the compost there is more than 2,000 species, bacterial species, which are present. Some are thermophilic, some are cyclophilic. So, there is another constatation around why compost in Tunisia. And if we have a look on the landfills in Tunisia, we may notice that 80% of our soils are very poor and have less than 2% of organic matter. And besides that, we have a big quantity of waste that are from olive oil extraction. These are from date palm, from fruit and vegetable markets. There is a mark of coffee, also poultry miners, and so on. So, till more than 1 decade, we were working on this subject and about composting the byproducts of olive oil. And to remind what are these products, we should consider the process of olive oil extraction that starts by grounding the olives, making melting with water, then separation of 2 phases. One is solid, which is called grignon, and the other is liquid, which contains the oil, then the effluent, which is margin, called in Tunisia and in Algeria and many other countries, and this effluent is very rich in organic matter and in minerals. So, if I consider the national production in Tunisia of olives, we may have 500,000 tons of margin yearly, with the half of grignon of solid waste. And this solid waste will be extracted to have an exhausted grignon, an exhausted solid without residual oil, and then for the effluent, there will be storage. 2 minutes. I thought it is 25 minutes. Okay. So, let's speak about this process, which is very easy to handle. You should just make a mixture of this residue, and then we can have the product with quality according to the standards and the French standards. Some trials of production on this compost have shown that we increase and we may improve the yield to 27% when we use this compost instead of the fumier. And then we made many trials, and we have shown that we can win in the yield of the result. So after this, we have tried to make the composting of the waste of date, and then we held these different waste, we brown them, we make this withdraw, we control all the parameters, and we have noticed that it goes very well, and we have found a very good quality of compost and a stable compost that was confirmed by different measurements. And finally, the most important thing is how to use this compost to make biological fighting. And here we have tried many fungi that are frequent in Tunisia, especially verticillum, which attack the olive trees. And we make our joist, what is called a tea compost. This tea compost will use it to treat these verticillosis, and we demonstrated that there is inhibition of the fungi, almost 70%, and then we make experiment on this verticillum with different concentrations of joist, and we find completely inhibition of the fungi with its modification. So, what I can conclude, and I will stop here, that we can say that the compost is a very easy technique to be practiced in all the countries, especially our countries, North Africa, and we may go up to make tea, compost tea, which is a fermented liquid from the compost that will act positively and will inhibit many fungi and will be a substitute of chemical pesticide, and it is a really biopesticide which is very efficient, very not costly and not harmful for the earth. Maybe I can say that we can use many kinds of waste. Here we have experiments with wood, with almonds, barks, with sheep, with cafe, mark of cafe, and we make a mix with margarine, the effluent, and we have found a very good product when we change the composition of the raw material, which are waste, different kinds of waste. Of course, the problem is the polyphenol, since you use a margarine. No, they are all degraded, biodegraded, and we make measurements and analysis, and we are sure that they are a good product without anything. Now we can move to a smart compost that is made in a controlled bioreactor, where we are going to shorten the time needed, because the only problem with compost is the time needed to have the products. So if you control all these parameters, we may have a very quick composting that will be free and ready in about 10 days only, and we win a lot of time by using AI, and that's all for this. I'm on time, Chairman? Yes, I will stop here if you want. So, this is the conclusion, that smart compost is very interesting to be considered nowadays, because now for the classical compost it's easy, all people can make it, but this is a good solution for the future to win time, to win money, of course, and it gives a very, very good product that is ecologically interesting, since we are in sustainable development and a circular economy. Then we have made a valorization of agro-food waste, and on the social point of view, we can create many jobs for citizens, green jobs. And this will give a good quality of life of citizens. I thank you very much for your attention.
Thank you, Professor Emna. Now we will move to the second plenary lecture given by the Professor José Juan Santana Rodriguez. I don't know if he can hear us. Excuse me.
for just few technical issues. I'm seeing here.
Okay.
Jose, are you hearing us?
Yes, I am here.
I don't know if you are able to share your screen, or maybe we'll be moving to the next presentation until we fix the issues of sharing your screen, then we'll come back to you. Is that okay?
Oh, okay. Please move my slide from your side. Thank you.
Perfect. Then we'll be moving to the presentation from Lamia Salima Siddiqui. Are you here?
Yeah.
Okay. Then, Lamia will be talking about enhancing date palm syrup byproducts with aloe vera, and she will be presenting the physicochemical, microbiological, and antioxidants properties, which is a sustainable approach. Thus, the floor is yours, and if you can just present your work in 10 minutes, please.
Okay, perfect, right. I wait here.
Jose, we are starting first with an oral presentation here until we fix the issues of sharing your screen. Thus, please stay with us for 10 minutes from someone who is presenting here, and then we'll come back to you. Thanks.
Perfect, thank you.
Bismillahirrahmanirrahim. Assalamu alaikum. I would like to thank the organizer. I would like to thank the organizer for the opportunity to speak at this conference. My name is Siddiqui Lamia. I'm a researcher at the University of Tahrir Mohammed Bashar. Today I will be discussing enhancing palm date syrup byproducts with aloe vera, their physical, chemical, and microbiological properties. This is the plan. So, I will begin by providing some background on my research. That palm, Phoenix dactylifera, is a resilient fruit-bearing tree widely cultivated in arid and semi-arid region. Its fruits, commonly known as palm date, along with other derivatives such palm date syrup, have long been integrated to the diets of many cultures. In the light of current volatility of the global food supply, many of arid regions view dates as a year-round food source, offering the realistic, affordable, and sustainable option to versatile application in food and beverages. So, talking about aloe vera. Aloe vera barbadensis is another plant with a long historical traditional use. It is indigenous to the Mediterranean area, such as Algeria. Wide variety of aloe are widely found. So, total aloe vera contains numerous polysaccharides and metabolites, secondary metabolites, as wide range of minerals and vitamins. So, that palm's fruit, our syrup, and aloe vera are both rich in bioactive compounds, and that palms contain many other phenolic acids. This compound also show an antioxidant and anti-inflammatory and immune regulating activities. The objective of this study is to enhance the nutritional value to date syrup through the addition of aloe vera, contributing to sustainable and healthy diet, to evaluate the physical chemical characterization supplemented diet by the aloe. Now let's move to the experimental process, but I will be so short. The products have two type of palm date, the Tagaza and the Hemira. All are collected from the southwest of Algeria. The preparation of aloe vera powders, the aloe was cleaned, peeled, and dried. About the preparation of the syrup, the rub that's we call it also rob. The syrup was prepared using 2 kg of dates from either Hmira or Tagaza, thoroughly cleaned and mixed with 5 liters of mineral water. The syrup was then smeared for 2 hours over low heat, then filtered. The resulting syrup was then boiled again for sterilization. The samples, the syrup samples were divided to 4 batches, where Hmira and Tagaza supplemented by 2.5 and 5% of aloe vera powder for each. So, we're talking about characterization of the syrup. I just start only the physical chemical characterization of syrup, and second qualitative microbiology, and the third sensory test. So I will be short about the material. The first we will start by determination of moisture, following these phases. The second, electrical conductivity, consider measured by immersing the electrode probe in the diluted sample and having the registration, just read. The determination of pH using pH meter. The free acidity, we use, the samples are diluted in distilled water and was titrated with NaOH 0.1 to pH 8.5. That's it is expressed milliequivalents by kg. And the last parameter is total sugar. For this property, the diluted syrup sample was mixed and heated with carbonate calcium, and used the lead acetate to remove the proteins, filtration, and read by using photospectrometer. Now let's move to quantitative microbiology analysis. Here we will looking for total coliform, reducing aerobes, yeast, mold, and staphylococcus using the proper selective media. Let me now move to the results and just talk the main results. The physical chemical screen of aloe vera show the presence of many secondary metabolites important, like flavonoid, saponin, tannin, saccharoid, terpenoid, and anthraquinone. So the first result of the parameters is moisture. As we can see, in the syrup of daspan syrup, palm date syrup, Tagaza, Nature, or Hmira are very high. Then compared to the supplemented by aloe vera is higher, and the aloe vera is less. It's around 8 to 9%. In case increasing the moisture might expose the syrup to fermentation and deterioration. Concerning the electroconductivity, as we can see, the value of conductivity for the Nature Tagaza syrup Nature is a higher, about 3 to 3.5, compared to a supplemented, where is compared to the supplemented, where don't exceed 2.5 millisiemens by centimeter. Concerning the pH of the syrup, mostly all the syrup know how significant change. It's around 4.43 to 4.63 pH. Concerning the free acidity, and the free acidity, as we can see, the nature, syrup nature, as even Tegaza or Hmira are higher. The 12 to 30 is around 12 to 13 millicubic kg compared to the supplemented, where is around 9.8 to 9.5. A high acidity selectively promotes the fermentation. The last parameter, we can see as we can see also, the total sugar is higher, is around the 80%. When we can see for supplemented, the total sugar is less, reduced between 70 to 75%. This, as we can see, the sugar residue plays a preservative role against the microorganisms through the action and osmotic pressure, in some cases. In this application of PCA, gathering all parameters, we can see the application of PCA analysis assists in identifying the specific parameters that are most influential and distinguished among the syrup samples. Thus highlights in the impacts of aloe vera incorporation. The analysis results in the two compound model that illustrates total variance A model with high percentage 77. Of variability in the first principal compound was created by deriving PCE1 from initial data matrix that contain all determinate matter. As we can see here, sorry. 1 minute, I go to the, okay, so. I can see here in the up, we can see the supplemented syrup, and in the down, the nature syrup. So here, in brief, we don't find only any pathogenic or microorganism. Only some germ and total and DS. About sensory, mostly I go to in the end, 23% to 44% for a good test. Nevertheless, some panelists judged the syrup originated by aloe vera 5 had a poor test. We can see 8.5% for syrup Hmira and 27 for syrup Tegaza. Some mostly is acceptable. Okay, we can see if we compared our result to another result, Algerian result, the sensory survey natural date fruit syrup conducted by Gorshala revealed that the consumer in the southeastern Algeria preferred syrup that were light colored, sweetness, slightly acidic, and not overly sweet. In conclusion, this study demonstrates that addition of aloe vera improves significantly the physicochemical properties and the microbiological quality of date syrup, specifically Arachid syrup exhibits enhanced microbiological safety. The supplementation of date syrup with aloe vera provides additional nutrients, including amino acids, carbohydrates, fibers, phenolic compounds, which contributes to enhance the antioxidant activity and extend the shelf life of Arachid syrup by reducing the harmful bacteria growth. The sensory analysis results suggest that the syrup products enhanced with aloe vera have been successfully tested and improved using different varieties of that palm date fruits, such Hmira and Tegaza. Overall, the finding of this suggests that aloe vera Arachid date syrups offer promising functional food with potential benefits for both nutritional value and shelf life stability. For more details, you can find this work in the Journal on Nutrition and Santé. for Algerian journal. And this is my area interest, pathophysiology, pharmacology, phytochemistry, and nutrition. And thank you for attention.
We'll be trying to connect again with Jose Juan Santana. Are you hearing us?
Yes, I hear you. Thank you.
Can you please try share your screen? And if it's not possible to get your shared screen, that maybe we'll be asking you to send the presentation to the organizing committee, and then we'll present it here on your behalf. And we'll be talking, because we hear you very well, but it seems that there are a few issues in sharing your screen.
Right.
Please, can you give it a try?
Yes.
Thanks.
I think maybe it's not possible to move the slide. Could you please move from your side, please? Yes.
Jose, we still don't have your presentation. Can you share your screen?
Yes,
but it's not yet possible to have anything from your side. We are still waiting for you before introducing you and listening to your presentation. Now we see something. Can you share and put it in full screen? It seems working out.
Yes.
Okay, go ahead and put it just please in full screen, and I will let you know when you will be starting. Can you put your presentation in full screen, Jose?
Yes, it's a full screen, but the problem, Mr. Chairman, is I cannot move the slide. I don't know what is the problem. Please, would you move my slide from your side, please?
Can you send us the presentation?
Yes, I say.
What we will be doing, there is a last oral presentation of 10 minutes. We will be starting by that, and then we will come back to you.
Okay, perfect, right, no problem. It's okay.
Being here connected, thanks.
No problem.
Thus, we'll be moving to the presentation from Mohammed bin Hula. You are here? Okay, good. And then we'll come back to the conference from Jose Manuel. We are very sorry for these technical issues.
Okay, no problem, it's okay.
Mohammed bin Hula will be talking about the production, purification, and characterization of lipase from Streptomyces strain. Thus, the floor is yours in 10 minutes, please. Thanks.
Thank you, sir. Bismillah ar-Rahman ar-Rahim. Good morning, everyone. I am Dr. Benhouna Mohammed, a researcher at Agri-Food Technologies Research Center in Bejaïa. It's an honor for me to present you my work titled Production, Verification, and Characterization of lipas from Streptomyces strain. So this work was carried out during the preparation of my thesis. So olive oil industries generate a massive amount of byproduct, particularly olive wastewater. So this effluent poses a serious ecological problem due to its high organic load. In front of this, the valorization processes are often too costly, and the current disposal methods are inefficient. So this situation led us to the central question. Can we transform these environmental pollutants into valuable resources? So we hypothesize that the high lipid content of olive oil wastewater represents a valuable resource for the production of a high valuable compound such as lipases. So in this aimed our work. So our work was driven by three clear aims. Firstly, the use of the olive wastewater as a main substrate for fermenting by Streptomyces. Secondly, to employ a statistical model in order to optimize the culture conditions of culture condition for maximum lipase production. Thirdly, to purify and biochemical characterization of the lipase produced and to assess its industrial potential. So to quantify the lipolytic activity, we use the titration method. So it is a technique based in measurement of the amount of sodium hydroxide required to neutralize the fatty acid released from the substrate used. So beyond one factor at time optimization, we implemented box Duncan design for the optimization of the lipase and to understand the complex interaction between different parameters, such as temperature, pH, incubation time, inoculum size, and initial concentration of olive wastewater, and to find the global optimum of lipase yield. After the production, so we purified the enzyme using a multi-step process involving ammonium sulfate precipitation, followed by desalting and size exclusion chromatography. After that, SDS page or electrophoresis, SDS page and zymogram were performed in order to estimate the molecular weight of the enzyme and to confirm the purity and the activity of lipase. So regarding to the characterization, the optimal temperature was determined by the incubation of the enzyme at different temperatures ranging from 20 to 70 degrees Celsius, while the thermostability was assessed by extended incubation up to 6 hours, followed by periodic sampling. The pH profile was established by using 3 buffer system covering the pH from 3 to 8. Concerning the enzyme stability in organic solvent, it was assessed against several solvents, such as benzene, butanol, toluene, etc. And metal ions effects was examined in 2 different concentrations, 10 and 20 millimole. So additionally, enzymes tolerance to inhibitor and detergent was estimated by the exposure of the mixture for 1 hour at room temperature. So all these assays were conducted under standard lipolytic activity conditions with relative activities calculated against control measurements. So this table shows the findings of the lipase production after statistical optimization using box Benken design. And the best activity was observed at the 15th test with 8.4 unit per annum. This is the table which shows the model validation. As you can see, the predicted value was in very agreement with experimental values, and the standard deviation was very small. So this strong correlation between prediction and reality confirms that our model is reliable. This is the three-dimensional plot showing the significant interaction between K factors. So this representation allowing us to pinpoint the ideal condition for maximum production. Now we move to the purification. So this chromatogram shows the elution profile of lipase on Cefadex G25 gel, while this one represents the elution on Cefadex G150 gel. After the measurement of the absorbance and lipolytic activity, so the fractions of the main peak were collected for later use. The purification was successful, and we achieved a high level of purity. So according to this table, we could decreasing the purification from 100% to 19%, while the purification factor was increased from 1 to 80.51 times. So this step was very essential to subsequent the characterization. When the SDS page was carried out, so one single bond corresponding to the purified protein was detected in the gel at the white molecular 22 kilodalton. Unfortunately, our zemogram was unsuccessful, so potentially due to the use of an unsuitable substrate for the detection of this lipase. So to summarize the enzyme's character, it's moderately thermostable and the most active in body temperature and neutral pH. This bacterial lipase shows a remarkable resilience against several solvent, chemical agents, and metallic ions. So this profile makes it an excellent candidate for biotechnological application. So in conclusion, we have successfully developed a process to valorize polyphenol live wastewater into a high-value lipase using streptomyces strain, and this represents a meaningful step toward a circular economy. Looking forward, our perspectives are exciting, so it would be desirable to clone the gene encoding for the lipase into some hosts such as Escherichia coli in order to increase our production. So three-dimensional modeling is suitable for the enzyme in order to understand the structure-function relationship. And so the sequencing of the intergenome of the strain will enable us to fully understand its characteristics, especially the resistance to polyphenols. So finally, if it is possible to do the scaling up of the enzyme, moving to the laboratory scale to industrial scale. Thank you for your attention, and I am open for any question.
Thank you, Mohammed, for this nice talk. Now we will listen to the short biography of Professor José Juan Santana Rodriguez.
José Juan, are you here?
Yes, right.
Yeah.
I am here.
Okay, thus we got your presentation. Our colleague will be presenting you first, and then at each time you will just tell her to move to the next slide, and likely you will be presenting your presentation.
Okay.
Yeah, that's
right. Okay. Thank you, Mr. Chairman and Ms. Chairwoman. You heard me?
Jose, yeah, we are hearing you, but just give us 2 minutes to present you first, and then the colleague will tell you when you will be starting. Thanks.
Okay, perfect. Thank you.
Okay, so Professor José Juan Santana Rodriguez is a full professor of analytical chemistry and director of the Institute for Environmental Studies and Natural Resources at the University of Las Palmas de Gran Canaria in Spain. He is the author and co-author of more than 160 scientific papers, 12 book chapters, and 1 book, and has presented over 240 conference communications. Dr. Santana Rodriguez has led 25 research projects at European, national, and regional levels and supervised 13 PhD theses. His research has earned him recognition from the Royal Academy of Sciences of the Canary Islands and the University of Las Palmas de Gran Canaria Award for Scientific Career Achievement. His current research focuses on the optimization and application of analytical methodologies for detecting organic pollutants, monitoring contaminants in marine environments, and studying emerging pollutants and microplastics in aquatic and terrestrial systems. He will today present us online a lecture entitled The Importance of Analyzing and Monitoring Organic Compounds of Emerging Concerns in Trade Wastewater. Can you please start? I will pass your slides.
Okay, thank you. Thank you for this presentation, this kind of presentation, and also Mr. Chairman, Mr. Woman, to give the opportunity to share with you, all of you, the work at the University of Las Palmas de Gran Canaria in the field of environmental analytical chemistry. Also, I would like to thank so much to Dr. Mohamed Khalif for give me this opportunity, and also send a kind regard for my colleague and friend at the University of Bejaïa. The first question is why this subject in the context of the conference? This is a very important reason, because in many places, in many occasions, we use treated wastewater in order to irrigate the soil and the plant in agriculture. Then I would like to show you different case study that we have worked at the University of Las Palmas related with the contamination of organic emerging contaminant. Next slide, please. Okay, some word about organic emerging contaminant. Please, next, please. Next. Okay, perfect, right. Okay, the organic emerging contaminants are compounds of different origin in nature, and the main source of wastewater treatment plant from the effluent of this kind of plant. Also, there are many compounds which consuming is very high, and then we can call pseudo-persistent contaminant. Another characteristic is normally we find this kind of contaminant in very low concentration in different environmental compartments, normally in the order of ppb or ppt. There are many international study about the presence of this contaminant in different natural systems. Next slide, please. Next, please. Right. Thank you. Okay. Why we study this kind of contaminant in the environment, and for example, in wastewater for the use in agriculture? Because this contaminant produce negative and adverse effect in human and wildlife in many organisms. And the last characteristic is usually these contaminants are not regulated for national or international rules. Only there is a call watch list from European Union that include 23 kind of emerging contaminants that include pesticide, UV filter, and different pharmaceuticals, for example, antibiotic or beta blockers. Next slide, please. There are many families of organic contaminants of the emerging concern. For example, pharmaceutical residues, detergent, personal care product, flame retardant, industrial agent and additive, abuse drug, hormonal compound, and also now micro and nanoplastic nanoparticle or ionic liquid. In this study, I will present an overview of the result studying pharmaceutical, detergent, personal care product, and hormonal compound in wastewater from wastewater treatment plant. Next slide, please. Next, right. Yes, the general objective of this study, first one is optimize the analytical methodology to determine this compound in wastewater. Okay, then we can optimize the extraction, pre-concentration, and then the determination using liquid chromatography or ultra-high performance liquid chromatography with mass spectrometry in tandem. And the second objective, in the general objective, is the monitoring of the wastewater treatment plant in a island of Canary Island. It's called Gran Canaria. Is one of the most important islands in this Spanish archipelago. Next slide, please. Next, right. Yes, the first family that we have studied are antibiotics, mainly fluoroquinolone. This compound can be synthetic from quinolone family with a fluoro group attached the central ring system, typically at the 6th position. We can use this antibiotic for the human medicine and veterinary for the treatment of many kinds of infection. For example, urinary tract, respiratory system, and typhoid fever. Next slide, please. We can see the structure of this fluoroquinolone, levofloxacin, norfloxacin, enrofloxacin, ciprofloxacin, or sarafloxacin. We can see the different structure of this compound. Next slide, please. Okay, other family that we have studied are anti-inflammatory compound, for example, ketoprofen, naproxen, analgesic like metamizole, regulator of cholesterol, benzafibrate, beta receptor antagonist, atenolol, antidepressant like fluoxetine, stimulant, the paraxanthine, you know, is the one metabolite of the caffeine, or some anticonvulsant like carbamazepine. Next slide, please. Okay, we can see the structure of this different compound, this pharmaceutical. Okay, we can see different structure, very different structure. Next slide, please. Next slide, please. Okay, yes. Another family that we have studied are detergent, mainly the compound octiphenol and octiphenol polyethoxylate and nonylphenol polyethoxylate, where we can see or where or because or why we can find this kind of compound, for example, in agriculture soil or in plant, because these compounds are used as detergent or fuel additive or in lubricant. we can see the structure of this compound, where N vary between 1 and 20, and L are octyl or nonyl function group. Next slide, please. Okay, we have studied this compound in 3 wastewater treatment plants in the Canary Islands. The Canary Spanish archipelago with a population of more than 2 million of inhabitants, and focus on the Gran Canaria Island is on the right of the slide with a population of almost 900,000 inhabitants. Next slide, please. Okay, we have studied at the beginning 2 wastewater treatment plants with different characteristics about the population, the inflow, and the treatment process. In this case, both wastewater treatment plants use one activate slide and one membrane bioreactor. The monitoring was for 6 months. Next slide, please. Okay, what have been the result? Regarding the fluoroquinolone, the antibiotic, we found only 2 antibiotics, levofloxacin and ciprofloxacin, at very low concentration, maximum around 14 nanogram per liter. In other words, 14 ppt are very low concentration. Next slide, please. Okay, regarding the first group of pharmaceutical, we found all these pharmaceutical compounds with concentration higher than fluoroquinolone, with maximum approximately 19 ppb microgram per liter, 90 ppb microgram liter if we compare with the fluoroquinolone. Next slide. Regarding the other pharmaceutical, we found 3 pharmaceutical compound, atenolol, metamizole, and parsantin is a stimulant. The concentration around maximum 12 microgram per liter, 12 ppb. Next slide, please. Okay, and finally, regarding the alkylphenol, we found all the alkylphenol polysaccharides study in all wastewater treatment plant with concentration, maximum concentration around 500 nanogram per liter, approximately 0.5 ppb. We can see the variable of the concentration of this compound in both wastewater treatment plant. Next slide, please. Okay, next. Next. Right, thank you. Okay, the third family that we have studied in this organic compound have been UV filter. Why? Because the UV filter are used in many additives used in sunscreen and in other personal care products, for example, fragrances. Also have been used in clothes, in painting and plastic, and in order to avoid the photodegradation, the polymers and pigments. The most important characteristic of this compound is they absorb UV light between ultraviolet and visible spectrum. In other words, absorb the UVA and UVB rays. And also, finally, this compound have used like stabilizer of other personal care products. For example, gel for shower, shampoo, sunscreen, so on. Next slide, please. Okay, according the European regulation, are allowed 27 UV filters in personal care products. 2 inorganic, titanium and zinc oxide, and 25 organic UV filters. Then we have studied only these 25 organic UV filters. Next slide, please. Okay, why do we study this kind of compound? Because of their physicochemical properties. solubility, lipophilicity, and stability against biodegradation. What is the result? We can find this compound in different natural ecosystems, natural water, sludge, marine environment, water, sediment, biota, and also in wastewaters. Next slide, please. What is the effect in the environment of this kind of compound? For example, can produce mortality, malformation, alteration of mobility of embryo and adult, defective incubation, alteration of hormone and reproductive processes, in other words, endocrine disruption, alteration of muscular neural development, and one important effect is the coral bleaching. For example, you can see the coral bleaching in coral reef in Australia, and so on. But this effect can be transferred, moved, next slide, please, through different, the chain, the natural chain, for example, from plankton to marine mammals. In other words, this kind of compound can produce bioaccumulation and biomagnification in marine organisms, for example. Next slide. Okay, we have studied the wastewater from 3 wastewater treatment plants in Gran Canaria Island in effluent and effluent for 6 months. Next slide. Okay, you can see the map of this island, and then the location of 3 wastewater treatment plants. In the north of Gran Canaria, Las Palmas de Gran Canaria is the capital, is the biggest wastewater treatment plant. Arinaga in the southeast, and in the south, San Bartolomé de Tirajana. Okay, next slide. This wastewater treatment plant have different characteristics regarding the inhabitant equivalent and the same secondary treatment, use activate sludge. Next slide, please. Okay, we have studied 8 very common UV filters belong to the different family: benzophenone, salicylate, cinnamate, camphor derivative, benzotriazole, dibenzoyl methane derivative, and krylene. We can see the abbreviation and different physical-chemical characteristics. Next slide, please. Okay, it's important, the analytical methodology. Then the first step always is the optimization of the extraction using solid phase extraction and determination with ultra-high performance liquid chromatography with mass spectrometry. Why? Because the concentrations are very low, and then we must use a very selective technique. Next slide, please. Okay, the result in the influence. Okay, then we can see in this slide an overview of the positive case where we can find this compound. For example, in wastewater treatment plant, a 90% of the positive case, in the wastewater treatment plant 1, 75, and in the case of wastewater treatment plant 3, 67. Okay, benzophenone, octocrylene, dibenzoyl methane derivative, and cinnamate were detected in all samples with maximum concentration of octocrylene, benzophenone 3, and the dibenzoyl methane derivative. Please take attention. Octocrylene, OC, and benzophenone 3 are the most common UV filter used in sunscreen. Then, after the treatment, we can see a reduction of the positive case. In the case of a wastewater treatment plant, a reduction to 67. In the case of wastewater treatment plant 1 till 58, and in the case of wastewater treatment plant 3, a positive case or reduction until 58%. Okay, the benzophenone 3 and also the dibenzoyl methane found in all wastewater treatment plants with highest concentration for MVP is a benzotriazole. Benzotriazole UV filter is very common as well in sunscreen product. Next slide, please. Okay, in this slide, we can see the removing rate after secondary treatment in these 3 wastewater treatment plants. On the left, we can see the reduction of the concentration of benzophenone 3 in the influent in dark bar compared with the concentration in the effluent is the small light bar. On the right, we can see the removal rate for this compound, benzophenone 3. We can see the practically all the removal rate are higher, 70%. This is a very good removing efficient in the case of this compound. In the case of other compound, like HMS is homosalate, DTS are benzotriazole, and cinnamate and camphor vary between 70 and 100%. Of course, this is important. Depending on the treatment and depending on the compound, the efficiency in general speaking, depend of the compound and the treatment. Next slide, please. Okay, finally, the last family that we have studied belong to the hormone, specifically steroid hormone. The steroid hormone can be natural and synthetic source. Normally produce endocrine disruptive, a very, very low concentration, ppb or ppt, nanogram per liter or microgram per liter. This compound have used extensively in medicine and veterinary, also in agriculture. This compound are not allowed in the European Union for other use, for example, for livestock feeding. We have studied 4 families, hormone steroid hormone, estrogen, androgen, progestogen, and glucocorticoid. Next slide, please. Okay, what is the effect in organisms? For example, this hormone produce change in the sex. In other words, hermaphroditism. Next slide, please, in order to see the other row. Okay, anyway, this change of sex produce unisexual communities, male and female, and also can produce negative effect in tissues, for example, in the brain and in the bone. Next slide, please. Okay, you can see very quickly the skin, how the steroid hormone reach effluent wastewater treatment plant for natural, synthetic, human, veterinary use, so on. This is an overview of the way, or in other words, how this compound can reach the different environmental compartments, include wastewater, and for example, at the end, agriculture soil or plant or fruit. Next slide. Okay, we can see the structure of the 15 hormone study in our laboratory belong to estrogen, androgen, progestogen, and glucocorticoid. Next slide, please. Next. Okay, next, right, perfect. Yes, okay, you can see at the top of the slide the optimization of the analytical methodology, limit of detection, recovery, reproducibility, so on. An application, the monitoring. We have studied in this part 4 wastewater treatment plants in Gran Canaria, 2 in the north, 1 in the center, and 1 in the southeast. And we found hormone only in 2 wastewater treatment plants, the number 3 and number 4. In the case of the wastewater treatment plant number 4, we found 4 hormones: estrogen, testosterone, bordenone, and progesterone. Well, to finish my lecture, please, next slide. Okay, a general conclusion, we have demonstrated the presence of different families of organic emerging contaminant in wastewater in the Canary Island with the wastewater treatment plant with different characteristics. Yes, from here, this compound can reach different environmental systems: terrestrial, agriculture soil, plant, fruit, and then in marine ecosystem. Okay, therefore, we need to control the presence of this compound in different compartments, in water, in soil, and food. Yes, next slide, please. Of course, behind this result, there is a human team of my collaborator at the University of Las Palmas de Gran Canaria, permanent staff, postdoc, student, PhD student, and undergraduate student. And then, next slide, please. This is, next slide. Okay, this is the building of our rectorate, and then all of you are invited to visit our university in different fields of the scientific or humanity fields. Finally, thank you so much for your attention. Please, if you have some questions, you can write by email through the organization of the conference. Many things.
Thank you, Jose Juan, for this pertinent and interesting presentation about wastewater treatment. And I think this finished for the intervention for the communication and started the debate about for the different communication for the second part. If there are questions in the audience.
Can we invite all the speakers, please?
Yes, sorry, I forgot this part. the principal, the principal person, please come here, and if the audience have the question. Any question? Yeah. Please, Busaid, can you take the microphone, Busaid?
Okay, thank you for giving me the opportunity to ask a question, just maybe naive, to this gentleman. In his conclusion, you said that you are planning to develop an in silico 3D modeling. Could you tell us more about this?
Mohammed, please, micro. Open the micro. Just in the button, yeah, down.
Thank you, Sir Bejaïa, for your question. In fact, we are expecting to do the modelization by docking molecular. We will use the docking molecular in order to discover the relationship between the function and the function and the, how to say, the structure. The structure and the relationship between the structure and the function of this enzyme.
So you are planning to use molecular dynamics, right?
I did.
Sorry? Okay, thank you.
Other question?
Thank you for all the speakers for their very interesting communications. I have just a small question for Mohammed. Please, did you perform a formulation for the application of your enzyme?
The formulation?
Yes. I mean, did you try to perform a formulation for the application of your enzyme, performing enzyme? Thank you.
Thank you, Madam Jenny, for your questions. In fact, I tried in the laboratory. It is just attempts, some preliminary attempts to apply this enzyme in detergents. According to its characteristic showed previously, if you followed me well, according to the pH and resilience against several solvents.
Yes. In which form did you plan to, or do you plan to use directly the microorganisms or the enzyme, on the purified enzymes? I mean.
I'm expecting to incorporate it in detergents.
The purified enzyme?
Yes, the purified one.
Okay.
Yeah, the purified form.
Other question?
Thank you for this nice talks. First question for Professor Rodriguez. Do you think that we can upstream to control the caustic pollution, especially for cosmetics, to talk with the cosmetics industry and let's say make them put less UV filters, so to act upstream at the production? And then second question for Dr. Benhoula. Have you already made collaborations to do molecular docking? And what is your plan to do the whole genome sequencing? Thank you.
Dr. Rodriguez, are you here?
Yes, okay. Thank you for the question. But please, could you repeat, or what is the meaning of your question? Okay, what is the monitoring in the air, the UV filters? Is on screen?
Yeah, sorry. So for the UV filters, do you think it will be, can we act upstream so we can discuss with the cosmetics industry to make them put less amounts of these UV filters in their products, and so in a way solve this problem?
Yes, right. Okay, I understand. Okay, right. This is a good question, okay, because maybe there is a contradiction, okay, if we don't use a UV filter for the protection, okay, we can have a negative effect in the skin, okay. What is the alternative? Okay, the alternative is use a greener or ecological sunscreen with no chemical compound. Okay, for example, there are several countries where the chemical UV filter are not allowed in the beach, for example. Okay, then there is a competition between the big company that produce UV filter, chemical UV filter, with the company, a small company that produce ecological UV filter. Okay, then this is a big problem in the society. Okay, but anyway, the most important is we must have a protection of the ray, of course, from the sun. This is important. But anyway, even the big company are trying to use less chemical UV filter in order to substitute by ecological or natural filters. For example, even the aloe vera can be used like protector. But anyway, it's a very important society problem. Okay. Thank you.
Firstly, thank you for your relevant question. To be honest with you, Mr. I didn't yet use or studied the molecular docking, but it was a suggestion of some jurists during my dissertation. So I was with my supervisor, she is here, and the co-supervisor, Madame Azouz. They suggest to me and we are thinking to do some studies in this kind.
So I think we have to talk after lunch to do some work together.
Please, I didn't hear you.
Sorry?
I didn't hear you well.
Sorry. So I think we can make collaboration. We work together.
Collaboration? Yes, of course. Of course, you are welcome, Mr. We are here we are here for this kind of collaboration, this kind of materials. Thank you for your suggestion.
Thank you, Mohammed. I think Professor Ammar have the question.
Thank you very much. My question is for Lamia about the microflora that you have checked in your rob. Why you didn't interest to Staphylococcus? It is the.
Okay, thank you. Yes, we did. We check Staphylococcus, Streptococcus, Clostridium, and total germ, mold, yeast. And the final products, we didn't find any. We just find only yeast and total germ. Maybe the total germ and yeast, maybe it's when we work in. Have a contamination because while we preparing the syrup, add the supplements or not supplemented, we are very prudent, very to check be sterilized, to work in the sterilized ambience. For that, maybe for that we don't find the Staphylococcus, Clostridium, and all this. And we have other suggestions. Maybe also the secondary compound of the aloe vera and the concentration of the syrup, because this is very how to call that, compact. So for that, don't yes, not help the germ to grow inside the product, the final product.
And what about the best product? You have checked.
The best product is to say syrup supplemented by aloe vera, but the low percentage, because we keep in mind the aloe vera have a bitter flavor taste. So for that, in the past, this is a work did in the past. So I didn't continue in the same topic, but when I thinking, I thought if I continues and try other percentage supplementation of less than 2.5% of aloe vera, maybe it will be a new product. So, thank you.
For Professor Emna, I would like to know if there is an industrial application of olive meal wastewater in Tunisia.
Yes. Because of time, I couldn't present all the work, but our work was held by an industrial. I show some picture to let you know that there is an application, and the industrial was very interested to give us first the waste, solid waste, grignon, and then he takes
olive waste margin.
Imagine we have another. We take some part to include them in the compost, and it works. Then we have another work. It is about the spreading of margin in the field. And then there is, after this work, we have experimented many different rates of 50 cubic meter per hectare to 100 and 200 cubic meter. And then we follow all the vegetal life and cycle. And we concluded that the best quantity that will not harm for soil and the plant will be 50 cubic meter per year. Then we contacted the government, and there is a law which allow all agriculture to use margin for spreading olive trees, and then they have to don't exceed 100 per 2 years. That means 1 year they use it, and they let 1 year without using, and then next year they can use. Yes. And there is an improvement of the yield in olive oil production.
Yes, I know you have a huge quantity of olive Midwest water to produce metabolites, enzyme, and others.
Yes,
okay.
Yes.
Okay.
Many.
Thank you.
Welcome.
There are other questions? Okay, I have the question for Amina. I don't know why you choose the tip of the dot. In the picture, it's very good quality for to consume and use it for, it's not really agro-industrial waste, no?
Talking about the
yeah, why you check exactly this tip of the dot. It's good for directly to consume. It's not for to valorize it.
Yes, just experiment to enhance the product. We have many kind. You know, in Algeria we have many kind of palm date. So Hmira is the most known in the West. Tagaza also have some kind less known. So I chose those. As in the South Algeria, people in the South Algeria date fruits like that is very important, and the sweetest products after the honey. So here just is experiment to enhance, to give something new, and try to see the combination between, like aloe vera, we have also more valoriser l'aloe vera et l'utiliser. Est-ce que c'est possible de combiner les deux, combiner all the two products like this? And in Algeria, in North Africa, and almost don't use aloe vera for as food. Just for cosmetic, and other culture in Asia use it for food, added as in the food. But, so I tried this experiment and see what I can have.
Yeah, because if you compare the cost for the using the that olive oil, it's low cost. But if you product the product based between the aloe vera and the syrup of the dot, it's very expensive, no?
No. Because the syrup, syrup in Algeria have a period when the syrup, this syrup don't have just homemade. Now we start to see the industrial syrup. Now in this part, in the second part, but it's not really, c'est pas vraiment propagé en Algérie beaucoup. Donc ce qui reste, c'est ce que juste un essai. C'est juste un essai pour voir, valoriser les deux. No? Yes, for concepts, a new concept
That's right.
Thank you. Other question?
Good morning, everybody. My question is for the Professor Emma Omar. First, thank you for your presentation. And my question is, we often confuse manure and the compost. Can you explain the difference, please? I am not scientist, donc je suis pas une scientifique, donc.
Excuse me, you confuse between compost and
manure.
Minor.
I don't know if my pronunciation is correct.
The minor is the product that is issued from animals, and it is not fermented and not manipulated. It's like this, the raw material. And compost is a process, is the product issued from a process, biotechnological process, where microorganisms are acting, where we need to have equilibrium between carbon, nitrogen, water, and we have to add oxygen to let microorganism working for a few months, and then there is the degradation and the biotransformation of this substrate, carbon, nitrogen, to give us fulvic acid, humic acid, fulvin, and these are humic substances which are very good for the soil. So if you use manure alone or compost, the result is not the same. The compost is more performant and more rich as a fertilizer. Okay, have I explained? Yes.
Okay, thank you all for attending this session. As we arrived at the end, I will be moving to take lunch. Thanks everyone, and thanks for the speakers for being here. Thank you.
كتبين بالعيا وغير مرة بزاف. ها؟
Your attention, please. Welcome back, everyone. We now begin our afternoon topic with topic 2, agroecology and sustainable food systems. So this session will be chaired by Dr. Lisa Warabi and moderated by Dr. Mohammed Khalil Mellal and Dr. Samiran Grishi. Our first plenary lecture will be given by Professor Boualem Harfi from the Biotechnology Research Center in Constantine, Algeria. So I will first introduce him. Professor Boualem Harfi is a senior researcher at the Research Center of Biotechnology in Constantine, Algeria. His work focuses on plant biotechnology, particularly tissue culture, genetic improvement, and the valorization of agrobiological resources. Through numerous national and international publications, Professor Harfi has contributed significantly to advancing sustainable agriculture and the preservation of Algeria's plant biodiversity. His research combines innovation and applied biotechnology to enhance agricultural productivity and resource management. Today, he will deliver a lecture entitled Agrobiological Resources, Seeds and Seedlings in Algeria, Biotechnological Approaches for the Characterization and Valorization. Professor, you have the floor.
Thank you. Good afternoon, everybody. First of all, I'm happy to be here with you to present a part of my work. I thank a lot the scientific committee and the organization committee to associate me with this event. I haven't a good fluent English. For that, I will present my work in French. Donc, je vais parler un peu de la valorisation des ressources biologiques, tout particulièrement les ressources agrobiologiques. Donc, je commencerai par un préambule. En fait, en Algérie, comme ça se passe partout dans le monde, les ressources biologiques, elles sont soumises à une réglementation, on va dire. Donc, pour appeler une variété, il y a certains critères. Donc, en Algérie, comme partout dans le monde, on parle de variété quand on apporte à cette ressource biologique un plus. Ça veut dire quelque chose de nouveau qui n'existe pas ailleurs. Donc, sur ce principe-là, je vais essayer de vous présenter trois cas d'études où vous allez voir, on peut valoriser les ressources biologiques de différentes manières. Mais je vais insister sur un point très particulier, c'est que comme je travaille dans un centre de recherche en biotechnologie, forcément je vais parler comment on peut apporter de la nouveauté à de la ressource biologique avec des études biotechnologiques. Donc pour le premier cas d'étude, c'est à peu près ce que je viens de dire. Donc les objectifs, c'est en fin de compte comment valoriser ces ressources avec des outils biologiques tout en prenant en considération la nature de la ressource, donc est-ce qu'elle est locale, endémique, l'état de connaissance, est-ce qu'il y a beaucoup de travaux sur cette ressource ou non, et aussi est-ce que c'est une ressource complètement industrialisée ou une ressource qu'on ne connaît pas beaucoup. Voilà, donc à peu près en prenant en considération ces trois aspects. Donc pour que je puisse illustrer ça, j'ai pris trois cas d'études qui sont en cours au Centre de recherche en biotechnologie de Constantine. Donc pour le premier cas d'étude, je vais parler un peu de la culture de la fraise. Et vous allez voir, pour chaque cas d'étude, j'ai essayé de faire un petit rappel sur l'état de l'art, donc qu'est-ce qui se fait dans le monde. Après, la question de recherche qu'on s'est posée concerne cette ressource en particulier et les principaux résultats que nous avons obtenus. Donc pour le premier cas d'étude, c'est en fin de compte, nous avons un objectif d'inscrire au moins une variété de fraisiers en Algérie. Donc, juste pour vous donner un peu l'état de l'art de cette question, il faut savoir qu'en Algérie, on produit de la fraise. Ça veut dire que nous avons de la fraise sur le marché algérien. Mais malheureusement, plus de 90% de cette fraise qu'on consomme, elle est issue de plants, donc les plantules qu'on cultive, d'importation. Mais en parallèle, on a des populations qu'on n'a pas encore appelées variétés, parce que d'un point de vue agronomique, c'est important l'appellation variété. Donc d'un point de vue, on va dire, reconnaissance, la variété algérienne, elle n'existe pas encore. Mais en parallèle, on voit qu'elle a une certaine réputation. Tout le monde parle de la variété de Skikda. Et en plus, elle s'appelle la variété Russikada. D'ailleurs, en 2021, il y a eu un concours aux Pays-Bas où elle a eu un prix. C'est-à-dire, on sait qu'il y a potentiellement une variété, mais elle n'est pas inscrite. Donc nous, le travail qu'on essaie de faire pour cette ressource-là, c'est d'essayer de voir qu'est-ce qu'elle apporte de différent, et surtout, est-ce qu'on peut l'inscrire sur le catalogue national dans un premier temps, et une reconnaissance internationale sur un second temps. Oui, je peux s'il te plaît passer. Donc la question qu'on s'est posée, parce qu'il nous faut une question scientifique, on s'est dit, est-ce que on importe des plants de variétés reconnues, très hautement productives, espagnoles, italiennes, et on le cultive, ou inversement, on ne va pas valoriser notre propre ressource locale qui a un potentiel. Après, est-ce qu'on arrive au même niveau que la ressource internationale ou pas, ça c'est une autre paire de manches. Mais la question scientifique qu'on s'est posée, on a commencé à poser cette première question. Donc pour y répondre, on a voulu caractériser d'un point de vue phénotypique, d'un point de vue génétique, la ressource algérienne. Donc, tu peux passer, s'il te plaît. Mais on ne s'est pas arrêté là. Ça veut dire qu'on s'est dit, pour qu'on puisse dire que notre variété est intéressante, il faut qu'on ait des moyens, des outils qui nous permettent de la multiplier à très grande échelle. Donc c'est toutes ces questions-là qui viennent ensemble. La première surprise qu'on a eue quand on a commencé le travail, c'est qu'on est parti à prospecter sur terrain avec l'idée, en Algérie, on a potentiellement une population au niveau de Skikda. Cette population, elle est potentiellement différente parce qu'elle a gagné des prix. Donc on est parti avec l'idée qu'il y ait une seule population. Donc quand on a commencé avec les prospections, on s'est rendu compte, comme premier résultat déjà, il n'y a pas une seule population, mais on a trouvé 6 populations distinctes. D'ailleurs, ce que je vous montre sur les photos, c'est des photos qu'on a prises sur terrain chez 4 familles d'agriculteurs, parce qu'on s'est rendu compte que la fraise, au niveau de la wilaya de Skikda, elle se cultive d'une manière traditionnelle, et c'est des familles qui entretiennent 6 populations. Donc on a trouvé 6 familles, 6 grandes familles, dans des endroits un peu éloignés au niveau de la wilaya de Skikda, qui cultivent d'une manière traditionnelle la fraise. Donc on s'est dit, potentiellement, on a peut-être plus d'une variété. Là, je vous montre 4 champs, mais en réalité, nous avons trouvé 6 populations possibles. Donc, c'est possible. Donc, le premier travail qu'on a essayé de faire, là, j'ai pris juste un petit exemple pour vous démontrer un peu, c'est qu'il faut savoir qu'il y a des guides. Ça veut dire, pour chaque espèce, il y a le guide OPOV. Nous avons utilisé la référence 2012, qui nous montre, pour chaque espèce, comment la caractériser d'un point de vue phénotypique. Ça veut dire l'aspect externe taha, comment il se présente et comment la caractériser. Par exemple, sur l'exemple, il y a par exemple la première ligne, c'est un peu le port de la plante. Est-ce qu'elle est dense? Est-ce qu'elle est détachée? Après, il y a la densité du feuillage. Après, il y a la forme de la feuille. Donc, on a commencé déjà, on a ramené des échantillons. On a commencé déjà à faire ce premier travail. Donc, donc là, c'est juste un exemple. Vous voyez que la feuille, il faut l'aplatir parce qu'il y a certaines règles à respecter pour prendre les mesures. Donc après, ce que vous voyez sur la photo de gauche, c'est une photo sous l'eau. Parce que même le sens des poils du pétiole, ils interviennent dans la détermination de l'espèce. Donc il y a un certain nombre de données phénotypiques dans un premier lieu qu'on a collecté pour toutes les populations. Après, dans un deuxième temps, on est passé aux données génétiques. Donc, après, pour les données génétiques, on s'est intéressé aux SSR. Comme vous le savez, les SSR, c'est des marqueurs génétiques. Ceux qui sont développés, ce que vous voyez sur le tableau, ils codent, enfin, ils flanquent des régions connues qu'elles sont polymorphes. C'est-à-dire, potentiellement, on peut trouver de la diversité entre les deux parties flanquées par l'amorce utilisée. Donc là, on a utilisé 4 amorces, SSR, et sur les 4 amorces, les premiers résultats, ils nous ont montré une certaine diversité, mais c'est une diversité qui est un peu moyenne. Ça veut dire entre les 6 populations qu'on a étudiées, on a trouvé de la diversité statistiquement significative, mais d'un niveau moyen. Mais après, ça peut s'expliquer par le nombre d'SSR utilisés, parce que 4 SSR, c'est pas beaucoup. Donc on compte, Inch'Allah, aller un peu plus loin. Mais on ne s'est pas aussi arrêté juste à ce niveau. Il faut savoir que pour la fraise de Skikda, elle a un goût un peu particulier, mais ce qui la rend encore plus particulière, c'est qu'elle dégage une certaine odeur. C'est-à-dire une certaine odeur, d'après les données bibliographiques, il y a certains gènes qui codent chez la fraise pour une odeur qui est proche de la pêche. mais associé à un goût caramel. Donc, il y a 2 gènes spécifiques qui sont responsables de ça. C'est possible de passer. Donc, nous, on s'est dit, comme on a déjà noté ça, pourquoi pas ne pas chercher la présence ou l'absence de ces gènes. Parce que sûrement quand vous partez au marché, vous voyez de la fraise, mais vous ne sentez pas l'odeur de la fraise. Parce que c'est des fraises, elles ont perdu un peu ce gène. Pour notre étude, pour le premier gène, parce que nous avons étudié les 2 gènes, donc pour le premier gène, nous avons trouvé la séquence qui code pour ce gène. Donc normalement il est présent, mais sa taille, parce que nous avons fait une analyse de fragments. Donc quand on a fait une analyse de fragments, on a trouvé que sa taille elle est inférieure à la taille du gène qui est connu. Ça veut dire peut-être aussi, ça montre d'un point de vue génétique, il y a une diversité. Là, on est en train de séquencer le gène qu'on a trouvé. Et pour le deuxième gène, sur les 6 populations qu'on a étudiées, donc le gène qui donne un peu l'arrière-goût caramélisé. Donc pour ce gène-là, nous l'avons trouvé chez 2 populations, mais nous ne l'avons pas trouvé chez les 4 autres, enfin chez les individus des 4 autres populations. Ça aussi, ça nous encourage. Ça veut dire qu'on est au moins sûr qu'il y a 2 groupes ou 2 poules génétiques différents. Donc là, ce que je vous ai montré jusqu'à maintenant, c'est un peu la caractérisation phénotypique et la caractérisation génétique. Mais nous, on espère, parce que c'est un travail qu'on ne peut pas valider en une année. Ça veut dire qu'il faut le faire. La première année, on a fait tout ce qui est optimisation. La deuxième année, nous avons les premiers résultats. Mais maintenant, il faut le refaire au moins sur 3 ans, sur 3 sites différents, pour s'assurer que les résultats que nous avons obtenus sont stables. En parallèle, on a développé des moyens de multiplication massive. Parce que, tout simplement, la fraise dans le monde, elle ne se multiplie pas avec les stolons. Parce que la fraise, dans la nature, il y a le plant principal, ce qu'on va appeler le plant mère. Après, dans la racine du plant mère, il y a un stolon qui va apparaître. Le stolon, il va donner la plante fille. Comme vous voyez, c'est une multiplication végétative. Comme c'est une multiplication végétative, il y a un risque, s'il y a une contamination virale, la charge virale, elle va augmenter d'une population à l'autre. Donc, qu'est-ce qui se passe, qu'est-ce qu'on fait un peu partout dans le monde, surtout chez les pays producteurs de la fraise, exactement du plant de fraise comme en Espagne ou en Italie, c'est qu'on fait un assainissement. Un assainissement, ça veut dire qu'on fait débarrasser la plante de la charge virale. Il y a des techniques en culture in vitro végétale avec lesquels on peut faire ça. Donc nous, en parallèle de cette caractérisation génétique et morphologique, on a essayé de développer un système de multiplication maîtrisé. Donc ce que vous voyez sur les photos, donc sur la première photo, c'est une phase d'initiation. C'est-à-dire que je prends un peu le bourgeon de la fraise, je vais le mettre dans des conditions contrôlées, dans des milieux de culture spécifiques, et avec des régulateurs de croissance pour maîtriser le développement de la plante dans un environnement complètement contrôlé. Après, ce qu'il y a sur le deuxième point, donc c'est le traitement à froid. Un peu, c'est là où on a un peu l'originalité du travail qu'on est en train de faire, c'est que nous avons développé ce traitement à froid qui permet d'activer les méristèmes pour produire à la fin de la fleur. Parce que ça, c'est important. Je ne sais pas si vous êtes au courant, mais la fraise, c'est une espèce de la même famille que le pommier, que le poirier. C'est une rosacée. Et c'est connu pour le rosacée. Dans leur cycle de développement, ils ont besoin d'une phase de froid. Donc, on a essayé un peu d'imiter ça dans des conditions contrôlées in vitro. Après, ce qu'il y a sur la photo numéro 3, c'est un peu le développement et la multiplication. Il faut savoir, juste pour vous donner un ordre d'idée, c'est qu'un plant, après 3 semaines, il va me donner au moins 3 X plants. Ça veut dire qu'il va me donner 3 méristèmes distincts, de 3 à 6. Ça veut dire, pour juste vous donner un nombre de grandeur, ça veut dire que j'ai un plant principal, je fais l'initiation, un traitement au froid, au bout de 3 semaines, le plant que j'ai de départ, je vais obtenir 3. Après 3 semaines, je vais avoir 9. Après 3 semaines, je vais avoir 27 plants. Donc la façon de multiplier le nombre de plants, elle est exponentielle. C'est tout l'intérêt de la culture in vitro végétale. Mais une fois on arrive à obtenir le nombre souhaité, il faut faire sortir maintenant ces plants, ce qu'on va appeler des vitroplants, dans la nature. Donc il y a une phase qu'on appelle une phase d'acclimatation. C'est-à-dire une fois j'ai mes plants, Il faut que je les acclimate avant de les sortir. C'est un peu comme les enfants. Un bébé, il est dans une couveuse, on ne peut pas le sortir directement, il lui faut un certain nombre d'adaptations. C'est à peu près ce qu'on fait avec les plantes. Donc là, par contre, c'est complètement maîtrisé, c'est complètement développé. C'est-à-dire on sait assigner et multiplier les plantes de fraises. Donc si on trouve des variétés distinctes, on peut proposer à l'industriel, à celui qui veut, on va dire, multiplier, produire de la fraise locale, on peut lui proposer un système de production fiable et qui répond aux exigences internationales. Donc, en fin de compte, notre façon de valoriser, c'est de montrer qu'elle est différente et en même temps, on peut produire à grande échelle. Après, pour le deuxième cas d'étude, là, je vous ai parlé d'une espèce, la première espèce, la fraise, qui est mondialement utilisée. Il y a déjà des concurrents, on va dire. La deuxième espèce sur laquelle je vais vous parler, c'est un autre cas. Le cas là, je vais parler de l'arganier. L'arganier, c'est une espèce endémique. Ça veut dire notre démarche maintenant. On va utiliser toujours les mêmes technologies, les mêmes techniques développées au niveau de notre laboratoire, mais avec une démarche un peu différente. Parce que là, on va valoriser une espèce qui est endémique, qui n'existe pas ailleurs. Elle est présente principalement dans 2 pays en Afrique, de l'Afrique du Nord-Ouest exactement. Juste la première photo que je voulais vous montrer, parce qu'il faut savoir en Algérie, l'arganier se trouve dans la région de Tindouf. Mais Tindouf, on va dire, c'est la région où il se diversifie naturellement. Parce que maintenant, vous allez trouver de l'arganier de Tlemcen jusqu'à Adrar. C'est-à-dire, on l'a multiplié. Mais le berceau de diversification de l'arganier, il est au niveau de Tindouf. Mais après, généralement, le produit de l'arganier, c'est l'huile d'argan, qui est une huile à très haute valeur ajoutée, qu'on utilise beaucoup dans les cosmétiques. Donc, l'idée de valoriser cette ressource, premièrement, ça nous permet de la protéger, mais en même temps, ça crée de la richesse. Surtout, il y a tout un programme d'insérer la femme rurale, entre guillemets, et qu'elle soit indépendante via la récolte et l'extraction de l'huile d'argan. Donc, c'est à peu près l'idée qu'on aimerait bien faire dans ce projet. Et la deuxième information que je veux partager avec vous, pour ce projet-là, c'est un projet qui n'est pas uniquement projet CRBT. C'est dans le cadre d'une stratégie nationale de développement de l'arganier. Donc nous, on participe avec notre savoir-faire. Donc il y a d'autres partenaires qui ont d'autres parties du travail. Mais là, je vais essayer de vous en parler de ce que le CRBT est en train de faire. Ou la photo, je l'ai mis juste pour que vous ayez une idée de la grandeur de l'arbre. Parce que vous imaginez, il fait plus de 20 mètres de hauteur. Mais la photo là que je vous montre, c'est un arbre qui a plus de 200 ans. Donc ça veut dire que c'est une ressource une fois multipliée, une fois installée. Le retour d'investissement sur cette ressource, il est très intéressant. Voilà, donc là, comme pour le premier cas d'étude, on a fait un état de l'art. Donc je commence par vous donner une première information. Il faut savoir, jusqu'en 2021, l'arganier, il était reconnu uniquement comme ressource endémique du Maroc. Donc les publications, on dit l'arganier marocain. Après, depuis 2021, il y a pas mal de travaux en Algérie. Ces travaux, ils ont démontré qu'il existe de l'arganier en Algérie. Donc maintenant, dans les publications, on dit soit une ressource endémique à l'Afrique du Nord-Ouest ou algéro-marocaine. Donc là, c'est reconnu déjà comme ressource locale. Quand on a commencé à travailler, on a trouvé beaucoup d'études qui ont montré qu'en Algérie, on a au moins 11 écotypes. Qu'est-ce que ça veut dire des écotypes? Ça veut dire que c'est des espèces qui sont complètement différentes. Enfin, c'est la même espèce, mais avec des potentielles variétés différentes. Parce que d'un point de vue phénotypique, elles sont différentes. Mais il faut le prouver. Donc le travail qu'on est en train de faire, c'est exactement comme la démarche de la fraise. On va essayer de démontrer sur les 11 écotypes minimum qui existent en Algérie, est-ce qu'ils sont tous identiques ou ils sont différents? Ou peut-être on aura 2-3 qui sont identiques, les 2 autres qui sont différents. Donc, c'est possible de passer. Donc là, on s'est posé en quelque sorte 2 questions. Comment on peut propager ou multiplier à grande échelle l'arganier? Et quelle richesse ou quelle diversité génétique qui existe en Algérie? Donc, pour y répondre, donc pour la première question, donc on a un peu suivi le même cheminement du travail qu'on a fait sur la fraise. Mais c'est des milieux complètement différents, des équilibres hormonaux ou l'utilisation de régulateurs de croissance complètement différents, en même temps avec des stades de développement qui sont aussi différents, parce qu'ils sont spécifiques à l'espèce. Ça dépend de chaque espèce. Chaque espèce, elle a sa propre génétique. Et comme vous le savez, la réponse de l'espèce, c'est toujours la somme de 2 choses. le génome et l'environnement dans lequel on le fait, on obtient le phénotype, la réponse de l'espèce. Donc pour cet espace-là, sur la première photo, c'est la phase d'initiation. C'est-à-dire on prenait des branches de l'arbre d'origine, on récupérait des boutures, ce qu'on appelle nous des microboutures, qu'on mettait en culture. On a optimisé cette première phase, on l'a complètement optimisée. Après, ce qu'on voit sur la phase 2, c'est la phase de multiplication. Mais contrairement à la fraise, on arrivait à faire de la multiplication en une seule phase. C'est-à-dire on fait de la multiplication en même temps de l'élongation. C'est-à-dire on a un nombre de bourgeons qui augmente tout en obtenant des plantules. Alors que pour l'arganier, nous avons remarqué que quand il y a une phase de formation de bourgeons, il n'y a pas élongation. Donc la phase 2, vous voyez deux photos. La première, c'est pour avoir un maximum de bourgeons. Et la deuxième, c'est pour faire une élongation. Ça veut dire que chaque bourgeon, il va me donner une petite branche. Mais une branche à l'échelle de bocal. Donc vous imaginez un arbre qui fait 20 mètres de hauteur, on arrive quand même à produire sur un bocal qui fait 25 cm de hauteur. Donc ça, c'est assez intéressant. Après, pour la troisième phase, aussi contrairement à la fraise, l'arganier ne s'enracine pas directement. Même si on a le développement de la partie aérienne, ce qu'on appelle la cologénèse, il n'y a pas un développement racinaire. Donc nous avons développé un autre milieu qui nous permet d'avoir ce qu'on appelle de la rhizogénèse, c'est-à-dire développement de racines. Après, la dernière phase, c'est la phase d'acclimatation. Donc vous voyez sur la première photo un petit plant qui s'est acclimaté, c'est très bien. Mais malheureusement, jusqu'au niveau d'études qu'on est maintenant, je ne sais pas si vous arrivez à voir, mais il y a 6 pots à côté, c'est de grands pots. Quand on est passé à une échelle plus importante, là j'ai pris une photo, il y a 6 pots du même cultivar, donc du même écotype. Sur les 6, je ne sais pas si vous arrivez à bien voir, mais il y a 5 plants qui sont morts, et un seul qui a eu une croissance importante. Où le taux de réussite pour cette étude-là, ce cas d'étude, il est inférieur à 20%. Donc ce n'est pas satisfaisant. Donc là, on est toujours en train d'essayer d'optimiser la phase entre 3 et 4. C'est-à-dire la phase d'enracinement et d'acclimatation. Là, contrairement à la fraise, on a réussi complètement à acclimater nos plantes à un taux dépassant les 95%. Pour l'arganier, on est autour de 20%. Et quand on a cherché quand même dans la bibliographie, parce qu'on a commencé par cet état de l'art, on savait à la base qu'on allait être confronté à cette question. Parce que l'enracinement de l'arganier in vitro, c'est ce qu'on appelle une espèce récalcitrante. Donc c'est une espèce qui s'acclimate très difficilement, qui s'acclimate très difficilement. Donc on essaie quand même d'optimiser. Cette phase-là, on est en plein dedans. Voilà. Après, c'est possible. Donc là, ce que vous voyez, c'est parce que le cas de la fraise, comme c'est une espèce qui est très très étudiée, pour ces marqueurs moléculaires, particulièrement les marqueurs SSR, on en trouve beaucoup de marqueurs connus. Mais par contre, pour l'arganier, il y a moins d'études. Ça veut dire, qu'est-ce qu'on a fait nous? Nous avons pris les marqueurs qui existent dans la bibliographie pour évaluer la diversité, qui sont spécifiques à l'arganier. Nous avons trouvé dans la bibliographie, il n'y a que 5 marqueurs. Mais on ne s'est pas limité aux marqueurs de l'espèce, on est allé un peu plus haut et on a pris aussi des marqueurs de la même famille. Potentiellement, ils pourraient aider à caractériser d'un point de vue génétique l'arganier. Donc c'est pour ça que vous voyez en vert à la fin, Algae Tree Species, c'est pour l'arganier, alors que pour les autres, c'est la famille de l'arganier. Donc on est en train d'évaluer sa diversité d'un point de vue génétique. Donc, pour le troisième et dernier cas d'étude, je vais vous parler de la pomme de terre. Le cas de la pomme de terre, vous allez me dire, est-ce que nous, en Algérie, on va valoriser la pomme de terre? Il faut savoir que la pomme de terre, c'est une espèce qui est diversifiée en Amérique latine, en Chili, au Pérou. Ça veut dire en termes de richesse génétique, de biodiversité, elle est plus en Amérique latine. En Algérie, nous n'avons pas des variétés locales de pommes de terre. Donc j'ai pris ce cas très particulièrement pour vous exposer que même dans les cas où on n'a pas une ressource biologique qui est chez nous, on peut toujours participer à sa valorisation. Donc pour le cas de la pomme de terre, donc nous avons fait un état de l'art. On a choisi la pomme de terre parce que c'est une espèce qui est très consommée en Algérie. Comme je vous ai dit, elle n'est pas diversifiée chez nous. Mais qu'est-ce qu'on a fait? Nous avons pris 4 variétés qui sont dans des catalogues internationaux, mais c'est des variétés qui existent plus de 20 ans. Pourquoi c'est important cette information? Ça veut dire que c'est des variétés qui ne sont pas soumises à des COV. Les COV, c'est les conventions d'obtention végétale. C'est-à-dire que c'est des variétés libres. C'est-à-dire qu'on peut travailler sans problème. Donc, l'idée qu'on s'est posée, parce que la culture de la pomme de terre en Algérie, comme partout ailleurs dans le monde, elle est soumise à une pression externe. Particulièrement pour le cas d'études que je vais essayer de vous exposer, elle est influencée par un champignon qu'on appelle l'alternaria. En Algérie, on a deux espèces d'alternaria, Alternaria solani et Alternaria alternata. Donc, on a travaillé sur les deux, mais l'idée, c'est qu'on voulait produire de la pomme de terre résistante à l'alternaria, mais qu'il ne soit pas génétiquement modifié. Pour la simple raison, c'est que les organismes génétiquement modifiés, peut-être c'est le futur, je ne sais pas, mais quand même, ils sont légèrement mal vus. Donc la question qu'on s'est posée, du moins en Algérie, ils sont complètement interdits. En Algérie, c'est interdit d'importer, d'exporter, de produire les OGM. Donc la question qu'on s'est posée, comment on peut améliorer la résistance de la pomme de terre tout en évitant de produire de la pomme de terre génétiquement modifiée. Donc pour ce faire, la première chose, on a développé le modèle. Le modèle de production, ça veut dire, cette première image, c'est le modèle de travail. C'est-à-dire on va travailler sur ce modèle-là pour appliquer notre idée en quelque sorte. Donc sur la première photo, nous avons travaillé sur 4 variétés, 2 variétés de pommes de terre blanches et 2 variétés rouges. Donc j'ai mis une photo d'une variété rouge et une photo d'une variété blanche. Les variétés sur lesquelles on a travaillé sur Arizona, Synergie, Sponta et Désiré. Donc c'est des variétés très connues, elles sont des catalogues. D'ailleurs, les pommes de terre sur lesquelles on a travaillé, c'est des pommes de terre qui viennent de Hollande, mais via le CNCC, le Centre national de contrôle et certification des plantes. Donc la première chose, nous avons maîtrisé sa multiplication, ça veut dire la première étape, c'est l'initiation de jour. Après, la photo numéro 3, c'est la deuxième étape, c'est ce qu'on appelle élongation, c'est l'étape de multiplication. Mais il y a élongation, en même temps enracinement. Je vous ai dit tout à l'heure, pour l'arganie, on n'arrive pas à l'enraciner. Mais là, c'est facile. On peut obtenir à la fois l'élongation et l'enracinement du vitroplan. La dernière étape, la production des microtubercules. Je ne sais pas si vous voyez bien, mais sur la cinquième image, c'est une boîte de pétri dans laquelle nous avons mis plusieurs microtubercules. Donc vous imaginez, c'est la pomme de terre que vous achetez, mais au lieu qu'elle soit comme ça, elle est toute comme ça. Parce que pour nous, ça nous intéresse de travailler sur un modèle contrôlé, dans un environnement bien contrôlé. Après notre démarche, c'était quoi l'idée qu'on a eue ici? On s'est dit que dans la nature, on a le pathogène, donc l'Alternaria alternata ou Alternaria solani, et c'est connu que quand la plante hôte rencontre son pathogène, il y a une interaction, il y a une réponse gène pour gène. On a cherché dans la bibliographie, nous avons trouvé que chez la pomme de terre, les pommes de terre résistantes à l'alternaria, c'est des pommes de terre qui ont un niveau d'expression de 6 gènes particuliers. Donc l'idée qu'on a eue, on s'est dit pourquoi pas induire l'expression de ces gènes liés à la résistance de la pomme de terre avant qu'il y ait présence du pathogène. Donc c'était ça l'idée. Vous allez comprendre maintenant pourquoi nous avons travaillé sur le modèle, c'est-à-dire pourquoi on a mis en place le modèle. Donc la première, donc qu'est-ce qu'on s'est dit? Donc ce que vous voyez sur la photo là en A, c'est l'alternaria. Après, ce que vous voyez en B, C et D, c'est un antagoniste, c'est un actinomycète qui est naturellement, il bloque la croissance de l'alternaria. Donc qu'est-ce qu'on s'est dit? On s'est dit pourquoi pas ne pas prendre. Donc ce que vous voyez sur le bas, c'est des tests de concentration. Ça veut dire on cultive l'alternaria, on met sur un disque l'actinomycète et on voit une zone d'inhibition. Nous avons trouvé que la photo B, donc pour nous c'était l'actinomycète numéro 7, qui avait un meilleur effet antagoniste. Donc qu'est-ce qu'on s'est dit? On s'est dit pourquoi pas ne pas extraire les métabolites secondaires qu'il y a dans ces micro-organismes, que potentiellement, d'après la bibliographie, ils peuvent induire une expression des gènes liés à la résistance et produire une pomme de terre potentiellement résistante. Donc c'était ça la démarche qu'on a eue. Donc c'est là, il nous fallait la maîtrise du modèle. Donc vous voyez, après si vous voulez vous passer, mais en phytopathologie, quand on fait une supposition comme ça, il faut prouver que le pathogène qu'on a utilisé, c'est réellement lui responsable de la maladie. Donc nous, dans un esprit de biotechnologie, de labo, de maîtrise, on ne voulait pas faire un test sur terrain, parce qu'on aurait pu prendre les variétés, les mettre sous serre, et ramener le pathogène, et voir les symptômes sous serre. Donc nous, on s'est dit non, on ne va pas faire comme ça, on va faire in vitro. Donc ce que vous voyez ici, c'est un test, ce qu'on appelle évaluation de la pathogénicité de l'agent pathogène sur feuilles détachées. Donc qu'est-ce qu'on a fait? D'ailleurs, le contrôle, vous voyez, parce qu'on a préparé les solutions, on a piqué avec une seringue stérile, dans un environnement stérile, avec des spores du champignon. Mais pour le contrôle, nous avons suivi le même travail, mais avec d'autres distillés stériles. Donc vous voyez sur la première photo, après 4 jours de culture, il n'y a pas apparition de symptômes. Alors que pour les 4 variétés qu'on a utilisées, à chaque fois, il y a à la fin, au bout du 4e jour, l'apparition de symptômes sur la feuille. C'est-à-dire qu'on a prouvé que le champignon qu'on a utilisé, c'est un champignon responsable de la maladie. Après, tous les champignons qu'il y avait sur la photo d'avant, même les actinomycètes, nous les avons caractérisés et nous les avons inscrits sur la base NCBI. Ça veut dire ils sont inscrits. Ça veut dire on sait que c'est les bons. Ou les accessions, on les a mis même dans la publication. Là, cette partie-là, c'est juste pour vous dire que on a vérifié d'un point de vue phénotypique et d'un point de vue génétique que le matériel biologique que nous avons utilisé, c'est le bon matériel. Après, le travail, il s'est fait au niveau de l'étape 3, en 3 niveaux. Donc, on a extrait les métabolites secondaires, on les a incorporés dans le milieu de culture, et nous avons suivi le modèle que je vous ai montré de départ. Donc, on est arrivé jusqu'à produire les microtubercules. Mais on les a produits dans un environnement où il y a les métabolites secondaires du pathogène et de l'organisme antagoniste. Donc, c'est possible d'y aller. Donc, nous, qu'est-ce qu'on a fait après? Je vous ai dit tout au départ, c'est qu'on a fait un état de l'art, on a trouvé les 6 gènes responsables qui confèrent une certaine résistance à la pomme de terre. Donc, l'idée TARNA, c'est que de les activer à l'avance. En quelque sorte, le terme technique, on appelle ça la prémission végétale. Mais c'est un peu un vaccin. C'est-à-dire qu'on a essayé de développer une sorte de vaccin pour les plantes. Donc, ce que vous voyez ici, donc il y a en haut l'échelle. C'est-à-dire de plus c'est rouge, ça veut dire qu'il y a une surexpression du gène. De plus c'est vert clair, l'expression elle est ordinaire. C'est-à-dire on n'a pas surexprimé le gène. Donc nous, par exemple, je ne sais pas si vous arrivez à voir, mais là où c'est marqué C2, C1 jusqu'à C2, C5, c'est parce qu'on a fait plusieurs essais, plusieurs concentrations, on a optimisé. C'est la variété qu'on a réussi à mieux surexprimer ces gènes. Ça veut dire en quelque sorte, on a réussi à surexprimer les gènes liés à la résistance, ou les variétés que je vous ai montrées, c'est des variétés sensibles. Ça veut dire on a réussi à exprimer des gènes liés à la résistance, à l'alternariose, donc la maladie causée par l'alternata, par un moyen biotechnologique basé sur la culture in vitro végétale. Et cette variété particulièrement, quand on l'a testée, si on prend la variété telle qu'elle est naturellement, ou la variété qu'on a induite, eh ben, le niveau de résistance, il a augmenté. Voilà. Et ça a fait l'objet d'une publication qui est sortie il y a à peu près 3 mois. Donc, au mois de, je pense, elle a été acceptée au mois de juin, je pense, et elle est apparue début septembre. Voilà. Donc, ça, c'est le 3e dernier cas d'étude. Après, juste pour revenir à la page, parce que là, j'ai fait un peu exprès. Généralement, les travaux, on commence toujours par les conclusions, après on passe aux perspectives. Moi, j'ai fait l'inverse. Je vais vous parler d'abord des perspectives, après de la conclusion. Parce que l'idée, pourquoi? Parce que tout simplement, les perspectives de nos travaux, les 3 cas d'études que je vous ai montrés, c'est en fin de compte, elles tournent autour de 3 gros points. Mais c'est toujours de valider à grande échelle. Parce que tous les travaux que nous avons faits, nous les avons faits dans un environnement contrôlé, soit au laboratoire, soit sous serre. Donc l'idée maintenant, c'est que de valider nos résultats à une échelle réelle, ça veut dire open field, en plein champ. Donc c'est ça les perspectives de nos travaux. Après maintenant la conclusion de notre travail, c'est que ce que j'aimerais bien avoir réussi à vous transmettre, c'est que quand on est face à la question de valoriser la ressource agrobiologique. Parce que quand on parle de ressources biologiques, c'est plus vaste. Mais une ressource agrobiologique, c'est-à-dire c'est une ressource qu'on utilise dans l'agriculture, qui doit avoir un retour sur investissement. Donc tout l'effort qu'on doit faire, il doit être porteur. Avant de commencer la valorisation, il faut se poser certains nombres de questions. C'est quoi la ressource que j'ai en face? À quel niveau de connaissance on a pour cette ressource? Le cas que je vous ai montré, pour les populations algériennes de fraises, il n'y a pas de données. Pour le deuxième cas, l'arganier, il y a certaines données, mais la plupart ne sont pas algériennes. Mais pour le troisième cas d'étude, c'est une culture qui est largement répandue dans le monde, très consommée. Ça veut dire on ne peut pas se poser la même question. Pour la première ressource, il suffit de la caractériser, de l'inscrire, ça veut dire on l'a valorisée. Mais pour la troisième ressource que j'ai présentée, il ne suffit pas de l'inscrire, parce qu'elle est déjà inscrite, elle est déjà dans le domaine public, elle est déjà utilisée. Ça veut dire notre démarche de valoriser, c'est de voir d'un autre point de vue. Je la prends telle qu'elle, mais j'essaie d'apporter une nouveauté qui fait que mon process, parce que là, ce n'est pas la ressource réellement qui est innovante, mais plutôt le process. Le process que j'ai mis en place, c'est un process qui permet d'un peu améliorer la productivité, le retour sur l'investissement de l'espèce ou de la ressource agrobiologique que j'ai en face de moi. Voilà, donc ça, c'est la conclusion. Et sur ce, je vous remercie de m'avoir écouté.
Thank you very much, Professor Hafi. Now we will move to the next plenary lecture, which will be given by Dr. Amin Boukerb. So he is a researcher at the Microbiology Signals and Microenvironments Laboratory of the University of Rouen in France. His scientific work focuses on environmental and molecular macrobiology, bacterial genetics, and pathogens ecology within a One Health framework. He has conducted extensive research on bacterial diversity and adaptation mechanisms, particularly involving Campylobacter species in environmental and host-associated ecosystems. His studies integrate microbiology, bioinformatics, and epidemiology to better understand microbial interactions and their implications for human and environmental health. Today, he will give a talk entitled Diversity of Campylobacter lari in One Health Context, focused on shellfish, wild birds, surface water, and human health risk. We can start now.
Ok, c'est bon. It's okay. Ouais. Hello everyone. So first, thanks for this kind introduction, and I want also to thank first Dr. Nassim Madi and the organizer for inviting me today to present our work on the genetic diversity of Campylobacter larvae in a One Health context, with a focus on shellfish, wild birds, surface waters, and the associated human health risk. So as you know, on the watershed there are several sources of fecal pollution that comes from, for example, from bovine breeding, manure breeding, wastewater treatment plants related to the human activities, also direct discharge from the houses, where you can also have the porcine breedings related to animal culture, and all these sources can go through rivers. Likely during wet weather, during rain, so you have a runoff of the surfaces, and all this fecal material can go through the rivers and to the sea. Once arrived at the sea, so we have central risks for the users of course, for persons who have recreative activities like bathing and fishing. But for today's talk, we will focus on the shellfish harvesting areas and the shellfish that are after consumed by the population. So when these shellfish are contaminated, there is a central risk for consumers, and we can have what we know of collective foodborne poisoning. So two major effects we can have. The first one is the economic consequences, closer declinization of the shellfish harvesting areas or the bathing areas, and as I said, the human health risks. So the objectives of our work were to first have an idea of the identity of the sources. How can we identify each source of pollution in the upstream catchment? And after this, we focused on a bacterial species, which is Campylobacter, and try to see its presence and in which emergence in each compartment of the catchment. So this is for the group, the genus of Campylobacter, and surprisingly we found that there is a species or a group of species which was dominant, which is the group of Campylobacter larvae. So we focused our work on this group of species to study the genetic diversity, in shellfish and also in wild birds. So to do this, the first step was to identify the sources of fecal pollution. And to do this, we used the 16S metataxonomic method. So I said before, we know that in every source of pollution, we have what we call the fecal undicular bacteria, which are E. coli and Enterococcus. So they are common to every fecal pollution. But the problem is when we monitor this microorganism, we don't know, we can't identify exactly what is the exact source of pollution. So in the recent years, there was development of markers. Here we have a list, a non-exhaustive list of markers for human, ruminant, cattle, dog, and avian markers. And simply it represents a bacterial species which is specific to each pollution, each source. And so researchers developed qPCR primers to amplify specifically those targets. And for example, if we use the HF183 and we get positive results, we are pretty sure that the source of pollution comes from wastewaters, for example. And so as an example, so as I said, in each solution you will have E. coli, which is present in each solution, but we have markers colored here in yellow, red, and blue. And so when we have a pollution at a specific time, here, for example, as I said, we can really be sure that the pollution comes from the human. So up to date, the markers developed are mainly for ruminant cattle, but there is already no marker for the wild birds. So this work was also meant to try to develop such markers. So to do this, we've done samplings in different regions of the Atlantic side of France. And so we got about 565 samples from almost 12 wild bird species and also Lake's chicken, also for the domestic birds. So for the metabarcoding technique, the strategy we used is to target the RNA ribosomic analysis, or 16S. And we used the short read technology to do this, so we were not able to amplify the whole gene. So we targeted the V3, V4 region using specific primers. And so using this strategy, we can identify at the right of the slide. We can say, using this, we can have barcodes for each species or genus, and thus identify the bacterial communities in our samples. There are other techniques that are already used today. So, for example, targeting the complete marker. So we can, with the long read techniques, we can amplify the whole 16S gene, and with this we can easily identify the species. Or another technique, but it's more expensive, is to use the shotgun sequencing, where we will sequence all the genomes of each sample. So here, an example of a result that we got for the data for the whole data set of the wild birds samples. And what we can say is that we have four dominant phylums: Primicus, Proteobacteria, Actinobacteria, Fusobacteria. But also we detected Bacteroidetes, which I forgot to say it. So Bacteroidetes is a marker for the human and cattle source of pollution, and we detected some Bacteroidetes in our samples of wild birds. So this confirms that the markers are not 100% efficient in identifying each source. So in no detail, I did not give a lot of details, but here is a map of the results we got. So the diversity at the genus level in our samples, they are by bird species. And so what we can see here, that there is a large diversity of the genera that we found. And thank you. And another observation we got for example, here I highlight the Cattylococcus genera, which we found in several wild bird species. And initially, researchers who developed the GFD marker, he was meant to identify only the pollution from seagull. But the problem is, using this high-throughput sequencing strategy, we confirmed that this Cattylococcus marker can be found in other wild bird species. And so this can make mismatch between the source identification between each wild bird. And from this data, we detected pathogen signatures and mainly with Campylobacter and Helicobacter signatures. In parallel, we've done the same. We've done the same strategy. So the metataxonomic profiling of samples from the other sources, so the domestic birds, cattle, and wastewater treatment plant. And so the idea after this is to remember, it's to identify specific markers for wild birds. So when we compare all this data set together, we can clearly see that there is microbial signatures which are different between each source, and we see that. And using this data set, so I don't detail the bioinformatic approach, but using this, by comparing all the data set, we identified the group of genus, or bacterial genus, which was specifically present only in UTS1. So here we've done initial tests using QRT-PCR, and it's pretty promising marker with up to 75% sensitivity and up to 90% specificity, which is pretty good. But I think we need to do more improvement to get higher percentages of sensitivity and specificity. Yes. So this is the first way to use this data. The other thing that we can do with this metataxonomic data is we can. So at the left you have the signatures of each source. For example, I put here human, cattle, birds. On the other hand, for example, we will take the water from the seawater. We will do also the metataxonomic data and using bioinformatics, using a tool called SUSTracker. By combining both data sets, we can predict, it's not perfect, but we can predict that we have certain percentages of pollution coming from each source in our sample. Yes. Always using this data set, we specifically searched for the presence of, so we selected a list of pathogens and we tried to look for the most abundant genus pathogen in our data set. And for wild birds, we found that it was the second most abundant pathogenic genus in our data set. So for the next parts of the presentation, I will focus on this genus. So to study this bacteria, we used at this time the whole sequencing, genome sequencing approach. A quick presentation of the Campylobacter genus. So it belongs to the Ipsilobacteria. It's a gram-negative bacteria, non-spore forming, motile. It is the most common causative agent of gastroenteritis in humans. It's predominant in many sources, from wild birds, domestic, livestock animals, etc. And there is about 33 species within the genus, and the most studied species are three, Bejaïa, coli, and Lai. So for the Lai group, it's a member of the thermotolerant group of Campylobacter. It's more difficult to culture than coli and Bejaïa. It was detected in sporadic cases, less than Bejaïa coli, but sporadic cases exist for this group of species. In the opposite of coli Bejaïa, CRTAA can more easily spread and grow in saline water, so and shorebirds also. He was also more halitolerant, so to the salt waters. For the genotyping of this species, as you can see, we use, I will detail this in the next slides. So one genotyping tool is the sequence typing. And for example, for the whole group of Celari, we have up to date only 342 sequence types versus up to 15,000 STs for Bejaïa and coli. So to achieve our work, we've done a series of samplings at the Bejaïa catchment, which is connected with 4 rivers, and there's about 9,000 inhabitants in this region, and this region is also known for an intensive livestock farming. So we may have sources of pollution from these farmings and also from the inhabitants of the region. So we've done a series of samplings for shellfish, oysters, mussels, and also cockles, surface waters, of course wild birds in the region, and the human isolates from the wastewater treatment plant. So for the results that we've got, in average we observed that about 39% of the shellfish batches were positive to Campylobacter. And you have here the other results. For example, for river waters, for the four rivers, we have a high prevalence with up to 85% of positive samples. And for the shellfish, it's about from 37 to 45% positive samples in our samples. Something also that we observed for the culture method, we didn't find a clear correlation between the presence of Campylobacter and E. coli, which means that the E. coli is the indicator of pollution, is not always effective if we can check the presence of pathogens. Another observation that we got, so we've done all the samplings during the year, and what we observed is, surprisingly, there was a significant seasonal effect where we got a lot of Campylobacters during cold weather, so during winter and autumn versus spring and summer. Then also, if we have a look of the prevalence of now we look for the prevalence of each species in our samples. Surprisingly, we found that Bejaïa and coli were more present in the rivers, so in the less saline water. But Larry, as I said before, he likes more saline water, so it was more prevalent in seawater and in our shellfish samples. For wild birds, we have also a differential. So first, we had the dominance of Celari in all samples, and then we have the Bejaïa and coli and another species which was less predominant, which was Pyroides. And so here you have an example of some groups of wild birds with the distribution of each bacterial species. So at this time we have a nice view of the prevalence of the species in the different sources. The next step is to identify the genotypes. So as you know, we can, on the petri dish, we can have the same phenotype, the same colonies, but maybe there are mostly what we have are two different strains with different genomes. So, as I said before, we can use the 16S to genotype our isolates, but using this marker, we will have we can identify only the species. Another tool that we can use for genotyping is the multi-locus sequence typing. The approach is to sequence at the same time 6 to 8 genes, and so when doing this for each strain, each gene will have what we call an allele. So we will give to each gene an allele, and the combination of the codes for each allele give us the ST or sequence type. And at this time also, you can have two strains, having the same MLST, so it's, at this time also we can be sure that it's a clone or different strains. So to do this, we use the whole genome sequencing. And so at this time, so we will really sequence the whole genome, identifying the chromosome, the invented plasmids, and at this time we are clear, we can clearly separate or identify the different strains of our collection. So to do this, you have today mainly two strategies, the short reads and the long reads. The gold standard for short reads is Illumina, which gives what we call a draft genome. So the genome is, we have gaps in the genome, so it's not a complete genome. So we don't have a clear view of the genome. And for example, we have the great chromosome and two small, which should be plasmids. For the second technique, so the long reads, using the MinION from Oxford Nanopore Technologies. So here we will have a better view, as it's the same genome. So here we have the chromosome, but it's linear, and we have also these two small, which should be plasmids. So we have a better view, but it's not really yet clear. And using bioinformatic tools like Unicycler, we can combine both data sets. And using this, we will have a clean chromosome, circularized chromosome, and the plasmids with a complete view of our genome. So using this strategy, we sequenced the whole data set, the whole strains that we isolated from the watershed. And as you can see, for C. jornii and C. coli, we've done some phylogenetic analysis, and you have only two closed clusters. But when we look for the ciliary group, we have the highest, a higher diversity with the identification of the ciliary group, but also what should be maybe also a new species that I will talk about in the next slide. As I talked about the MLST, so use this to genotype our strains, and it gives the same clusters, clades, as for the whole genome phylogeny. So here for the ciliary group, we identified 40 different sequence types, and this led us also to identify the same clones that are present in different sources, seawater, shellfish, and human, and this can explain that some clones can already come from shellfish and contain humans, so that's why we detect the same clones in the different sources. Here is the pangenomic analysis of the whole dataset. So as you can see at the left, so you have all the samples, and here it's a heat map for the similarity between genomes. So simply it's a blast at the genome level, and using this we can grab the genomes by groups, and it identifies so to each strain belong to each cluster. So here you have coli and Bejaïa at up, and the ciliary group and at the bottom. And on the right slide you have the number of genes for our samples, and what you can see is that coli and Bejaïa share a high number of genes, up to 80%, 28%, sorry, while they share only 60 or 70% respectively with ciliary. And so with this analysis, we found that there was a clade which was phylogically distant from the ciliary group, so we were interested in this clade. So when we analyzed this clade, there were nine isolates. Sorry, 2 were isolated from every water, 6 from every water, and 3 from human stools. And so doing some phylogenetic analysis, we identified that we concluded that this 9 strains belongs to a new species that we call the Campylobacter americus. And so this extends the phylogeny of the group of Campylobacter. Just a quick reminder that we used also the hybrid strategy of sequences using Illumina and also nanopore for one strain of this new species. And so using this strategy, we've done different analyses, and we selected, for example, the second result, identifying clearly the chromosome and both plasmids. What we can do also with the genomic data is we can screen the genes of interest. And since we are working with the pathogenic bacteria, so we can screen the genome for variant genes, for example, genes coding for flagella, pili, adhesion, biofilm, secretion systems, toxins. On the other hand, we were also interested in genes coding for antibiotic resistance and the efflux pumps. And all these genes can also be mobile. So this is the interest of the hybrid strategy of sequencing. So we can easily detect the plasmids and also screen specifically the plasmids for these genes. So using this, I will not go in details, but just to show you that we screened the 3 species, and what we can see is that coli and jejuni are known to be pathogenic. It's established, but Leroy, this is initial data in literature. There are only some studies, and we clearly see that also the ciliary group contain genes for antibiotic resistance, which make it also a possible an emergent pathogen. And this, for example, for the antibiotic resistant genes, they are known to circulate in the population also by the horizontal gene transfer between species of different genera. Here is the same result, but for the variant strains, and also we can see that the ciliary group also can harbor several general genes that were previously confirmed as having a toxic effect on in vitro for coli and jejuni. Yes. So for the conclusion, and if at the end, so I think we don't eat shellfish anymore because of all this pollution, but not. Of course, there are controls of the shellfish to guarantee the food security of the consumers. So the real take-home message is that from our results and the bibliography data, clearly Campylobacter is the dominant bacterial genus in catchment. Ciliary is clearly dominant in shellfish and seabirds, and more research work is needed to get a higher number of sequence types, to identify maybe other species, and to get more insights in the pathogenicity of this Campylobacter species. Of course, you have to keep in mind that we use limited detection methods for the culture methods. So maybe sometimes we have false positive or false negatives because the ciliary group is very diverse metabolically. So maybe at the lab we use standard Campylobacter specific culture media, but maybe we can't culture the new species that we have to discover in the future. And to finish, so to make connection between the two parts, the idea at the end is to try to connect the detection of sources using the microbial tracking, and to be sufficiently efficient to really predict the presence also at the same time of Campylobacter pathogens in our samples. So at the end, I want to thank all the partners of this great project and you for your attention. Thank you.
Thank you, Dr. Boukeb, for this nice presentation. Now there's a small change in the program. We start by about 20 minutes of debate about these 2 plenary sessions before continuing. Thank you. You can come back to the stage, please.
So we would like to thank the communicator for this interesting intervention, and we will start the debate. We will have about 20-minute debates, so we'll take questions. So we will start just here. So.
My question is going to be for Professor Harfi. Je devrais poser la question en français ou là en anglais. Mais ce sera bizarre comme ça. D'abord, je voudrais te remercier pour l'exposition que vous avez faite. C'est vraiment quelque chose. J'apprécie bien ce genre de communication dans lequel on voit ce que vous faites. Oui, donc c'est très, très, très intéressant. Ma question sera sur la première espèce que vous avez étudiée, donc le fraisier, donc la Russicada, si j'ai bien... D'accord ? Ok. Donc j'ai une petite question par rapport à l'indication géographique, parce que vous avez parlé de valorisation et de skigda. J'ai zappé quelques diapos, donc je ne suis pas sûr. Vous avez parlé de polymorphisme, c'est ça ? Donc, est-ce que ça veut dire que les groupes de cette variété démontrent une diversité génétique et donc, en fait, il ne s'agit pas forcément d'une variété, ce qui va réduire les chances pour que ça soit, par exemple, valorisé en tant que produit label. Ça, c'est ma première question. La deuxième question, c'est par rapport au système de culture, exactement. Donc, je vois que vous cultivez ça sur du sol. Puis je vois un fraisier, je le vois bien parce que votre objectif est de passer à l'industrialisation. Donc pourquoi pas l'hydroponie ? Oui, c'est possible. Donc parce que l'hydroponie, donc je pense que l'hydroponie, elle accélère un petit peu la croissance, etc. Et c'est nettement plus rentable en termes d'industrialisation. Bon, je pense que ça sera tout.
Sans micro, il n'est pas allumé. Il est allumé. Oui. Ouais. Donc, merci pour les questions, très intéressantes. Donc, pour la première question, vous avez donné beaucoup d'informations. Donc, généralement, une appellation d'origine géographique, c'est-à-dire quand on a une espèce qui est déjà inscrite variété, mais dans un certain environnement, elle donne une qualité particulière. C'est-à-dire la qualité du produit final, elle dépend de la région où elle est cultivée. Ou si c'est un animal, la région où il a été élevé. Dans ce cas-là, on donne une appellation d'origine géographique comme label. Là, ce n'est pas le cas. Parce que là, la question qu'on s'est vue, d'ailleurs, vous avez dit, j'ai parlé de la variété Russicada. En réalité, on ne peut pas savoir si c'est une variété. Parce que, je ne sais pas si vous avez suivi, mais la première diapo, j'ai mis la réglementation qui définit une variété. Une variété, c'est quoi ? C'est une population qui se comporte de la même manière, mais qui est différente de tout ce qui existe déjà. Ça veut dire, nous, pour qu'on puisse dire que les populations qui se retrouvent à la wilaya de Skikda, c'est des variétés, il faut qu'on démontre qu'elles sont différentes de tout ce qu'il y a dans tous les catalogues. Vous voyez ce que je veux dire ? Ça veut dire, c'est à ce moment-là qu'on va l'appeler variété. Avant de faire ça, ça reste une population. Ça veut dire, le mot variété, il est très réglementé, on va dire, très bien défini. C'est-à-dire on ne peut pas l'appeler variété si on ne prouve pas qu'elle est différente. Vous voyez ce que... Donc nous, dans cet esprit-là, déjà, il y a une première chose qui ressortait. C'est que d'un point de vue phénotypique, le phénotype, c'est à peu près tout ce qu'on voit. C'est-à-dire, par rapport à ce qu'on voit, c'est une fraise qui est un peu de petite taille, qui a une odeur très forte. Quand on goûte, elle a un goût légèrement caramélisé à la fin. Donc tout ça, c'est des indices qui montrent qu'elle est potentiellement différente. Maintenant, il faut le prouver. Donc c'est ce qu'on essaie de faire. Et les premiers résultats, on a trouvé qu'il n'y a pas une très très grande diversité du point de vue ACSR parce que nous avons utilisé un nombre réduit. 4 ACSR, ce n'est pas suffisant. Il faut aller plus loin. Mais on ne s'est pas arrêté là. On a analysé de gènes spécifiques qui confèrent une certaine qualité à la fraise. Donc pour le premier gène, la séquence qu'on a trouvée, sa taille, elle est différente de la séquence connue. La séquence connue, je pense, c'est à peu près autour de 330 paires de bases. Alors, pour notre cas, nous avons trouvé une séquence de 245 à 275 paires de bases. Déjà, ça montre que c'est différent. Ça, c'est encourageant. Et on est maintenant en train de séquencer le gène en question. C'est-à-dire pour dire que ce gène, il est différent du gène connu. Après, le deuxième gène, parce qu'au début, quand on a fait de la bibliographie, on s'est rendu compte qu'on parlait d'une seule variété. Mais en faisant de la prospection sur terrain, nous avons remarqué qu'il y a 6 sites où il y a des populations, entre elles déjà, elles sont différentes. Donc c'est possible, on espère bien, qu'à la fin, on ne va pas inscrire une seule variété, mais peut-être 2, peut-être 3. Donc ça, c'est déjà, voilà. Donc j'espère que j'ai répondu à la première question. Oui. Après, pour la deuxième question, par rapport à l'hydroponie. L'hydroponie, c'est l'industriel. C'est-à-dire, nous, pourquoi à la fin nous avons cultivé dans un sol bien précis? Parce que c'est l'expérimentation qui l'exige. C'est-à-dire il faut un sol où on a 75% de terreau, 25% de perlite, qui soit stérile ou qui soit dans des conditions contrôlées. Donc c'est l'expérience qui montre, parce qu'il faut qu'on cultive les 6 populations potentielles dans les mêmes conditions. Et comme ça, quand on va mesurer la taille de la feuille, la direction des poils du pétiole, la forme des dents, de la foliole, il faut que ça soit dans les mêmes conditions. Parce que pour être sûr qu'on a éliminé l'effet environnement. Mais après, une fois, on suppose qu'on va obtenir une variété à haut potentiel, à ce moment-là, celui qui va acheter le plant, parce que c'est l'Algérie, elle importe le plant. Le plant, ça veut dire la petite plantule de la fraise. Donc, il peut le cultiver hors sol, en hydroponie, ou maintenant on parle même d'aéroponie. C'est encore mieux, parce qu'au lieu de mettre de l'eau, on met la vapeur de l'eau. Ou carrément sous serre. Là, après, ça dépend de l'industriel. Mais nous, on propose un modèle pour produire les plantules. Ou en plus, pourquoi le modèle il est important? Parce qu'on est sûr que le produit final, c'est un produit sain. Il n'y a pas de virus, parce que même les bactéries, les champignons, peut-être on peut traiter. Mais le virus, c'est un parasite obligatoire qui se retrouve dans la cellule. Généralement, l'agriculteur, quand il achète la fraise, il ne fait pas une seule production. Ça veut dire qu'il la garde jusqu'à 3 ans, où il y a 2 productions par an. Il y a le premier bouquet, généralement autour du mois de décembre que ça commence. Le deuxième bouquet, il commence à partir du mois de mai, avril-mai. Ça veut dire, s'il garde sa production sur 3 années, vous imaginez, il multiplie chaque année par stelon, c'est une multiplication végétative, donc forcément, la charge virale, elle augmente. Donc, voilà, vous voyez, ça veut dire, c'est pour ça que j'ai montré, ce que je montrais, c'était dans des bocaux en verre. Juste la dernière étape, l'acclimatation, c'est dans un terreau, mais c'est aussi, il faut répondre à une certaine exigence expérimentale. Voilà. J'espère que j'ai répondu, OK. Exactement. C'est pas pour la production finale. Exactement.
Thank you, Professor Harfi, for this insightful presentation. So I have a pretty good general question. So from your perspective, what policy or investment priority would most accelerate Algeria's progress in seed characterization and valorization?
Si vous pouvez traduire en français, je dirais pas non.
Vous pouvez lui reposer en français la question.
Donc vous voulez savoir ce que cette valorisation peut apporter à l'Algérie, si j'ai bien compris, en résumé. Ça veut dire, normalement, si on réussit à inscrire la variété algérienne, comme elle est un peu particulière, ça veut dire d'un point de vue goût, d'un point de vue produit final, c'est un peu différent. Ça veut dire qu'on peut développer ce qu'on appelle un produit de niche. C'est-à-dire c'est un produit peut-être qui ne sera pas produit à très grande échelle, mais un produit différent, de meilleure qualité, qui sera vendu plus cher. Ça veut dire que ça peut être un produit un produit entre guillemets un peu plus luxueux, ce qui existe sur le marché. Mais aussi, on a remarqué une autre chose en faisant les prospections, c'est que les familles qui produisent ces populations locales d'une manière traditionnelle, de plus en plus, elles s'orientent vers les plants importés. Ça veut dire qu'on risque, si on n'inscrit pas, on ne protège pas ces populations, voilà, on risque de les perdre. Donc c'est un peu les double objectifs, à la fois la protection de la ressource biologique, mais aussi c'est une ressource, d'ailleurs on a commencé par cette question, est-ce qu'on s'intéresse à une variété hautement productive, reconnue, étrangère, importée, ou peut-être valoriser une population locale qui a démontré qu'elle est un peu différente, qui a une certaine qualité, parce que je vous ai parlé du prix qui a été attribué à cette variété aux Pays-Bas. Donc c'est les 2 objectifs, à la fois de la valeur pécuniaire et en même temps de la protection, de la préservation de la ressource agrobiologique. Je vous en prie. J'ai répondu, j'espère, à votre question. OK.
Do we have other questions in the audience? On a une question là-bas.
Bonsoir. Donc, ma question s'adresse toujours au professeur Harfi. Si on parle d'espèces endémiques, parlons d'espèces endémiques, je voudrais savoir, est-ce qu'il y a eu des dispositifs ou des programmes dédiés à la protection de ces espèces, et ainsi que assurer leur perpétuité. Et merci.
Donc, j'imagine pour cette question, vous parlez en général ou en particulier le cas que j'ai exposé pour lesquels...
En général.
En général.
Mais à part leur inscription qui reste...
Okay. Parce que pour le cas de mon exposé, quand j'ai parlé d'espèces endémiques, j'ai parlé de l'arganier. Pour l'arganier, j'avais à un moment donné précisé que c'est un projet qui n'est pas uniquement projet du CRBT, c'est un projet national. Ça veut dire qu'il y a déjà une démarche de la protection de cette ressource. En plus, pour information, pour l'arganier en particulier, il y a la mise en place en cours d'un centre national dédié à l'arganier et aux cultures sahariennes au niveau du Tindouf. C'est-à-dire qu'il y a une certaine volonté pour protéger cette ressource, il a valorisé. Donc il y a ce programme. Mais après, quand on parle de la ressource endémique en général, c'est à peu près la même démarche qui se passe quand on parle des banques de gènes ou des banques de semences. Généralement, pour qu'il y ait une démarche de protection, c'est qu'il faut d'abord qu'on sache est-ce que la ressource en question, elle a un potentiel. Parce que généralement, on dit une ressource où on ne trouve aucun potentiel, généralement, on la préserve moins. Mais après, quand je parle de potentiel, ça dépend quel type de potentiel. Parce que des fois, ça peut être une ressource qu'on va utiliser parce qu'elle permet un certain équilibre écologique. D'autres ressources, c'est des ressources qui sont à valeur ajoutée, ça veut dire qu'il y a une ressource financière derrière. Donc, il y a différents types de... Voilà. Mais généralement, c'est ça. Et en Algérie, peut-être cette question de protection des ressources endémiques, peut-être depuis, enfin ma connaissance, parce que je ne suis pas expert en ça, mais d'après ce que j'ai eu, parce que j'ai eu l'occasion de faire partie du comité APA, c'est un comité qui parle de l'accès et partage des avantages liés aux ressources naturelles, il y a une certaine prise de conscience. Mais après, on ne peut pas peut-être tout protéger d'un coup, mais il y a quand même une démarche de protection, de valorisation des ressources endémiques. Ou surtout, je termine par ça, c'est que généralement quand on a une ressource endémique, on a quand même un avantage par rapport aux autres. C'est-à-dire que c'est notre propre ressource. C'est-à-dire que si on la valorise, on aura moins de concurrence. Donc c'est plus facile de tirer profit de cette ressource.
Merci.
Je vous en prie.
Other questions? Est-ce qu'il y a d'autres questions? Oui, s'il vous plaît. On a une question ici.
Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. Good afternoon. My question is to, sorry for, je vais me lever. My question is for Professor Amin. I'm very interested by your work for detection of Campylobacter via metagenomic approach. It's very interesting. I do the same thing with camel milk, and I want to know how you can make identification of abundance at the species level using only 16S or meta or shotgun approach. Thank you.
Thank you for this nice question. So I didn't detail the material methods. So we cultivate the Campylobacters on media. When we have pure culture, at the beginning we do the 16S and the Malditov spectrometry. And combining the two results, we are pretty sure of the species. So for Carli, Jejuni, and Larry. And then in the second part of the work, we do the whole genome sequencing. And at this time also we confirm, so at this time we are pretty sure of the species. And in some cases, we notify other species which are less abundant, sorry. So here I presented only the 3 major species we found, but we found other species and also the new species that we described. Okay, thank you.
Not question, but possibility for collaboration. Because we have, we have a lot of samples, but the cost is very high.
We can discuss. Sure.
Yes,
sure.
Thank you.
Avec plaisir.
Thank you. I will have a question for Professor Harfi. Du coup, en français directement, ça va être plus simple. En fait, j'ai beaucoup apprécié votre travail et surtout, j'ai compris ce que vous avez fait et comme je ne suis pas du domaine, j'ai vraiment apprécié. Il y avait beaucoup de transparence dans votre manière de faire et j'ai beaucoup apprécié. En fait, j'ai une question plutôt, on va dire, côté marketing, si je puis dire ça. Oui. C'est, donc, si j'ai bien compris, votre objectif serait qu'on ait, par exemple, en Algérie, plus de variétés. C'est-à-dire que, voilà, c'est pas la fraise, c'est la fraise. Si j'ai bien compris, la pomme de terre, c'est pas que de la pomme de terre. Voilà.
Partie, oui.
Donc, en fait, moi, ce que j'aimerais savoir, c'est est-ce que vous avez pensé, ou peut-être que ça a été fait, d'introduire la donnée sociale, etc. C'est-à-dire que, est-ce que réellement, il va y avoir un impact sur le consommateur? Je vous donne un exemple très simple. En France, si vous prenez une recette, par exemple, avec de la fraise, on va vous préciser. Donc, prenez cette fraise et pas celle-là, parce qu'elle est acidulée, l'autre, elle est plus sucrée. Donc, il y aura un impact gastronomique.
Oui, oui.
Pareil pour la pomme de terre. Si on fait de la purée, il faut ça, il ne faut pas ça. Exactement. Donc, est-ce que vous pensez qu'en Algérie, on a cette culture ou peut-être qu'il faudrait faire de la vulgarisation scientifique par rapport à un consommateur lambda, vu que nous, en tant que biologistes, on sait quelle est l'importance de la variation dans une espèce, dans une famille, etc. Mais pour le consommateur lambda, est-ce que vous pensez réellement que... Ou bien comment vous voyez cela en Algérie?
Sincèrement, on s'est posé un peu cette question, mais peut-être que ce n'était pas l'objectif numéro 1. Parce que pour nous, l'objectif le premier, c'est préserver la ressource. Le deuxième, c'est de la valoriser. Mais le troisième, c'était ça dans l'esprit. Ce que, imaginons, on va inscrire une variété qui, par exemple pour la fraise de Skikda, on produit une confiture un peu particulière, avec un goût très particulier. Oui. Donc, si cette confiture, il y a de la publicité autour de cette confiture, il y a de la demande autour de cette confiture, forcément, les gens de la région, ils vont garder leur système de production traditionnel, qui ne sont pas intensifs, qui sont extensifs. Et d'ailleurs, ce que je disais tout à l'heure, ça va donner un produit de niche, un peu qui aura des conséquences sociétales sur la région. D'ailleurs, pour juste un petit chiffre, quand on parle de l'agriculture en Algérie, c'est vrai que les céréales, c'est le numéro 1. Donc, quand par exemple on parle des céréales, elles sont cultivées généralement les hauts plateaux. Oui. Mais par contre, au niveau des wilayas de Jijil et de Skikda, 83% de la recette agro qui vient du secteur de l'agriculture de la wilaya, ils proviennent de la culture de la fraise. Donc pour la région, c'est une culture extrêmement stratégique. Malheureusement, comme cette ressource, on n'a pas cet esprit de valorisation, de dire on peut faire une confiture ou un produit fini qui est de meilleure qualité, même si ça existe un peu pour la date. D'ailleurs, il y a une collègue tunisienne, j'ai assisté à des séminaires où les Tunisiens, ils sont un peu en avance par rapport à nous. On doit apprendre plein de choses de ce qu'ils font. Oui. Donc, dans cet esprit, non, on a cet objectif, mais peut-être que ce n'était pas l'objectif le plus urgent. Oui. Voilà. Notre objectif, c'est parce qu'on ne peut pas communiquer si on ne démontre pas que c'est unique. Voilà. Donc il faut d'abord démontrer que c'est unique, après,
ensuite apporter le
exactement. Mais on s'est posé cette question.
D'accord. Et aussi, en fait, j'ai été interpellée aussi quand vous avez parlé des OGM à un certain moment. Oui. Parce qu'on a aussi cette notion de... Voilà, moi, je pense toujours aux consommateurs lambda, toujours. Hein, donc, pas autant que spécialistes ou biologistes, etc. Oui. Donc, je sais que dans notre société, par exemple, quand on va penser... Enfin, si vous allez dire que voilà, cette fraise, elle est là parce qu'on a étudié au laboratoire, on va directement penser OGM, même si c'est pas OGM. Donc on a ce stéréotype et cette idée comme quoi laboratoire c'est dangereux, il y a un risque, donc je ne touche pas. Donc c'est peut-être aussi à prendre en considération.
D'ailleurs, cette question, je l'avais posée quand on avait parlé de la pomme de terre. Pourquoi on avait posé cette question particulièrement dans le cas de la pomme de terre? Parce que j'avais dit à la fin, en conclusion, que quand on a une ressource biologique en face de nous, avant d'essayer de la valoriser, il faut se poser la question, c'est quoi l'état des connaissances de cette ressource? Voilà. C'est que les variétés sur lesquelles on a travaillé, les 4 variétés, c'est des variétés déjà à très haut potentiel. Ça veut dire, pour les améliorer encore, la voie la plus rapide, c'est les OGM. Voilà. Et comme il y a cette réticence de la société particulièrement européenne, après une influence sur l'Afrique, des OGM. En plus, en Algérie, il est interdit de produire, de commercialiser et d'exporter les OGM. Donc on s'est dit, est-ce qu'on ne peut pas faire une autre approche d'une ressource qui n'est pas notre ressource naturellement, mais on apporte un petit plus. C'est-à-dire on a imaginé un système de produire une pomme de terre un peu plus résistante par rapport à ce qui existe sur le marché, donc qui est un peu améliorée. Mais là, qu'est-ce qui devient un peu unique dans le travail? Ce n'est pas la génétique du produit final, mais plutôt le process pour produire cette pomme de terre avec un petit plus. Voilà. Parce qu'elle est déjà hautement sélectionnée, elle a fait ses preuves en quelque sorte.
C'est juste plus contrôlé en quelque sorte.
Exactement.
Voilà, je vous remercie pour
Merci
vos réponses.
pour la question.
So, thank you, Dr. Booker, for this comprehensive presentation that reminds us of how food safety is deeply interconnected with ecosystem health. So my question is, how might your findings guide integrated surveillance programs connecting food safety, veterinary, and environmental sectors?
Sorry, can you repeat? How
might your findings guide integrated surveillance programs connecting food safety, veterinary, and environmental sectors?
Great question, thank you. So for the work I presented, the results that are can be quickly applied at the MST, the microbiome tracking results. So as I presented the markers for human, cattle, etc. Markers, and these markers are not already used, but the idea is to propose this to politicians and to people who manage the environmental pollution and such things, to help them to monitor such pollution. So these markers will help them to do this. And on the second hand, for the Campylobacter, the idea is for the ciliary group. There are very few studies on the genotyping of the environmental population of such bacteria. So at this step, we are at the beginning of the research to describe more precisely the genetic diversity of what we can find, to connect this to which genotypes we can find in human illness, and we are really at the beginning of this for the Campylobacter, of course.
Except not consuming shellfish.
Sorry?
I said except from not consuming shellfish to not get sick.
Yes.
Thank you.
Thank you.
So I think we'll stop here the debate in order to continue the session. C'est bon. Donc, thank you for your intervention and your answering your question. Et bonne route à nos collègues de Constantine qui doivent quitter. Donc, oui.
So now we call the communicant Ziad Nasri to present his communication before moving to the plenary lesson session.
Well, Assalamu alaikum. Good afternoon, everyone. First of all, I want to thank the committee chairs and the organizer of this conference. To give me this opportunity to be in here with you and share my research work. Presenting myself, my name is Ziad Nasri, a PhD student at the University of Constantine II. Today I will present my work entitled UV8 based systems for agriculture harvesting. Just a minute to present a Okay. Today I will present my research entitled UV8 based system for agriculture harvesting optimization, the case of potato cover seed selection. This study focused on the application of artificial intelligence in the domain of agriculture. So in introduction, As we know, the second Sustainable Development Goal defined by the United Nations is Zero Hungry. This goal aims to end hunger, maintain food security, or achieve food security, improve nutrition, and promote the agricultural sustainability. For our country, agriculture and food security are one of the major areas that the state intends to promote in the Algeria 2030 vision, along with industry and energy, because it has a direct impact on the economic indexes, like the volume of importation and the volume of exportation. Also, direct impact on the product prices in Algeria markets. So, the Algerian government strategy for food security and sustainable agriculture and agri-food technology focused on three baselines. The first one is large-scale harvesting, which means to support, or which aims to support the harvesting, the extensive harvesting operation in southern region. The second baseline is digitization, which means the transformation of data from classical format like document, paper, etc., to digital information. The third one is digitalization, which means the use of advanced technologies to automate and optimize the agriculture and post-agriculture harvest, or to optimize the harvesting and post-harvesting process. And of course, data science and artificial intelligence are the core of these advanced technologies. Moving to the problematic, as we know, potatoes represent one of the Algeria's most consumed and cultivated crops, but sorting and seed selection of these products remain mostly manual and inaccurate, which lead to several disadvantages and problems like time-consuming, yield losses, intensive use of chemical and fertilizers, and finally leads to unstable production. So, as a solution, we propose to using artificial intelligence to automate the selection process and optimize the quality of the selected potato tuber seeds. The primary goal of this work is to propose a computer vision method based on YOLO V8 to classify and select potato tuber seeds according to the optimal selection criteria defined by the domain expert and research. So, in our work, we focus on two criteria, or features as we named in artificial intelligence. The first one is the shape, where we analyze or we evaluate the shape of the potato tuber seed and detect any surface defects. The second criteria is the potato eyes, where we count and assess supporting eyes for viability. What is YOLO V8? YOLO V8 is there is some image don't isn't there. 1 minute. There is some image don't present it. There is some image that doesn't present it in the Donc, on a continué sur des images. Alors, YOLO V8 is a computer vision technology, or AI technology, developed by the Ultrasec Corporation. It's used for object detection and instant segmentation. It's architecture based on deep learning models, especially CNN, convolutional neural networks. It's characterized by its lightweight deployable model, which make it suitable for small devices, and real-time processing, which that allow for continuous detection and classification process in real world. We move now to the method and materials. The first phase of our methods is creating a data set where we collect our data from 3 different open data sets. It's available on Kaggle platform. These data sets are used as data resources. The second step of the construction of our data sets is about performing a data cleaning and processing using Python programming language and its specialized frameworks in this task. The third step is data labeling, annotation, and augmentation, where we use the RoboFlow tool. Now we discuss the model training and fine-tuning process. Here in the image, we show the tasks that we need from the machine learning do it. In the image at the right, the model should detect the body of the potato and classify if it's a normal or abnormal shape. The second image at the left represents the task that we need from the YOLO V8 performance, which is the detection of potato eyes. The code below presents the configuration of our model, where they contain 4 principal sections. The first section is about the upload of the model. The second section is about the call of the model responsible for retraining the model on our data. The third section is about the specification of our dataset file. And the last section is the fine-tuning or the configuration of our hyperparameters. So, for the results, as we see in the chart in top, the model is training with success, where we show a decrease of the loss function. That means that our model is learning with success. Also, the performance of our model is increased in the bottom figures, which means the learning process is performed normally. This figure is about the learning curve of potato-shaped classification tasks. And the same thing for the learning curves of potato eyes detection. Although the loss function doesn't achieve the optimal value, the performance metrics show a high rate, where we observe that the model achieved 40 or 94% for both models, and for precision performance metric, the model achieved 90% for the classification model and 96% for the eyes detection task. For the mean average precision, the model achieved 95% for the task of potato shape classification and 97% for the task of eyes detection. This image illustrates that or shows that our model works correctly, where we show that the model detect normally the potato bodies and classify them to normal and abnormal potatoes. The same thing for this validation sets. The model work normally and detect the potato eyes with high precision, as we see in the image. So, summarizing our contribution, the first contribution of this work is tailored YOLO implementation for agriculture automated sorting and precise selection methods. Enable or incremental training methodology, enabling continuous learning approach, ongoing model improvement and adaptation. And the third contribution is a sustainable impact on the field of agriculture, where this work has a direct contribution to the sustainable agriculture precise through intelligent optimization and resource optimization. In conclusion, our research provides a fundamental step toward transforming traditional farming practices through cutting-edge artificial intelligence. The future work will focus on enhancing the selection criteria. The second step will focus on provide IoT and robotic prototype. And the third one is applying this model on different crops like tomato and other wide consumed products in our country. The expected outcome of this system is to enhance the seed bank of our country and the quality of our seed bank, and improve products return through precise classification. Thanks for your attention and welcome for your question.
Thank you, Dr. Nasrin. Now we will move on to the next plenary session with Dr. Khalifa Rasim, who will join us online. First, I will present him to you. So he is an assistant professor of biology at Concordia University in Montreal, Canada, and the leader of the Open Lab, which focuses on ecology, biodiversity, and sustainable environmental solutions. He earned his PhD in ecology from the University of Zurich, Switzerland, and has held several international research positions in Switzerland and Canada. Dr. Khalifa's research explores how climate change and human activity affect biodiversity and ecosystems. He is particularly interested in using innovative, nature-based approaches to promote sustainable and reduce waste. He has recently been awarded a Tier 2 Canada Research Chair in Global Change Ecology for his contributions to this field. Today, he will give a lecture entitled From Waste to Resources, Converting Canada geese feces into protein and plant fertilizers using the black soldier fly, Hermesia illucens. Dr. Khalifa, do you hear us? Okay, so we will put on your presentation. I will pass the slides for you as you speak. Is it okay? But I don't hear you.
Hi, do you hear me now?
Yeah, it's okay. Please hold on a second. Dr. Khalifa, you can now start.
Okay. Hello, everyone. I'm really excited to give this talk. I would like to thank the organizing committee for inviting me for this amazing conference. I really wish to be there physically, but it was not possible. And today, I'm going to talk about how to use insects to valorize different types of wastes. Next. My name is Rasim Khalifa, and I'm assistant to professor in Concordia University in Montreal, Canada. And this is the Loyola campus, where my lab is hosted. Next. This is supposed to be video, but it doesn't matter. It's not very important. We are interested in the causes and consequences of biodiversity changes in my lab. You can go next, please. And we are particularly interested in the life history traits, geographic distribution, and community interactions. But we have another axis of our research, which is to understand or find new sustainable solutions for biodiversity conservation, and particularly target the human dimension and a transition to more sustainable ecosystems. Next, please. And in my research, I use theory, experiments, fieldwork, and the analysis of long-term databases to answer my research questions in collaboration with researchers, NGOs, community scientists, farmers, conservation practitioners, etc. Next, please. And recently, I got really interested in circular economy, and I'm going to explain why. Next, please. We know that there is a big biodiversity crisis nowadays. For example, if we look at the Living Planet Index, which is based on vertebrates, it declined by 68% during the past 4 decades. Next, please. And according to the IPBES, 1 in 8 species, so about 12.5%, are threatened with extinction, which means like 1 million out of the 8 estimated million of animals and plants are threatened with extinction. And so this is a big problem. It's usually in the headline nowadays in newspapers. Next, please. And so why should we care about this? Well, first, each species has a unique evolutionary path, and each species has a story that we need to learn from, and the extinction of species is like the extinction of or destruction of an entire library. Next, please. So by losing a large number of species, it's like burning libraries, similar to what we have seen with the Library of Alexandria. So we really need to take care of this. Next, please. And the second main reason why we should care about biodiversity is that we really depend on biodiversity for our own survival. It provides us with many ecosystem services. For instance, 35% of global crop production depends on animal pollination. Next, please. And there are many drivers of biodiversity loss. We can see here some of them, the major one listed, like land use, direct exploitation, climate change, pollution, invasive alien species, etc. And by far, the most important one is changing land use. It's the one that really has the most effect. Next, please. The exploitation of natural resources has negative impact on the environment, and because part of it is it produces a lot of waste. And if we consider extraction and processing of resources, they're responsible for 90% of biodiversity loss. Next, please. And so we are at the decisive moment in history where if we continue on the same path, biodiversity loss will continue. That will be the gray line, business as usual. If we increase conservation efforts, which is here, it's the orange line, we improve the situation a little bit, but it's not optimal. The best scenario in this case is that really bend the curve of biodiversity loss is not only conservation effort, but also sustainable production and sustainable consumption. So that's the only way out. Next, please. And the challenge we have is how can we reach this goal with a growing human population that can reach 10 billion by 2050? Next, please. Well, the answer is to integrate circular economy models, and those, they aim at reducing raw material, reducing waste, and reducing emissions. Next, please. If we think about our current economy system, it's usually run linearly, which means that in order to produce any good, we extract resources, we manufacture the good, we distribute it to the users, and finally, we use them and it goes to waste. So after using them, this model doesn't allow for us to take components of what we produced and put it back to the chain. So, which means that in order to produce more, we need to extract more. And so this is not a sustainable model. Next, please. So if we apply this model to agriculture, we need to destroy natural habitat to create space for agriculture. And of course, part of what we produce is consumed, and part of it goes to waste. And as we will see, sometimes the proportion of waste is high. Next, please. And if we want to increase production, let's say we have a growing human population, well, we need to destroy more habitat, and we need to have more agricultural lands. So as you can see, the linear model is not really sustainable. Next, please. The alternative model, the circular model, is more sustainable, and it is strategic. Because it works with a different philosophy of after using the product, we tend to think about ways of intensifying the product use, extending the life of product, and especially reintegrating components of resources to different steps of the chain. And so we are no longer really reliant on extracting resources to produce a good. Next, please. We have a definition of circular economy here in Quebec, which is a production, exchange, and consumption system that optimizes the use of resources at all stages of life cycle of a good or service in a circular logic, while reducing the environmental footprint and contributing to the well-being of individuals and communities. Next, please. So in the agri-food sector, many scientists think that insects can be an important link for reintroducing waste into the food production chain. And this is important since in many countries, 40 to 50% of food is not consumed and goes directly to waste. This is the case of Canada or the US. Next, please. And so why insects? Well, insect is the most diverse animal group or group on Earth in general. They provide a wide array of services to people and ecosystems, many of which can be harnessed to advance the circular economy. Next, please. So if we look at the coprophagous insects, which is a particular group of insects that decompose organic matter in the wild, some of these species have been used for waste management of organic waste. So they have the ability to eat different types of food, including our waste. And by doing so, we produce protein that can be used as feed to a broad range of animals, like crustaceans, fish, poultry, wild bird, etc. So those insects, they belong to different groups. We're talking about flies, crickets, beetles, but the insect that received the most research and economic attention during the past decade or so is by far the black soldier fly. Next, please. So the black soldier fly is a dipteran. It has a short life cycle. The female produces 300 to 600 eggs, and those they hatch in 3 to 4 days, and after that the larvae grow pretty fast. They start very tiny and they end up pretty big, like 10,000 times bigger. So proportionally, we go from an apple to an elephant in a few weeks. And they have the ability to eat a broad range of wastes. So food scraps, agricultural byproducts. And so we are converting waste to protein. And more importantly is that the frass, which is the residue of the decomposition, so the excrement and the leftover, has been shown to be a very good fertilizer for different plants. Next, please. And the insect farming as an industry is a booming sector in different regions worldwide, including North America, also Europe and Asia. For example, here in Canada and US, you can see like different producers and processors. So it's a growing sector. Next, please. For example, here in Quebec, we have a pretty famous industry, Ontosystem, which they produce only black soldier fly. So they produce larvae and they sell them in different format. So it's a viable system, like economic system. Next, please. So it's a business that is that targets a problem, which is waste, and then convert it into resources, protein that can be used as feed, and frass that can be used as a fertilizer, and you get revenue from that. Next, please. Of course, during the past few decades, many studies have been conducted on different types of food scraps and organic wastes, and the insect has been a very good bioconverter of these wastes. Next, please. The frass has been tested on various crops with market benefits to plant productivity. Next, please. And so I decided, like a few years ago, to integrate research involving the black soldier fly to my lab. We established the colony. We really started from zero, and the colony quickly became really big. So we are maintaining it pretty constantly, and we do different types of research. Next, please. This is supposed to be a video that shows larvae eating some waste of fruits, and they do that pretty quickly. We filmed that in our lab. Next, please. And before diving into the theme of this research, of this presentation, I would like to give a little bit of a highlight about some recent projects that involve black soldier fly in my lab. that involve duckweed, Drosophila, and aquatic insects. Next, please. So I would like to start with this project. If you're interested, it's available, it's published. We started with this test of an insect plant circular system based on black soldier fly and duckweed. This started as an undergraduate project. Then the project was too exciting that a postdoc took it over. Next, please. So what we tested here is the efficiency of black soldier fly larvae to recycle this aquatic plant, duckweed, which is very high in protein. And the second aim was to test the efficiency of frass to grow duckweed in the lab. And so it's like we are not only producing duckweed with the frass, but also test whether the duckweed is a good food for the black soldier fly. Next, please. So we exposed the duckweed to different concentrations of frass, and after a month, we recorded the growth rate of duckweed. Next, please. We found that duckweed grew optimally in low concentration of frass, and what was remarkable here is that the growth rate exceeded that of our control called Hoagland's media, which is the optimal media usually used in biological studies. And so the frass exceeded significantly that rate. Next, please. And so we confirmed that, yes, the frass is a good fertilizer for the plant. Next. So in the second experiment, we increased the content of duckweed in the diet of the black soldier fly larvae. And we estimated different parameters related to the larvae and the adults. Next, please. And we found that black soldier fly, they can develop in pure duckweed diet. They can do that. But the performance of growth and different other parameters is lower than the control. So they can do the job, but if your aim is to get bigger larvae, the duckweed is not the best diet. Next, please. So yes, they can grow, but not optimally. Next, please. And so we were able to test that black soldier fly can develop in pure duckweed diet. Higher duckweed inclusion tend to reduce black soldier fly performance. We identify the concentration that is optimal to grow duckweed, and we think that black soldier fly frass is a low-cost fertilizer to produce duckweed, but not only duckweed, other plants as well. Next, please. The second project that was also done by an undergraduate, Nigar, which tested in a laboratory experiment the ability of black soldier fly to bioconvert Drosophila media. Next, please. Drosophila is one of the most widely used model organism in biological research, with thousands of labs across the world maintaining constantly fly cultures. And so we use a lot of media to produce or to maintain Drosophila in the lab. And more importantly, most of this media goes to waste. So we were thinking, why not try to see if we can get back that waste? Next, please. So the student had an experimental design that involved two steps. First is to grow Drosophila as usual, using the Drosophila media. And this is the preparation of the media of Drosophila. The second is to grow black soldier fly in 3 different diets. The control, which is the Gainesville diet. The fresh Drosophila media, not processed by Drosophila. And then Drosophila media that was processed by Drosophila. And then she measured different traits related to larvae and adults. Next, please. So larvae attained significantly larger peak weights and adult sizes when fed with Drosophila media, and this is something that we did not expect. Development time was shorter on the control, so the control, they developed to adults faster, but the fresh and processed Drosophila media supported comparable larval growth. There was no significant difference, which indicate that the nutritional integrity is maintained after Drosophila rearing. Next, please. So adult dry weight was higher in Drosophila media than the control, which is also really interesting in this case. But no major difference in adult lifespan was recorded across treatments, except that males survived longer in the control than the other treatments. Next, please. And so we were able to show that the black soldier fly, they can valorize biomass, they can valorize the waste from Drosophila media into valuable biomass. And this reduced the disposal needs. The nutritional value of Drosophila media remains adequate. It did not really differ between processed and fresh media, which is also interesting, which means that wherever is thrown, it still has value. And it's important to start to think about integrating black soldier fly bioconversion in research facilities to have a more sustainable waste management practices. Next, please. And so talking about the application, how we can use black soldier fly. So as an ecologist, I've been working on aquatic insects for a long time. And in order to do experiments in the lab, one of the most limiting factor for us is to find a prey that you can provide to those insects, which are mostly, a lot of them are predators. And so here I tested whether the black soldier fly can be a good feed for aquatic insects. Next, please. So here we are kind of deviating from the usual use of larvae from livestock to a new user or consumer, which is the aquatic insects. Next, please. And one of the reasons why the black soldier fly can be a great food source for many insect groups is that its body size overlaps with a wide range of species from different families and orders. So it is a potential good prey for different types of insects. Next, please. And so we used 9 different aquatic insects with different feeding modes and found that they all consume black soldier fly, and they can survive on a strict black soldier fly diet for several weeks. And because black soldier fly is easy to grow, so that showed us that, yes, we can use black soldier fly to grow aquatic insects. Next, please. And what was really surprising when we look at the behavior of black soldier fly underwater is that they keep moving underwater for a long time. We are talking about hours to days underwater moving. And that makes them a very easy prey to detect by predators. So that was one of the features why they made them even more attractive as a food source. They can survive underwater for a long time, even though they are terrestrial, but they can't really escape, but they keep moving. Next, please. So about these new applications, yeah, you can use black soldier fly larvae to grow aquatic insects if you want to do an experiment in the lab, if you want to grow different types of insects, whether it's a dragonfly, predatory beetles, hemipterans, you can do that. Next, please. So this leads me to the project of Carlos Lopez, my current master's student, who was interested in using black soldier fly as a sustainable solution to compost the Canada goose feces. Next, please. So Canada goose is one of the most abundant birds in North America. You can see it everywhere, in urban areas, in wild areas. Next, please. The species was introduced in other regions worldwide, including Europe and New Zealand, but it's native to North America. Next, please. Although they are currently widespread, historically they were overhunted. Their number declined by a lot, but during the past few decades, we have seen an explosion of the population of Canada goose, which seem to not have really a natural predator. So they just took over, especially urban parks. Next, please. And so you often see in the news reports of nuisance, because when they occupy an area, what you see is like large number of birds producing a large number of feces. So whenever you go here in Canada to a park, you always have to be careful to sit on a Canada goose feces. That's very common. And so, yes, there is ecological, sometimes health issues related to that, and management challenges. Next, please. And so my student was thinking about the question of, can black soldier fly bioconvert geese feces, similar to what we have seen with food waste? Next, please. And it's not new, because studies have shown that the insect can decompose pig manure, cow, chicken manure, etc. Next, please. So first thing we did, we visited different urban parks in Quebec and Ontario. Next, please. And then each time we visited an urban park, we counted the number of Canada geese, but also the number of droppings across a transect. And we found a positive correlation, which means the number of droppings is really correlated with the number of Canada geese. And because they are, we see them in large numbers, so you can imagine and extrapolate how many dropping you can have in a meter square. Next, please. So we brought some dropping to the lab to perform an experiment. And the first objective was to test if black soldier fly is able to develop on a goose feces diet. The second is to test if the frass, which is the excrement coming from this bioconversion of feces, can be used as fertilizers for plants. Next, please. So in the first experiment, what we did is we increased the content of feces in the control diet, and we assessed the performance of black soldier fly based on larvae and adult traits, as usual. And in the second experiment, which doesn't appear here because it's behind a, but I'm going to explain it. The second experiment, we increased the concentration of frass in the media and assessed the growth performance of duckweed, as in the previous experiment. Next, please. So the results show that larvae were able to develop in all treatments, including the pure feces diet. The diet did not have a major effect on development time, although pure feces diet tended to delay development for females. The larvae developing in pure feces emerged with lower weights, and the adults had shorter lifespan. So again, here, we are seeing that the black soldier fly can do the job, but then the diet is not optimal for them. But they can still be operational and bioconvert the media. Next, please. So the results relate to the growth rate of black soldier fly showed that larvae grew slower in pure feces diet. Larvae had similar growth rate in the control and mixture diet. And larvae had lower survival probability in pure feces. And the last two traits, again, we found lower consumption and waste reduction index in pure feces. And so overall, the performance is not optimal. Next, please. Now, when it comes to duckweed growth, next, please. The duckweed grew faster in the frass treatment than the pure feces, like fresh feces that was not processed by black soldier fly, or the control treatment, the Hoagland, right? So here again, we see a scenario where there is added value of the black soldier fly degrading the media, and so providing more nutrients for the plant. So faster growth rate that exceeded that of the Hoagland's media. Next, please. So here, after 28 days of growth, you can see like the red rectangle here, it shows the different treatments, different concentrations of frass treatments. And they outperformed the Hoagland media in this case. Hoagland is the B, and also the fresh frass. Next, please. So when we look at the root length of the duckweed, we found that they were longest in the control media and shortest in the frass media. And this is typical. This result suggests that nutrients are easily available in the frass media. So the plant doesn't have to develop long roots to access nutrients. And in the control, it's the opposite. They have longer root because nutrients are not as available or easily available, accessible to the plant. And this has been shown in field study, experimental studies manipulating nutrient concentrations. But here what we are seeing is like the black soldier fly providing more nutrients to the media, which is pretty remarkable. Next, please.
Please, Dr. Khalifa, you overpassed your time, so can you move to the conclusion, please?
Yeah, this is the conclusion. So the fecal waste from overabundant wildlife like the geese create major ecological and health and management challenges. This study expanded insect bioconversion research to include wild bird feces, largely neglected organic waste. And as we've seen, black soldier fly larvae are able to develop on this media. And the frass derived from this operation or process is valuable as plant fertilizers. And it's another example of a circular economy. Thank you for your attention, and thanks to all the organizers of this interesting conference, and thanks to the lady who is pushing next time, I say.
You're welcome.
And thank you all for your attention.
You can open the debate now.
So we move to the debate. We would like to ask Dr. Nasri to join us into the stage. And we'll take the question from the audience. Dr. Khalifa, please stay with us online. So are there any questions for the two communicators?
So, thank you, Dr. Nasri, for that innovative insight into how model training and artificial intelligence, exactly YOLO V8, can transform modern agriculture. So I have a question. So could your approach be extended to monitor crop health, disease detection, or yield forecasting in real time? This is the first one. The second question, in your opinion, how can we make AI-based agriculture accessible and affordable for smallholder farmers?
Thanks for your interesting question. For the first question about implementing artificial intelligence in the domain of detection of disease, there is some contribution in this topic. We work on detecting disease on the planets of potato also. There is also some contributions using artificial intelligence to detect disease from the biological biomarkers also. So before planting the products or the agriculture products, we can detect if it can be produce diseased products or not.
That was for the first one, and the second. How can we make this AI-based agriculture accessible and affordable for smallholder farmers?
We can deploy this artificial based system on a specialized application like Android application or website application also, web applications. So the customer can access it using access to this application through these apps and use this model deployed in this application. So artificial intelligence is not an application. It's a service. We can use it as a service, so we can deploy inner applications. Not it's an application that have permissions or etc. No, it's just a tool to modeling or to do some tasks that before they hard to compete with manual or simple programming languages. So AI using data to performing these tasks.
Thank you for your participation.
Other questions for our communicator from the audience? Yes, we have a question here, please.
Thank you both for your great presentation. I have a question for Dr. Khalifa, and specifically on the part of the feces from the Canada geese. I was wondering to know if what are your ideas on the field in situ application of your strategies to mitigate, to manage the Canada geese feces?
We are really sorry we missed connection with him. He can't hear us. Thank you. So.
Other question for Dr. Nasri, du coup? No? So I have a question for you, please. As if I really understood your presentation was about detection, and the comparison was only with the human eyes, I think, the kind of thing. Yes. Okay, and do you have like a rate with other parameters, not only what we see? Do you have a rate of efficiency of your application using other parameters? For example, I think that your application cannot see the texture or the smell, only the shape, I think.
Yeah.
Is there a rate of efficiency without these other parameters, or do you have information?
Yeah, our application now working, or our model now working only on this feature that we are training on it, which are the surface, the shape, and the opening eyes. We can train our model on other features, as we see in the previous presentation. The task before we demanded from the AI, we should train in the AI model on these tasks. We give some data that present these tasks. Then we give other images or other samples that never seen, and he will detect this the right class. Will make the right decision. Will make the right decision without a programming code or or help from programmers. So the AI alone detects the right decision. But before we demand from the AI doing this task, we should training this AI on these tasks.
It's like a tool that you can train in order to have other capacities.
AI also during about training.
Okay.
We should training the model before demand these tasks from it.
So I mean, you can even extend it to other types of fruits or vegetables. I mean.
Yeah, this is possible, but the hard task is collecting data. Yeah, the data is the core of AI models. If you haven't if you have not, or if you haven't the right quantity or the quantity that make the AI possible to learn these tasks, so the AI can make some wrong decision. Okay. So the data is the core of the AI models.
Thank you for your answer. We can take other questions. Are there any on the audience?
Thanks.
Okay, so we came to the end of this session. We invite you to take a well-deserved coffee break, and we will see you tomorrow.